year for life sciences and a few appreciation. So to be one among the 15 in the country is a great privilege, and I'm sure many of you who are going to present today will have that joy. The second thing I want to talk about her is that what the case study that she is going to share with us. And when I read it in the morning, 6 o'clock in the morning when I got up, and I realized that, my goodness, this is such a great story. I don't want to take away the charm by telling more about it, but it's a story of how an idea in a lab can make a global impact, And I think this is something I would like all of you to feel inspired by. And also, I would request all of you to realize that this kind of solution, this kind of innovations, what is the process she has followed? So I think innovations will be different, but process can be replicated. And I would hope that Dr. Vandana will highlight the process also that she followed, which gave her such a huge response in more than 55 countries. So Dr. Vandana, the floor is yours.
Thank you so much, Professor Gupta. In fact, it's my really honor to be among GYTI awardees. First of all, congratulations to all of them for getting this very, very prestigious award. And I'm sure they are going to make a difference to our nation's economy. Can someone confirm my
Yes, slide is visible.
Thank you so much.
Yes.
So, in fact, Professor Gupta, I call you are like institution by yourself. I've been a real follower of you even on Facebook. And those of you who have not done it, please do anytime, whenever I have time. I really like to go and visit what he is doing. I mean, that is something which I can't put in words. In fact, I call him Mom. Mom for me is mentor of mentors. And of course, the other part of Mom also, he is really taking care, because as you've seen, if you see his Facebook profile, it's written, he's a show. There are 3 supporter of course, inclusive education, grassroots creativity, frugal innovation, but focuses on empathy. And who else can do that better than a mom? So thanks once again for steering this, for our own India. And let's get on to the case study which we were talking about. It is drug-eluting devices. And I'll take you through ideation and how did we actually transfer the technology. So roadmap, if you need to look at from bench to bedside, it starts with one simple idea. Obviously, once you have idea and you've done certain search on it, you need to protect your IP. And then starts the journey of developing the product. Of course, product development on one side should go on, but it will really not be put to use if you are not studying the market, looking at how you can position yourself, what would be the commercialization strategy, is it feasible to manufacture on large scale? Many times in pharmacy, people forget about feasibility. As a result, even if idea is innovative, it doesn't reach its full potential and is not exploited commercially. So once that is done, of course, you start with prototype development. Stability studies forms a very, very important part here. And often people neglect that because, in country like ours, it has to sustain temperatures as low as 4 to 8 degrees and as high as 45 and above too. So if that is the case, it is really very, very important part. And we need to look at that aspect too. And then either you would start marketing your idea or you would look for technology transfer, a scale up to manufacturing level. So obviously it could be that you would be developing it here in Mumbai, but you need to transfer the technology to manufacturing site, which is in Baddi. I mean, it could be anywhere for that matter. So if that has to be done, of course, the environmental controls play a very, very important role. And then that validation has to be done. And validation, as you know, in pharma world, we say that it's 3 consecutive batches which will be manufactured, and you need to test whether they perform the function what they are supposed to. So that means the quality target product profile is listed beforehand, and then you are checking whether your product is behaving the same way or not. And obviously, if you look on the right-hand side, the graph which is shown, initially when you're filing the patent till commercialization, there'll be a negative cash flow because you are in the development stages, you are registering the product, etc. But when commercialization takes place, there is no looking back. So obviously, it's a very, very steep curve if you've studied the market dynamics well. But it shouldn't happen that as soon as a patent expires, the cash flow starts declining. So you need to re-innovate, and that's what I have written. If you look at the roadmap, you need to re-innovate. That means you need to look at strategy which can make the same product better and better. And you'll see how we have done in this case. Now, to understand stents, let's understand the pathophysiology. What I have shown is a cross-section of arteries, and you'll be surprised till the time the plaque goes to this stage. That means it's actually blocking the artery. More than 70%, it is not clinically apparent. That means there is again innovation awaiting all of you in case you can think of a strategy so that who are the people who would develop this plaque over a period of time can be judged. It would be very, very highly useful. So once, of course, it becomes clinically apparent, you can see that by that time, erosion already has resulted. Thrombosis has taken place, and that can lead to acute myocardial infarction, or as we say, heart attack. It can lead to death too. So it's a real need of the hour that we develop something on the diagnostic side, which can actually check. And I saw, of course, yesterday something where people have looked at cholesterol. Maybe it can be extended to plaque formation too. So more than 7 lakh people worldwide suffer from heart attacks. Within 6 years of heart attack, about 18% of men and 35, nearly double women have another heart attack. 42% of those who have heart attack die from it. And out of the total discharges which happen from hospitals, half the discharged patients are diagnosed with acute coronary syndromes. That means they may be admitted for something else, but when all the testing are done, they are the potential ones who can have these episodes. So of course, earlier what was being done was the bypass surgery, which is open heart surgery, as you know. But now we do have an alternative, which is stents. So you will see what these stents are. They just look like springs, tubular structure, mesh-like structures. But what is actually done is they are mounted on balloons, and through your shoulder or through your thighs, the stent would be taken to the place where the plaque is seen. Balloon is inflated. So balloon will compress the plaque. And then after the stent is placed in place, it would be in fact deflated. Balloon would be deflated and brought back, whereas stent will take its position. So it's a very, very simple thing which is done, but you need to understand that it's a multidisciplinary approach, and you need to understand engineering aspects of it, drug delivery aspects of it. When we talk about biocompatibility, of course, many, many tests which are biotechnology related also creep in picture, and then preclinical and clinical studies. So we started with looking at market growth, and you can see that we started actually this work in 1997. And how did we start? The company who came to us was actually cutting diamonds using laser machine. So they were looking at what other uses these laser machines could have. At that time in pharma, what was prevalent was osmotic tablets, where a small hole is drilled into tablets and the release occurs through that orifice in a very controlled fashion. We thought, let's look at something else. We wanted to give them something else. So after a lot of discussions, they said whether we can enter into devices. Said, you will need a very good, strong team for that. They said, engineers, no problem. Then I said, pharmacy, no problem. I'll handle the total drug delivery part. And that's how, in fact, vision of Dr. Dheeraj, rather Mr. Dheeraj, but I call him doctor because his idea of serving the nation has helped many. More than 6 lakh stents have been implanted now. So you can see it's an ever-growing market, 6.1% CAGR. And in fact, I've also given you a picture of how the prices were before DPCO came into picture. $1,800 per stent, drug-eluting stent. DES stands for drug-eluting stent, and BMS is the bare metallic stents. So $670 went for bare metallic stent. So that means there was actually opportunity waiting for us. And you can see that after 2017, when pricing policy came and there was capping on the prices, how the prices have dropped. And believe me, still there are profit margins which are more than 200%. So if that is the state, we became, in fact, the first CE marked stent in India. And we changed the total scenario. And how you will see, we studied the market dynamics. Of course, a lot of literature survey was done at our end because they were not familiar with respect to drug delivery systems. And in fact, world over, people were using bioerodible polymers to coat drug. We were the first ones who suggested to them that we should go in for biodegradable, because bioerodible polymers in the body finally can create loci for thrombi formation, which also can be fatal. Imagine if there is a locus, there will be blood clot forming around that, and blood clot is the one responsible for many of the infarctions. So after studying what are the opportunities for us, what are the challenges, what could be the restraints, we understood all the market dynamics and moved forward. Now, my first step always, and I tell students that too, is if you're working on a problem, you need to focus on the problem. So understand the pathophysiology of the condition very well. Now, restenosis, you'll see on one hand, it is a wound healing response. Stent would be something which you're putting in the body, it's a foreign body. So in foreign body, always our body will give some response. And what is the response? Initial days, there is inflammation, then smooth muscle cells proliferating, and these smooth muscle cells will go on proliferating to give you extracellular matrix production. And since you have actually compressed the plaque, and you saw in my previous slide that plaque already is bleeding, there are chances again of thrombosis. So that means, though it is a wound healing response, if it goes uncontrolled, it is a problem. It can actually become a clinical complication of stenting. And how? Very simple slide I have depicted. You are putting the stent. You can see what is the light color thing is a plaque. You have inflated the balloon, so plaque is getting compressed. And then stent has taken its position. You are taking away the balloon. You are removing the balloon. But once you have removed, if this restenosis continues, that means the extracellular formation continues, it is again blocked the artery, it is again become narrowed. So we do not want that. And that's the reason why drug has to be coated onto these bare metallic stents, so that it can be kept in this position for all the time stent is there. And especially clinicians said at least for 3 months that is required. So this is a cross section of artery which is in close contact with stent. It could be bare metallic or it could be drug-eluting. So bare metallic stents, of course, it resolved arterial remodeling because it's providing strength to your artery so that blood flow can happen smoothly. But neointimal hyperplasia, that's nothing but extra ECM. That problem was not solved. Neither was instant thrombosis. So we went on to DES, which is drug-eluting stents. By doing that, you can keep ECM formation under control. But instant thrombosis is still an issue. And you'll see how we are solving. The small video which I'm going to play, and I'm hopeful that this will actually show you how did we go about. This video is about Infinium, which was the first drug-coated stent, which is paclitaxel-coated stent. After this, we have introduced about 8 other products in the market. So you can see that if there is artery where there is a plaque, you need to insert on a guiding catheter, which you can see, balloon, which is on the guiding catheter, and then there is stent, which is crimped onto the balloon. So obviously the balloon has to have specific properties. It has to be very, very thin balloon. Polyamide is the preferred one. It has to be flexible because arteries are tortuous. You have to have on one end radio opaque markers so that you can see the position, the clinician who is inserting on CCTV monitors. So these markers, that's another area which requires innovation. Right now we are using platinum, which is very, very expensive. So platinum, radio opaque markers are being seen. Then balloon has to have wings, 3 wings, and have shape folding memory, because only when it reaches a place it should open up. And obviously when you are putting stent onto it, it shouldn't happen that it holds a stent so much that it does not release a stent, then balloon will not come out. Obviously path is tortuous, so maximum flexibility. Now you can see the yellow part or the orange part indicates plaque. Platinum markers have located the position, and obviously now you start inflating the balloon. So it is not pushing the plaque forward, it's just compressing it to the side so that arterial lumen opens up. These stents have to have a specific geometry. So there, of course, role of mechanical engineers is very important. In fact, one thing which they appreciated was, I said, okay, you will expand it. It will expand radially, it will expand longitudinally, but it should have a locking mechanism so that it stays in place. And that's why, again, on the stent design side, some suggestions from our side were really very useful, besides on the DDS side too, drug delivery system side. Now this balloon has to have zero foreshortening. That means it should not go to area where it is not needed. So obviously that is being seen using platinum radio markers, but the geometry of stents has to be such that they don't leave the balloon anytime and move forward too. So there has to be minimum radial recoil. And now you can see one stent has been placed, the balloon is deflated and the guiding catheter goes back from the body. So obviously now these stents are excellent scaffolds. And there has to be again innovation in this happening, because we should have those, they are finally foreign bodies. So you should have very, very low metal. So can we look at metals which can give us thinnest of struts possible? You'll be glad again. India, meaning this company with which I'm working, has the thinnest strut right now, which is 60 micron, and we are moving slowly towards 45 micron. And that would be really, I'm sure, would make a real good headlines in future. So once the balloon is made, obviously you need to coat. So for coating, you need a drug, you need a polymer base. Drug in this case was paclitaxel, because already there were products from other countries like Abbott, J&J, Boston Scientific, which were coating paclitaxel. Paclitaxel, as you all know, is an anti-cancer drug. Comes from, of course, herb, Texas brevifolia. Now, properties of this drug are such that it's very insoluble, but it has good anti-inflammatory property, anti-proliferative action. It prevents a smooth muscle cell migration too, which is a required property. So what we had to do is see that we formulate it in such a way that drug onto the stent remains, but from the stent it goes into the arteries and gets localized there. So this is our own invention. That means we suggested for the very first time world over that we should be using biodegradable polymers. And the initial stent design had 4 layers. But later on, Now we have transitioned to only a 2-layer device. That means we are moving towards thinner and thinner polymeric layers. And our idea is now to remove polymer over a period of time, because whatever some side effects are still seen, they can be looked at. This is how the stent is coated. I had given them just a paper design. Their team was so dynamic that they changed it totally into equipments, and from batch process, now they are going into continuous mode. And this is a release profile which we suggested to them, that initially let there be a burst so that condition can be controlled, and then it can be very, very slow release over a period of time. So that is how, we went ahead and looked at and made drug-eluting stent first of its kind. And obviously, as I said, then there is no looking back. This is a company, Sahajanand Medical Technologies Private Limited. Very recently, in fact, they have taken me on as independent director on their board too. And it is marketing, of course, in India, and is presented now in 75 countries worldwide. Meaning this number only goes on increasing, and we are just hoping to hit US market very, very soon. Now let's look at what are the problem areas which we had to tackle. Obviously, with respect to stents, there are issues. With respect to drug, there are issues. And when you are checking them in biological lumen also, there are issues. So physico-mechanical, drug-related, and biological, all factors had to be seen. Now, drug dosing is very critical. If you load too much of drug, you will not get smooth muscle cells at all. That means healing is not taking place. On the other hand, if you put less of the drug, instant restenosis will happen. So we had to really carefully study and select the dose of the drug which can be coated onto these stents. Second thing, we actually went to cardiologists and those who are doing this kind of intervention, percutaneous intervention, and understood what were the problem areas with other global stents. They said sometimes they do not reach the place, we have to retract them back. That means there is not a flexible coating. Second thing which they said is sometimes patients are coming back within 3 to 6 months with heart attack, which is absolutely not acceptable. So we thought, being drug delivery scientists, let's look at their geometry and look at and try and correlate what kind of release mechanisms are there. So Corners, the company which was marketing Cypher stent, this was the best profile. In fact, we've tried to match this profile. But problem with Cypher stent is that since they had only 2 layers, base coat and top coat, all the drug from base coat over a period of time used to move to top coat, and over a period of time, that means during shelf life, this release profile used to change. So there we had again intervention, and we thought, okay, we can go in for, as I mentioned, initially 4 layers, and now we have actually mastered the technique, and depending on drugs' physicochemical properties, we are able to tell them what proportion of polymer to use and which polymer to use too. Other very important stent which was there in the market was Texas. Now, all people always think if it is something which has to go in market, go into the body first, has to be silicone. Why? Silicone elastomers, as you know, are biocompatible. So same thing Texas thought. So they coated it with silicone, as a result of which the drug was not coming out. And if there was some crack in this stent, there used to be a burst. Remember, these drugs are cytotoxic. We want absolutely control release. Otherwise, they themselves can have hundreds of adverse effects. People had also worked on stents in a way that in the pockets, they created pockets into stents and loaded drug polymer matrix into it. Of course, it could not sustain for a long period of time. And people, just to be innovative, thought, why coat the outer surface? We'll coat only the inner surface. But it was also giving erratic. So we came up, as I said, with biodegradable polymers, thinking that bioerodible polymers will definitely create rough surfaces. And those rough surfaces can be loci for thrombus generation. And this idea, in fact, You'll not believe now world over people are going towards biodegradable polymer. But we took the lead. So when we started, of course, coating, there was another challenge. Now this is metallic and we are trying to coat some polymer onto metallic surface. What is a guarantee that this polymer will stick onto the stent surface? Now here, engineers could not help them, but our literature survey done by our students helped. And we indicated that maybe you will have to electro-polish the surface. And of course, for electro-polishing, they went to Engineering Institute and did that, and then optimized the methodology. But again, the idea came from our end. And after that, of course, coating was done. Next challenge was, okay, you've coated, but how do you study? Whether it is releasing over a period of time, can you study it for 2 years, 3 years? No, that means you have to come up with the technology which can in some time give you idea how these stents are going to behave over a period of time. So this also we designed, we are now using type 4 method, and we suggested that they could use porcine serum instead of the regular PBS, that's phosphate buffered saline. They could use porcine serum, and months data can be generated in weeks time frame. Obviously, simultaneously work was going on. Animal studies they had planned and had to be done because in vitro, in vivo correlation needs to be established. Other challenge was sterilization. Now they are going inside the body. So, but if you do not sterilize, that's not acceptable. If you sterilize using autoclaving, everything fails. Dry heat, everything fails. So we had to look at what could be mode of sterilization. So here ethylene oxide sterilization helped. But of course, you know that when you're using ethylene oxide, no residue of ethylene oxide should remain. So they had to give post-sterilization, also keep it for some period of time before they could release. So there again is a challenge. Again, we are looking at whether we can use flash sterilization. Now plasma sterilization has come in. So there are challenges of making even those devices which can sterilize such kind of interventional devices. That means equipments which can actually sterilize such kind of devices. That's also a challenge. We went ahead, did biocompatibility study. There should not be any adhesion, be it of platelet or of leukocytes. And then we were ready to do study in pigs. Now this has to be done in pigs, cannot be done in smaller animals. You have to use mammals to get the fair idea because arteries have to be really the kind of arteries which we have in humans. So this study, of course, was outsourced and it was done, you can see in Yorkshire swine. So obviously all the studies are done in Europe. What you need to look at is a late loss. Late loss indicates that vessel diameter is as when you had opened it. So that means it has to be near zero. And in all cases, you can see that it is maintained. Now, what I had told you earlier, if you use BMS, no drug, such is a kind of extracellular matrix. It just goes on. If you use only drug, you can see the incomplete formation of ECM. So these are the actual, pictures from porcine. And of course, when you have infinium coated stent, which has, of course, both drug and polymer, this is maintained. So that means there is good degree of endothelization, thin in neointima. You do not have too much of ECM, no instant thrombosis. So after this, we went into humans. And of course, in humans, by that time, we had coated even sirolimus as a drug. So matrix is only bare metallic, infinium is paclitaxel, and supralimus is sirolimus coated stent. So this study was done, of course, 9-month angiogenic follow-up. Now we have data even of 2 years, which I'll be showing. Of course, any such study, you have to have baseline characteristics same. There'll be generally diabetic population, those who are hypertensive, they are the ones who are more prone to such angioplasty, has to be done in them. So baseline characters were same. Look at the clinical outcome after 12 months. I would like you to focus on TVR, target vessel revascularization, because we do not want after stenting to target vessel to become smaller and smaller. So you can see in the bare metallic, it was 17.6, whereas it reduced considerably in drug-coated. So that means drug-eluting stents were giving better protection. After 36 months, also the same story. So, but after 36 months, it was very clear that sirolimus was giving us much, much better protection as compared to paclitaxel. These are the pre-procedure shoot, and you can see a constriction here. So that means this is a place where stent needs to be implanted. And after the stent is implanted, it opens up. You can see a mesh-like structure. It opens up. But idea is after 1 year or 2 years, whether it remains in place, whether this again becomes narrower. So that doesn't happen. You can see that this is a post-procedure shoot immediately after post-procedure. This is follow-up at 6 months, and whatever I'm showing are of the same patient. And obviously, this went on for 2 years. And as I said, now we have very thin strut S-link cobalt chromium. Initially, we started with stainless steel. We have gone to cobalt chromium, another opportunity waiting for all of you. You can look at metals which can give thinner and thinner struts. I told you now we have gone from 60 to 45, but if we can do something with 30, it definitely would be great. So we are moving slowly towards that. And this trial actually came in all the newspapers because what we did is compared our Supraflex with everolimus eluting coronary stent, which was of Abbott. Now this uses biostable polymer, 81 micrometer thickness. And I told you, we are the thinnest at present, 60 micron, ultra thin. We are calling it ultra thin. I don't know after we have 30, what will we call it? So talent trial results also indicated that obviously you can see here myocardial infarction rate is much less, as well as TLR, that is clinically indicated target lesion revascularization, is also less with Supraflex, which is our own Indian make stent. 2-year outcome again has shown that we are better. So obviously in all the parameters, if you look at all the parameters, we are actually better than the Abbott stent. So this came and became really very popular. So that was in controlled population. But now what happens in real world experience? People have kidney disease, maybe having liver disease along with hypertension. So what happens there? So there also, when the study was done, it gave sustained clinical safety, low rates of restenosis, no instant thrombosis, and of course, up to 3 years after a Supraflex was implanted, the data is with us. This is a status. Now SMT holds about 61% share in India. Many people left SMT and started their own company. So obviously, we are moving ahead with innovation in that direction too. Therefore, they are not able to compete because they are right at stage 1, 4 layers stent. And then 20% share worldwide, which I hope, and I am really sure that it will increase. As a result of which, the company has got many awards, be it from Frost and Sullivan or National Award or maybe Ramkrishna Bajaj Award and many others. Now, this is an unmet challenge. What if you have such legends? And what if you have peripheral arterial disease? So I'll be glad that now our Prime Minister Fellow, which I have just appointed, will be working on this problem. What is necessary here? To reach such diabetic wound, you require absolutely ultra thin struts. So nitinol is one metal which can be used, but you can think of other metals, other polymers, which you can actually make use of and make such stents, which will really help many population, because you know that if such a condition happens, many times you need to amputate. So basically, that needs to be prevented. And not only in this, it can be used in biliary duct, you can use in esophagus. My son's a surgeon, he says you are using stents even in kidney. So there are many challenges. Brain aneurysm happen, you would have heard that people have aneurysm. So can we not design stents for brain? I mean, challenging, but nothing is impossible. So this is the roadmap which I showed you in the beginning. I'm again showing you to show that what was our idea. we said that we will use biodegradable polymers and we will make drug-eluting stents by putting drug in either micro or nano form. So by this time, whatever I've shown is your micro form, IP protection. Of course, Indian, we have to do first, but we have IP in place, be it US, be it European. In fact, for any technical discussion, I had to go because they did not have a single drug delivery scientist in the full company. Now this company, of course, is in Surat, has 2 plants in Surat itself, and one is coming up in Hyderabad now, a very big plant. So our development strategy, of course, we looked at feasibility and saw that there was a high market potential. Then we developed key features was, we looked at very aptly at which polymers should go in. What could be our coating methodology? I told you I gave them paper drawing. I still remember, and they have preserved that paper drawing. I said, this is how we coat tablets. For tablets, we, those of you who are pharmacists will know that you are rolling the tablets and the coating gun is fixed at a distance from these tablets. So same way, I said, you can fix the coating gun and you can rotate the cylinder. So obviously we had to design in such a way that we can hold and still rotate the stent without any issues, and we could do that. We worked on in vitro release. We worked on in vivo testing. So net result was, of course, it was a scalable technology. We could commercialize it. And we had to work really very, very close to them. And believe me, every week, either their people used to be in our premises or we used to be in their premises. And then, of course, there's a multiple product line. So we went on re-innovating. So what are we doing now? Now we are loading nanoparticles onto this. Why nanoparticles? Because we want to reduce whatever you have seen, as 1.2% death. Why should we have one death, single death? So that needs to be taken care. And one thing which others have not done, and I have seen, I've read many patents, they make nanoparticles and finally they dissolve nanoparticles in a solvent and coat. Where is the integrity of nanoparticles there? So what we had to do is look at coating, and after coating, see whether they remain in nano. And you can see what I have given here. We scratch it from the stent surface and then again do SEM to show that it is really nano. Because why are we making nano? If it is in nano, obviously it will permeate on the artery side. It will go to intima, media, adventitia layers and get deposited there. And from there, for a long time, it can offer protection. So this is what we showed in vivo actually to them, in vitro to them in cell line studies, that yes, it is getting deposited into the arteries. And then we came up with the ex vivo permeation assembly. First time we have designed this assembly. Before going to pigs, they can do it in this assembly. So it's a simple, tube in which aortic tissue is mounted. And then with the help of peristaltic pump, we are mounting the stent. After mounting, after a few hours, we'll be taking readings, HPLC readings as to how much has gone to the tissue side, how much is onto the stent surface, and so on. And this has given them really good indication. And now we have gone even to next step, wherein we are actually coating 2 drugs. Why 2 drugs? Because you saw that one drug will take care of SMCs, it's an anti-proliferative drug. But what about thrombosis? Can we see that the clopidogrel or aspirin, which people have to take lifelong as blood thinners, need not be given? Answer is yes. So can we not incorporate other drugs? We can. So this is what we have already done, actually published also now, wherein we showed that you are getting really good prolongation of coagulation time over a period of time. Not only that, in fact, now we are moving a step ahead and designing polymers which can give anti-thrombotic action. So that definitely is another platform. So you need to work at different levels very closely. And I really believe what Mr. KG Ananthakrishnan said, and what in fact GYTI is doing, working very closely with students, that means academia, government partnership, BIRAC has come in, They are trying to identify industry partners for you. And I think with this, I will end. But these are some of the points which we need to ponder if you are doing translational research in India. So do we create a sound hypothesis before we undertake any research? In fact, many of us do not do market survey. That needs to be done, identify good unmet needs. That's the reason why 3 of our students have got, GYTI award, because they understand what they are doing. Okay, you have come out with a solution, but are you looking at whether this drug data which you have created is authentic, reliable, reproducible? Because data integrity on one side and reproducibility on other side is very important if you are thinking of commercializing or if you are thinking of technology transfer. Many times we do not have predicted preclinical models. People can open, I really believe we hardly have any BCS class, class 3 facilities for animal testing. There's only one facility in India to do such kind of studies. So there again, our entrepreneurship is waiting for you. If someone has space, can get all the expertise and can start this. Other thing is, many times we take up industrial projects and give it to students to work, but how do we maintain confidentiality of data? So maybe you can have internal CDA signed with students so that they understand. On the institute sides, we are modifying rules, rewriting rules every time. We do not have ready drafts of confidentiality, milestone payments, royalty. I have faced each of this even at our institute, so I can imagine what is happening in other institutes. So all these need to be really looked at. And of course, can we dissolve our big ego? And think that's what GYTI is doing, trying to bring all of you together in a way that such a conducive environment is generated that all work with one goal. And that's how our own India, can become, obviously, it is world leader, but can still become better and better, and we can cater to needs of all. So opportunities, dear students, are right there. Key is to grab it. And this is the quote which each of my students actually reads, because we say this is our mission. So focus. Nutshell, it just means once you have started working on an idea, please focus. These are many of the technology platforms which we have. About 20 technologies have been transferred from our lab to industry. And of course, I take pride in saying that it's all because of these dear students. When I started, I didn't have a magnetic stirrer also. Let me tell you, Udacity gave me some seed money of 10,000 rupees. But over a period of time, all the facilities, whatever you need, have been generated within the lab. Of course, ICT by itself has many labs. And without collaborators and funding agencies, nothing is possible. You'll see, I have put many manufacturers who are from UK. So we are working very closely with UK, maybe Europe, many places. In fact, many of the technologies during COVID times have been transferred. I could identify students who are in Ireland, send them to the company premises because Mumbai was totally closed. And giving direction from here, they could actually make a veterinary product, which is for foals. So, I mean, such is the capability of students. So we have to take the best out of them. And we are lucky at ICT to get such students. I'm so sorry. I didn't realize it's 11:47.
No problem. We will extend the point. We have some flexibility. So instead of 12:40, we will finish at 1 o'clock. So 20 minutes for Tasleem are still intact. Not a problem. But what you have told us, 2 stories that you have told us are very inspiring. So I will take 1 or 2 minutes. If anybody wants to ask any question, any student wants to ask any question, any clarification, please go ahead. We will leave 2 minutes, 3 minutes for questions. Please go ahead. Anybody? You can, of course, put it on the chat also, but I would wish if you want to ask a question. Yes, Amar.
Good morning, ma'am. It was a very nice presentation. had a lot of informations from your slides. I would like to know that you said you are spraying the stent with a drug, right? Putting the drug on the outer side or from the inner side?
So we are coating right now on all the sides. We
All
have
the
not restricted. It's on outer as well as inner side.
Okay. So if you're coating on the inner side, that drug will be released into the bloodstream, right? On the outer side, it will be released into the tissues. On the inner side, it will be released into the bloodstream.
Right. That's
So
correct.
what is the life of the shelf life of this drug? How long it stays in the body?
No, so please try and remember that though these drugs, as I said, are either immunomodulators or they are cytotoxic, the dose at which they are used is 1/100 of the usual dose. All we want to do is stop smooth muscle cell proliferation. So we do a lot of studies to arrive at a critical dose mass, and these drugs will be out of the body. The shelf life of the drugs which I have quoted right now, I have quoted paclitaxel, sirolimus, and everolimus. So they are out on, meaning if they go into the bloodstream, they'll be out in days' time.
Okay. Thank you.
Thank you very much. Anybody else has any comment or question? All right. Thank you, Dr. Vandana. Please stay on for a while so that you can comment on the presentation by the GYTI awardees also. But I'm really grateful for the huge impact that you have had. And at some stage, we are going to get you to write all of that in a manner that, apart from the technology, the institutional processes that you had, you mentioned you began with a 10,000 rupees grant. I think that's how the story should begin. That when people look for resources first and innovations later, they are doing your biggest mistake of their life.
Absolutely.
What you have done is you have talked about innovation first and resources later. And I would like everybody to note this point. This is what the story of Honeybee Network is. We had nothing with us. For 4.5 years, I managed it through my consultancy income, which I diverted toward this network. That's how we managed. There was no funding. We didn't begin with funding. We began with an idea, and that's how everything happened. So I think what you have said today is it resonates so well with me, because that's the spirit of our country. We should not look for resources. We should not think of big ideas only when resources are there. In fact, when resources are less, your ideas are bigger. There is a view we use in science policy research. We talk about tool view of science. We get constrained by the tools we have, the big machines that we have. We see what can be done with these machines, not what we don't ask what we need to do. What you did was you asked what needs to be done. And if machines are there, all right. If they were not there, you might have even maybe redesigned some of them. I mean, that is something we have to hear in the next story. Some other time, you should tell us what modifications you made in the lab equipment itself. I'm sure you would have done that. So just one small example, if you can give us, and then we move to the slate.
Yeah, I will tell you, sir, when we actually started, of course, coating was a far-fetched idea at that time. So whether my drug and polymer actually gets coated on the stent, we were dipping the solution to a certain length, a test tube, keeping it for some time and then taking it out, and then looking under microscope whether we had to put a stain in the a dye to see whether there's a uniform coating or not. So with the help of magnifying glass, what we do today with SEMs and TEMs, we used to actually do it with magnifying glass at that time. Sir, we didn't have even HPLC. We had only one in ICT, our own department. So I used to coat, and we had a nearby analytical lab. So I struck a deal with them, and it used to go for analysis there. Because by that time, even company with whom I was working, they were novice in all ways. So they didn't have anything. So many things, but I thought I'll give you
Thank you so much. Thank you so much. Taslim Arif, he has championed an extraordinary platform, CCAM and Index, which I have been listening to the meetings every Saturday more or less regularly. I'm so inspired by what CCAM has done, particularly in diagnostic area, in building a platform for testing, certifying, helping in regulations, arranging distributors, whole chain of support system that CCAP and its different platforms have done for the startups and biotech entrepreneurs in our country. So I would request all the GYTI bodies to know about that, use their facilities. Taslim, floor is yours. Thank you so much, Vandana.
Thank you, Professor Gupta. Good morning, everyone. And Dr. Vandana, phenomenal kudos to you for this exciting work. I had known about the work, but never heard you directly. I think it was a great opportunity, thanks to Professor Gupta, to learn this story. These stories are rare and need to be told again and again to excite small people like me. I think, thank you for encouraging. Thank you. Excellent work. And thanks, Professor Gupta. And this is, I mean, literally, this is what science is all about. So thank you. I think, Professor Gupta, we have 10-15 minutes. I'll try to just give the
You can take
glimpse.
20 minutes, not a problem. You take 20 minutes, not a problem. We will extend the session, not a problem.
I'll try to be as brief as possible, sir. I will do that. Very quickly, just want to share with them what it is about, just to introduce myself. Friends, I'm a neuroscientist by training. I have at my PhD understanding how neurons talk to each other, and at Max Planck Institute of Brain Research, and then did my postdoctoral work at University of California, San Francisco. And then I have been interested in translating scientific knowledge into impactful innovation. That has been my larger interest, and something that I get an opportunity to do that at CCAM, something closer to what Professor Gupta mentioned just now. So I'll give it a glimpse of it in a way to you today, and happy to take questions and so on. My email ID is written, so for any questions that I may not be able to answer or something that I can add to, I'm happy to expand on it. We all know about scientific entrepreneurship. You all are, in a way, building those things with you. But as we all scientists dream, and you just heard from Dr. Vandana, clearly, I think scientific knowledge reaches its maximum impact when it solves society. And I think we have seen how it gets done and how translation gets done. And something that we are creating a platform, a kind of a innovation hub where deep scientific ideas can be nurtured. Not all ideas will be always successful. We all know that, and we must know that. But there is a fun in journey. As a scientist, when you actually attempt to do something, as Dr. Vandana mentioned, can we have stents for brain, arteries going there and all that? Sounds like, very futuristic, but that's the fun part of it. Even if we fail, we attempted something that was challenging. And that's where the journey's fun comes around. So that's such ideas and such journeys are something that we want to support. So if you have ideas that you think you want to get supported for further on, we will be very happy to do that. Just for the context, CCAMP is based out of Bangalore Life Science Cluster, which also has INSTEM and NCBS. Together, we form a community of around 1,500 to 1,600 scientific researchers and support staff, which aims to build science, translate and build innovation and help commercialization of scientific knowledge. And we are hoping that this cluster phenomena will be the new way how thriving scientific and innovation ecosystems will be built in India. This is where CCAMP is. We have largely two verticals. I'll not go into them. One is technology platforms, one is entrepreneurship programs. And in technology platforms, we have, as one would say, who is who, and one can say what is what, whatever. In terms of high-end bioanalytical capabilities, in terms of instrumentation capability as built in here, top of the notch, absolutely global standards, and with experts who can help you take your research further in microscopy, in flow cytometry, in mass spectrometry, in genomics, in structural biology, transgenic animals, the only place in India which can build transgenic animals using CRISPR-Cas, micro devices, microfluidics, microfabrication, and so on. So this is really something where we aim to ensure that give those high-end capability platforms for you to utilize them and go further. And this is where all of the work has been done across India. In terms of entrepreneurship, we have different capabilities starting from funding, incubation, mentorship, acceleration. More importantly, these are the tools, and one cannot be really, happy about what tools they've created, but what have they done with the tools is more important, as we all can believe, right? So what we have done by building capabilities is we have now around our own funded 250 organizations in life sciences, biological sciences, in healthcare, agri, animal husbandry, and environment. And some of them are definitely, we are very, very proud of in terms of the kind of science they are doing. They are absolutely globally challenging, top notch in terms of what they have done. Yesterday, one of our companies, Bugworks, published a paper in Nature Communications. You can imagine the kind of science they are doing in terms of building new antibiotics against antimicrobial resistance. We have a company like Koyo Labs, which has actually built 2 products from regulatory approved, including US FDA for respiratory support, including COVID-19 and so on. And those are in market and doing phenomenal job across India. Pandorum is doing stem cell work for mini liver as well as corneal transplant as we speak. I'll not go into details of it, but I just want to give the flavor of it, how each story sounds phenomenal, but that has been a persistence, as Professor Gupta mentioned. Find the cause first and then go to means. Don't go to means and then find a cause. So I think the cause is very important. If you have something that you want to do, you can build the means around it. So, and if we are all here, across this to help you support your cause in terms of building innovations for the impact that you want to make together. There have been work in terms of string bio, which is using synthetic biology, using microorganisms to convert global warming gas like methane into non-human food and futuristically other capabilities as well. There are many more. iStem, which is using stem cell therapy for vision disorders, including genetic disorders like retinitis pigmentosa and also age-related macular degeneration. There's really this is challenging healthcare, challenging agriculture, challenging environmental problems at the global level, and how we can do that. 66 Energy uses algae as a new way into looking at for agricultural enhancement in terms of crop production, and their technology has been already licensed or marketed by Mahindra and Mahindra. There are many, many more such examples. I won't really take too much of time, but just to give you a perspective that this is really what we want to do is that takes innovators like you and support them further. As you can see, there are many more. We already have at least 25 to 30 products in market serving people as we speak across the healthcare domain, agriculture domain, environmental domain, and so on. And many more in the making, because life science, it takes longer time. So many more are towards phase 1, many more are doing their clinical studies, many more are building their understanding. There are work in terms of, as one would call it, for precision medicine in cancer. There is work in terms of the, I think last slide you saw, so Yostra Lab has 2 products in a way in market, one for diabetic neuropathy detection, and the second one is for treating diabetic foot ulcer using warm oxygen therapy that they have built and have shown phenomenal data out of it. Hanujan is working for Duchenne's muscular dystrophy using single nucleotide gene therapy approach change, and they are getting into the phase 2 trial of that. SpotSense is building what one would call it as a neonatal saliva-based biomarkers for understanding neonatal infection and neonatal sepsis. So these are really top-notch, excellent work, and what we are doing with the tools that I mentioned is that identify those good ideas, nurture those good ideas, bring the environment that can help these good ideas and take it forward step by step across the value chain of the journey of the innovation. And those numbers continue. We have dedicated focus in maternal child health. The reason we think is that India needs more and more innovation in maternal child health, so both from the mother perspective as well as the neonatal perspective. We will do it. We all know about the birth asphyxia and how many deaths we actually see in terms of neonates. So we have a product which is now going to be launched in a month for neonatal birth asphyxia. Really amazing work. We are looking at a product that will come out very soon for minimal invasive uterine fibroid surgery for women, which is a major cause in terms of their health issue and so on, and many other diagnostics and monitoring capabilities we have. Of course, agriculture is one important aspect, as well as the antimicrobial resistance we all know about, from the prevention capability, diagnostic, rapid detection, and therapeutics, both in terms of the antibiotics and phage therapy, as one calls it. This is a larger number and older slide, but something that Professor Gupta mentioned about. Last year, we all realized that there was a challenge in terms of healthcare tools, if I may use the same word again. We were not having enough tools. We were largely always relied on international import from collaborators or other countries. But when things really got challenging, we didn't have test kits, we didn't have other things. And at that time, we thought that can we now make a program which will make it as an indigenous diagnostic capacity building program. So with the support of first, we set up NBRIC in May, and then around June, with support, the NBRIC was supported by DBT, and in around June, July, we got support from Rockefeller Foundation to set up index program. Collectively, we have been able to actually bring together the capacity of nearly 800 to 900,000 RT-PCR testing per day capability in terms of manufacturing, with a number of companies across India. And there are many nuances that went into it, but the most important thing, as you heard from Professor Gupta, the cause was important, the means came around, the methods came around. It was not planned from day one. We actually had to work on the ground. The idea was to see how our Indian companies can produce high quality reagents as well as kits at the scale that should be good enough for India and hopefully export beyond. And for that, many scientific, regulatory, and technology collaborations had to happen. And that's what we were able to do it on this platform. Similarly, we had another thing that we worked around on, which we call it CCDA, CCAM, COVID-19 Innovation Deployment Accelerator, launched around 2020, March, April. And we got 1,100 applications for COVID-19 innovations. And out of that, we selected around 30. And out of that, I'm happy to share that 23 of them are in market, which are either in diagnostics, respiratory devices, screening devices, vital parameter monitoring, and preventatives, and so on. And we, not only that, we supported them and nurtured them as a phase 2 of that. Now we have many of this COVID-19 healthcare solutions we have now deployed them in hospitals. And 14 states from CCAM, we have actually worked with a national health mission. We have worked with the state national health missions, district hospitals, pediatric hospitals, and now these innovations are sitting there. This was another challenge, that while supporting is one, how do we actually help them to the next? So that has been done as well. So this was possible, friends, because to build a larger understanding of how the innovations or the good ideas are identified, supported, taken to the, as one called, through the regulatory, and most importantly, deployed further on. So this was an effort towards it. I would say that, as we all, I think this is the previous talk by Dr. Vandana was clear an example of how scientific entrepreneurship is very, very important. And we need to, all of us need to continue to take on good scientific ideas, more innovation bets or more innovation experiments, as we call them. We should do more and more. Even 10% of those after a while become successful. We would be actually doing a phenomenal job in terms of how it has been done. We clearly know how government has been supporting all this for many years now. And because of their support, we have been able to build all that, like DBT, BIRAC, NITI Aayog, DST, and so on. They have been really doing a phenomenal job in supporting that, as well as state governments. For people who are, at the cusp of taking their work forward, I think I was just forward this from my team, that as of today, we have 5 calls for funding open for innovators. One is, deadline is today, that is for biotechnology ignition grant. So, please feel free to apply for that if you have it. Then we have something which we call it as a national bio-entrepreneurship competition with the deadline tomorrow, which might get extended, but this is even for student teams. It is allowed, so look it up, apply for it. We have up to 8 crores of funding, by the VCs, as well as 65 lakhs of funding cash prize available. We have an Agri grant challenge. We have 3 problems in Agri that we want to solve. This is a long, Professor Anil Gupta is on the core committee for that. So we have 3 problems that we identified over last 8 to 9 months, and we want to solve them. So we are looking for that. We have food and nutrition social immersion program, where we are hoping that fellows will go and find problems in terms of food and nutrition around us. And then the last one is we have a Japanese collaboration to fund up to 2.3 crores investment equity funding. So those is also available. So please feel to reach out on the email entrepreneurship@ccam.res.in, of course, or me. I'm happy to take those. But if I may conclude, all this is to ensure that your really brilliant ideas are taken forward after GYTI or in parallel to GYTI. I think this is very, very important thing. We really want to support those ideas, because our hope deep down from all of us is that some of these ideas will actually solve societal problems on the ground and help to take the growth of the society and community further. I will possibly stop here from with my coordinates, the website, the email, the Twitter handles, both for organization as well as for myself, and happy to take any questions. Thank you for patient listening. Thank you.
Wonderful, wonderful. Tasleem, Arif, this is an amazing journey. I know I personally have absorbed a lot of energy from what you have achieved. Now, we are going to have the presentations by the GYTI bodies, but I would like to ask one small question. The kind of support that Index is providing to diagnostic, can that be generalized for other solutions? Is it possible? Are you planning to do that? Because that is what's doing it. Only you can do that.
No, thank you, sir. I think that is actually going to be key, sir. I think this is what was if I may say, that the conversion block between the potential and the reality in the MSME sector. What we did not have is a scientifically oriented infrastructure and validation support. And we badly need it across, as you rightly said, across setup, because the tools are available, but the scientifically oriented, validation centers are not there. UK has done it under the name of catapults, as they call them.
Yes.
And they have done a brilliant job. The Europe has done it under the name of test beds. Like the Sweden and all have a phenomenal big test beds and so on. And I think we need to do that with not only just by setting up machines, but also have scientific teams which can handhold these projects and correct them. It is not about only saying yes or no, but what we did in Index, as you know, sir, is that when they went wrong or was not up to the mark, is to tell them how to correct it and then make sure that they reach the correction mark and then go further. And that is very, very important, sir. And we are putting a couple of proposals for that, sir. We are hoping that something comes through.
You have all my support, because I believe, and I strongly believe, and I want to say that publicly, that what you have achieved, what Lalit and his team has achieved under your guidance, is something that needs to be generalized and made available to all GYTI awardees to begin with.
Absolutely.
And obviously big grantees, because none of them have a platform of the kind that you have built, where whole range of support system, not just, yesterday we had questions about regulatory support. Now you have a lot of consultants who can be mobilized at low cost, maybe sometimes without cost, to these entrepreneurs, these startups. So maybe you should start holding a small part of the equity, maybe 1%, 2%, 3%. I would call it technological equity. I mean, in the sense that for the support that you're providing, have a small equity in the enterprises, so that when they become successful, you recover all your costs. And in that sense, your viability gets linked to their growth. Today, you are not doing it. You're doing it at all pro bono, isn't it?
Correct.
But I will strongly urge that set up a Section 8 company which will hold equity in every startup that you do, voluntarily, of course. It is not mandatory. It is voluntary. But I'm telling you that when I hear many of the startups who have deeply been influenced and helped by the platform, I don't think any one of them will stint in offering a small percentage of equity to you. And with that fund, many of our awardees who are at early stage students, they can't give you anything. Correct. But they can be grateful recipient of your support. And I can tell you that when you have seen GYTI awardees, when you have looked at our platform, you know, some of them are making breakthroughs. We have got 133 publications till 2020 from these people. Some of them in Nature and other such publications. These are brightest of the lot that we have in the country. Every year when we select 15 of them, they are the brightest of the lot. And, you know, all IITs, ISC, JNCSR, all of them compete for these 15 awards. So they are really the choices. And I think if they can get a support, we can work on that jointly. We can provide them institutional support, policy support. We can provide them the technological support, business support. It will transform their journey. They will transform to become more vigorous startups faster than otherwise. So I would strongly urge that.
We'll do so, certainly, sir.
Thank you so much. So we will now start the presentation. Both of you are now in charge, Vandana and Taslim. So you can start inviting. We have got Rohit, Viraj, Shibu, and I think Mani Kuntla is also there, and Harsh. These are the 5 who will like to present. Please start. Please invite them.
We can start with the first candidate, incidentally, my own student.
Yeah, you had one yesterday, you have another today. That's wonderful.
Okay, so should
Rohit,
I start
please
the presentation?
Start. You
Yes,
can
please.
share. Yeah, you can share your presentation.
Am I audible and my screen is visible to everyone?
Yes.
Okay, so good afternoon and namaste to everyone. My name is Rohit Pawar, a PhD Tech Research Scholar and a GYTI awardee working under the guidance of Professor Vandana Patravle at the Department of Pharmaceutical Sciences and Technology of Institute of Chemical Technology, Mumbai. And I'm here to present my work on topic point of care nanodiagnostic kit for brucellosis. So coming to an introduction, brucellosis is a neglected global zoonotic infectious disease that is caused by ingestion of unpasteurized dairy product or direct contact with infected animals. Here, the causative agent is Brucella species, which is a class 3 organism, and CDC has labeled it as highly weaponizable. So these species were weaponized and used as bioweapon during World War II. So you can imagine its severity. The clinical signs and symptoms of brucellosis in animals generally involves abortion, infertility, and low milk production, which keeps no other option in front of farmers than killing their own infected livestock. While clinical signs and symptoms of brucellosis in humans include continuous or intermittent fever, headache, weakness, profuse sweat, chill, joint pain, weight loss, and it creates general malaise, which takes almost a year for a person to revive back. The diagnosis of brucellosis is still based on costly and time-consuming cultural techniques involving serological sample, while treatment, it involves vaccination, antibiotics, and common antibacterial. This slide talks about the market analysis and the statistics. So as you can see, the veterinary diagnostic market at present is about 5 to 5.5 billion US dollar and is expected to shoot up to 9.5 billion by the year 2026. By next year, that is 2022, Asia will be the best and the biggest market to invest in the point of care testing. And at present, India invests a huge amount, that is 1.5 billion US dollar in a diagnostic market, of which 30 million USD is invested only in point of care testing and with a CAGR of 8 to 10%. So this was the problem which we identified, because of which we started working on the diagnosis of brucellosis. So the medical diagnosis of suspected cases in farm animals such as buffalo and cattle is often delayed, as the detection majorly relies upon the previous history of abortion and time-consuming cultural techniques, which continue to persist as a gold standard for the detection of brucellosis. Apart from these techniques, agglutination, complement fixation, rose Bengal test have been developed, but they also suffer from false positive and non-specific result. A major drawback associated with these techniques is that they require blood sample, and hence it subject patient to painful and invasive sample collection procedures. The diagnosis of brucellosis has been achieved using non-serological samples such as milk, using new techniques such as PCR and ELISA with great sensitivity and specificity. But again, these systems, they rely on the availability of skilled personnel, costly immunoreagent, complex instrumentation, and sophisticated testing facility. If you see Indian scenario, India has a huge resource of livestock, and dairy farming plays a substantial role in country's rural economy. India restrains largest buffalo population and second largest cattle population, making its highest milk production country in the world. Brucellosis being highly contagious in many parts of the world, including India, it is responsible for causing significant morbidity and enormous economic loss. If you see the incidences of human brucellosis, they also occurred worldwide, which indicate that this disease is endemic in Mediterranean basin, Mexico, and Asian countries. So by taking all these factors into consideration, we developed a very simple immunochromatographic diagnostic test or a kit, which is based on fluorescence silica nanosensor that could serve as a rapid, point of care, sensitive, specific, and cost-effective tool to analyze non-serological sample. Our diagnostic kit is based on lateral flow assay, which makes use of unique dye-loaded fluorescence silica nanosensors, which are conjugated with lipopolysaccharide. Here, lipopolysaccharide or LPS being the antigenic component of the Brucella species, we extracted it from Brucella species and then we conjugated it with dye-loaded silica nanosensors. So these pictures generally represents the assembly and the working principle of the immunochromatographic diagnostic kit. If you can see here, the kit generally consists of the sample pad. You have the conjugate release matrix, which is coated with the antigenic fluorescence silica nanosensor. You have the nitrocellulose membrane, which consists of the test and the control line, and an absorbent pad. So when a sample comes in contact with this nitrocellulose membrane, it reacts with the test and the control line. And in case of positive sample, both the test and the control line or a spot the fluorescence, while in case of negative sample, only the control line fluorescence upon excitation. This kit, it gives a qualitative result in yes or a no form in less than 15 minutes. We had also filed an Indian patent on this technology, which recently got granted. So this is how our diagnostic kit looks like. So the left-hand side of the picture or the image, it shows the diagnostic kit with the plastic cassette, while the right-hand side, it shows the refill, which consists of the different components. So the developed diagnostic kit, we evaluated in terms of its sensitivity and the specificity using samples from animals and humans. And it was found out that the test results were comparable with that of the currently available test with ICDT showing 100% success rate. So these are some of the factors which makes our diagnostic kit a unique one, such as the currently marketed ELISA-based diagnostic kit, they employ gold nanoparticles for visual detection of color change, which following the antigen-antibody complex reaction. However, they are not only costly, but also time consuming. So the use of silica nanosensors here could be an economic approach. Also, the use of non-invasive or a non-serological sample such as milk, saliva, urine could reduce the overall cost and it could ease the screening procedures. These factors makes our kit a novel one. Also, the currently available diagnostic kit for brucellosis, they rely on the ELISA as their test principle and mostly work on the serological sample. But our kit, since it uses non-serological sample, it could be available at a cost of as low as 200 Indian rupees. Also, our developed diagnostic kit is cost-effective, sensitive, specific, non-invasive. It does not require blood sample, neither it require any advanced facilities. So these are the outcomes which we are expecting out of this project, such as the developed technology is novel. Yes, it is cost-effective yet scalable, and it fits into Make in India initiative to foster the innovation, enhance the skill development, create the intellectual property, and generate revenue. This kit will benefit a large masses of animals and the human population, including the farmers, veterinary doctors, slaughterhouse workers, distributor, wholesaler, and so on. The diagnostic kit with large-scale manufacturing as well as the efficacy data will be the step towards its commercialization. So this is a unique or first of its kind based on the non-serological sample as well as the nanosensors, with which giving the sensitivity and the specificity comparable to that of the PCR as well as the ELISA kits. At the same time, it is affordable to be used by the farmers. So this is our platform technology, which can be applied to diagnosis or detection of numerous diseases. Earlier also we had, and we are also currently writing many proposals to industry as well as to the private or the government grants. And currently, using this platform technology, we are working on the diagnosis of dengue infection as well. Thank you. Thank you so much.
Thank you, Rohit. So if anyone has any questions or suggestions, as Professor Gupta has already written in the chat box, please post or you can ask.
Rohit, how far is it from the go-to-market stage?
Okay, so basically, we are currently working in collaboration with our industry partner for the scale up of this diagnostic kit. And once this is done, it will be commercialized immediately. So we are in that stage of scale up.
Okay, so how long do you think it will take?
Sir, if everything falls well and if the large scale manufacturing, there won't be much hurdle with respect to the large scale manufacturing. Soon it will be commercialized.
Soon
Because,
meaning how soon?
Do you want to be yourself a startup entrepreneur? You will do postdoc and let it be the fruits of your labor will be harvested by some company, isn't it? Is that what you're saying? You don't want to harvest those fruits. No, it doesn't look like. From your facial expression, I can make out.
Initially, when we started, in fact, he was very keen to get into this journey. But as the time passed, we felt that it's now better that we
license it out.
Join hands with industry and license it out.
Okay. Yeah, that makes sense. And in this regard, I would only say that if you wish to explore the startup community, biotech is a startup community, then Index and CTAN has a very large network. So Taslim, if you think that some of these people need to be, and some of these young boys and girls need to be linked with your platform sometime, perhaps we could have a separate presentation by those who want to license their technologies, who are at that stage. We can arrange a presentation by them of 10 minutes each to one of your weekly meetings or something like that.
That would be very good. Absolutely a good idea.
So that we can ask them to diversify. Many they have solved a problem. They have solved the COVID-19 problem. Now we can't stop there. So many of them need other business ideas to expand there. So maybe they will not pay you, Rohit, as much as a big company will pay. But helping our indigenous entrepreneurs to grow big is something that is what nation building is all about.
True, sir.
So I think we should encourage. They may not give you much upfront, but the royalty, if they grow, then you get royalty from there. And I think we should not really go for too much of upfront payment, which obviously many of these companies can't pay because they're also struggling with their own growth problems. But the fact that they have come up this far, we should think about it.
Yes, sir.
Thank you.
Absolutely.
And Taslim, yeah, if you want to say something.
No, absolutely. I think, as you rightly said, sir, some of these are platform technologies, to be honest. Right, platform technologies where you can use the same platform and you change the sensors, you change the detection, the probe and so on, and the rapidness, the affordability, the portability is anyhow built in. So as you rightly said, those are absolutely there, and those can be taken forward. And some of those can be, through the slides presentation or for 5 to 10 minutes, a discussion will be very useful. Absolutely.
Because yesterday we were asking everybody who presented that, what is your platform approach? You have solved a problem, but what is the platform element of your work which can be applied to more than one domain? So that is where we will get real multiplier effect of your work. The spillover effect will be much higher than what Taslim said can be done. So thank you so much.
Thank you so much, sir. Thank you.
Thank you, Rohit. So we can move to the next GYTI awardee of 2019, Biraj Mehta from IIT Hyderabad.
Yes, ma'am. Am I audible?
Yes, you are.
Yeah, I'll just share my screen. Okay, so is presentation mode visible?
Yes, it is.
Yeah. Okay, good afternoon, everyone. So my name is Viraj and I am 4th year PhD student at IIT Hyderabad in Biomedical Engineering Department. We applied with a project called Microfluidic Technology for Combinatory Anti-Cancer Drug Testing. So me and along with Dr. Sharanya and my guide, Dr. Shubhanarayan Rath, applied in 2019. So Sharanya actually completed her PhD in 2019. So since then, I am continuing this work. So I am GYTI 2019 awardee and Prime Minister Research Fellow at IIT Hyderabad. So before going into presentation, I'll just give you a brief idea about this whole project. So we have made a microfluidic device which can test 3 anti-cancer drugs at a time and all the possible combinations in the microfluidic device itself based on tumor spheroids that we culture in microfluidic device. So our ultimate goal is to isolate the primary cells from patient biopsy sample and culture it in our microfluidic device, and can we recognize the most efficient drug combination among the other combinations. So we all know that the combination of anti-cancer drugs can produce better results compared to monodrug therapy with lesser drug toxicity and better outcomes within a short time span. So so this is the basic design of the device. So this was the first prototype with which we applied originally for the award. So as you can see, these 3 drugs have been illustrated with yellow, red, and blue color, drug A, B, and C. And there are 3 pairwise combination possible if we think. Then at these 3 junctions, we can, where these 2 drugs are meeting, here you can see. So these 3 junctions, we can test 3 pairwise combination and in the center, all the 3 drugs are coming together where we can test combination of all the 3 drugs on tumor spheroids. So, so here, as you can see, this whole device we tested with, as a proof of concept, we tested with on A549 lung cancer spheroids with 3 chemotherapy drug, paclitaxel, vinorelbine, and etoposide. Here P is paclitaxel, E is etoposide, and V is vinorelbine. So as you can see, when we tested with all the 3 drugs at a time, we found that within 24 hours, the killing efficiency was the highest among all the other monodrug therapy as well as pairwise combination. So which shows that the combination of the drugs works on our device. So after this, what we did was there were some limitations that we need to overcome with the first design. So here, as you can see, we have just one microwell, means just one spheroid for each drug combination that we can test in one platform. So we have to at least go for another 2 chips for statistically better results. So what we did was we come up with the next new modified design with the same concept, of course. So here what we have done is we have increased the number of wells from 1 to 3. So at least we can get better results within one microfluidic platform. And the same concept we have applied here. Here also the 3 drugs have been illustrated with 3 different colors. And here at 3 junctions, we can test pairwise combination. And in the center, we can test combination with all the 3 drugs. Moreover, here we have included serpentine loops to pre-mix the drugs before they reach to the cellular area where the spheroids are cultured in the microwells. Moreover, the serpentine loops also increase the hydraulic resistance to prevent any sudden fluid flow if there is any mismatch of the fluid level in the reservoir part. And with this new design, we have also applied the provisional patent. So we get the whole 1 year to file the final patent by the next year, July next year. So another thing that we have modified here, when we are applied for the award, we were fabricating this device considering the commercial perspective. We were fabricating this device using a technique called photolithography, but that is not commercially viable technique because it requires series of steps and clean room environment. So what we have done here is we have tried, try to incorporate 3D printing in the fabrication of microfluidic device. So of course, there are many challenges when we try 3D printing based fabrication method. So there are many challenges, but one of the challenges that we had to overcome was, so we are fabricating the master molds first of all using photolithography earlier, but now we are doing with 3D printing. So in 3D printing, if there is a roughness present on the master mold surface, it can prevent the bonding of the two layers. So that is why we had to explore many other 3D printing technologies and resins. And then we finally came across one technology which produced very good surface finish and of course facilitate very good bonding between the two layers. So as you can see, this is a lower layer mold in which there is serpentine loops and microwells present. And this is an upper layer mold which holds the fluid in the reservoir, each reservoir can hold 35 microliter of the fluid. So once then we pour PDMS, which is a well-known microfluidic material and biocompatible as well. Then we cure the PDMS and we have two layers, which of course need to be bonded using plasma bonding technique. And later on, we coat the channels using pluronics to prevent any cell attachment and facilitate spheroid formation, rapid spheroid formation in microwells. So here, after incorporating 3D printing, we are able to make more than 10 or 15 replicas of the device within 6 or 7 hours. Of course, here curing time, based on the curing time, we can reduce the fabrication time. So here we for 50 degree it requires 12 hours, but if we cure at 60 degree, it can be cured within 6 hours. So within 7 to 8 hours, we have more than 10 to 15 devices ready for testing. So this was the benefit of 3D printing, which we incorporated in the new design. So now to check the spheroid formation in the newly applied design, what we have done is we have culture MCF7 breast cancer cell line in the microwells. And this is a tumor spheroid diameter that we have measured for till day 4. As you can see, on day 1, on day 2, there was a slight reduction in the mean diameter, which suggests that spheroids are compacting. And after day 3 and day 4, the mean diameter was found to be increasing, which suggests that they might be proliferating. Similarly, when we did the fluorescent-based cell viability assay, we found that the spheroids were highly viable in the wells, which suggests that there is no toxicity coming from the 3D printed material to the PDMS, because that problem is well mentioned in many literature that 3D printed material often transfer the toxicity to PDMS material that might lead to cell death. But that was not in our case. They were highly viable on day 4. So now what we are doing is after initial proof of concept with the first design, with the drug testing, we changed the design. We involved the 3D printing, considering its commercial potential. And now our final goal is to isolate the primary cells from the patient sample and provide the proof of concept in the same device. So we are going with that same goal. Currently, we are optimizing the isolation protocol from the primary biopsy sample that we are getting from two hospitals, Care Hospital and Indo-American Hospital from Hyderabad. And we are optimizing the isolation protocol, and soon after isolating the cancer cell, the drug testing can be done, and maybe we can provide some data to the hospital that which combination works well on particular patient. So we are going with the precision medicine approach here, although it is very ambitious, but we are hopeful that within 1 year we'll get some really good result, and we also come to know about the commercial potential of the whole technology. So with this, I would like to end my presentation, and I would like to acknowledge my guide, Shubhanarayan Rath, of course, the coordinator of SISTI, Professor Anil Gupta, and whole GYTI team. Without this funding, this was not possible. And of course, we have also raised 10 lakh funding from Prime Minister Research Fellowship for the same project. And of course, I would like to thank my family and all the lab colleagues. Thank you.
Excellent idea and work, Viraj, let me say. I just had one suggestion, because since you've started with MCF, breast cancer, and there HER2 is one very, very important, some of the population is indicating. So maybe you can extend your work and make multiple channels with these cell spheroids. Simultaneously, can you test whether the drugs are effective against normal breast cancer cell lines and HER2 positive?
Yes. Yeah. So we can of course have a multiple of device, and multiple of device we can have for different HER2 positive and HER2 negative breast cancer. So, yeah.
And another
that's
thing, sorry, sir. Please.
Yeah, go ahead, please.
You quoted it with I saw poloxamar, if I'm not mistaken, right?
Yes.
So that there is no contact between PDMS and cells. So
yes.
how uniform is that coating, and how are you making sure that it is uniform?
So ma'am, like uniformity of the coating we haven't checked, but we have cultured the cell till day 7, and there is no cell attachment on the channel for 7 days.
Okay, so moving ahead, when you go for large scale, I think that will become one more criteria, because how much is the well depth, that would also be very important. Yeah. Yes, sir.
Just a small question, Viraj, that the effectiveness at early stage and effectiveness at later stage may not be similar, because proliferation rate of cells is much higher and targeting may become much less as the cancer cells diffuse. Is it true?
Yes, sir. So
if it is true, then you must partner with either a GYTI awardee or somebody else who has a good early diagnostic tool. And I remember, I mentioned it yesterday, I'll repeat it today. There was a student of a professor at ISC, I'm forgetting his name, 2012, he got GYTI award, where they had used the this potassium to sodium ratio in the cell as an early stage using visible light scanning facility to detect cancer even before aggregation begins. So that when there are very few cells, we do not have the current methods of diagnosis to be effective, because they are generally working when their aggregation already takes place, So if early stage diagnosis can be linked to your product, I think the effectiveness of your solution will become much higher. So think of partnering with some diagnostic enterprise and diagnostic startup or a student who has an early stage detection.
Yes, sure.
Yeah, thank you. If anybody else has any comment, please.
If I may, sir, chair, and I think sir both, very quickly, I think Viraj, very, very exciting. And I think precision medicine is going to be the way forward. We know that. And this is one way of looking at it. And of course, people are going to look at it from a genetic markers, right? Which would be based on the trend, what they would respond to, like what kind of population would respond to that. That is still a precision medicine. What you're doing is actually a personalized medicine, right? For the individual, right? Whether it responds to or not. So I think very, very good. If it would be very good to get a corroboration data from the, how this actually happened, and show the sensitivity of your, solution and the platform to what really happened. So show the follow-up on the clinical side of the after, therapeutic intervention, what happened, and show how sensitive your outcomes, because that's where the proof of the pudding is, right? So my view with that, if you can bring that aspect out as you go forward, not the product, but the sensitivity of the product is a very important. And keep that, saying that this is where my test lies, right? This is very clear. Say that, no, I have created it, but I will say that myself successful when it works this way, right? Device. At the same time, device is something I see that you are spending a little bit more time. I would say that if you can outsource to the places where already devices capabilities are, and I'll send you the link that we have. We have been funded by BIRAC to do exactly what you have been doing. So you won't spend time doing it. We have experts who have been paid by BIRAC, who have been paid at CGAM to take care of all that, right? Something that Professor Gupta mentioned. So you'll not spend months to do this. You can actually let it have, discuss the design with them, your rationale, why do you want it, and then test out a few designs and then test it out in your place and see which one works. I'm just This is a suggestion from my side. In the chat box, I'll send you the link as well. But
Yes,
there's a couple of suggestions from my side.
that will be really helpful, sir. Yeah. Thank you so much.
That's very useful.
Any questions from other awardees? So if there are no other questions, we'll move to the next candidate, Shibu, IIT Hyderabad, 2018 awardee.
Yeah, yes, ma'am, I'm joining.
Okay.
Is my PPT is visible, ma'am?
Yes, it is. Not in presentation mode though. Yeah, now it is.
Yeah. Okay. Yeah. Good afternoon, everyone. Actually, I'm Shibu from IIT Hyderabad, and I have received this GYTI award in 2018. So when I was applying, for this award, we just prepared some hydrogel from like bovine cornea. So, and we had a lot of hypotheses at that time, like this cornea can be used to treat many corneal indications, but nothing was clear at that time. But we proposed something that we can use this hydrogel for severe keratoconus. Keratoconus is a disease condition where the cornea gets protruded outside. So because of that, there will be blurred vision. So actually, it was during my PhD coursework, it was my just a PBL, like a problem based learning. So I just presented it, but then we have applied for this GYTI award, and then we got the award, and then we started working on it. We have collected bovine cornea, decellularized it, and then we have checked its all biocompatibility and its potential to become a biomaterial to treat corneal indications. So what happened later, we also started to collect human cornea from eye banks, and then decellularize it and prepare a hydrogel out of that. The peculiarity of this hydrogel is, since it's a tissue-derived hydrogel, its main component is collagen. And when we keep this hydrogel at 37 degrees Celsius and at a pH of 7, it will start forming a gel. So, so that was the status when we have received this award in 2018. So now cornea is a transparent layer which is located outermost of the eyeball, and it also protects the eye, and also it focus the light towards the retina. So, and there is as you know, the blindness. So because of this blindness, once childhood really gets spoiled, and then education, employment, mental health. So overall impact will be there in somebody's life, if a person is blind partially or fully. So that was the motivation of this work. So so we have focused, narrowed down our objective to a few clinical conditions, and because this damage in cornea leads to blindness. So in cornea, the clinical condition, there are so many clinical conditions which causes blindness, but we have picked few, for which there is no option of treatment or prevention. And also for keratoconus, there is only option is a complicated surgery or else transplantation. And for scarring also, scarring means the opacity at the center of the cornea or anywhere in the cornea. But when the scarring occurs at the center of the cornea or the visual acuity, the person will be blind. So and also injury, most of the scar develops after an injury or an infection. But after an injury, that really we can prevent, by using our hydrogel. So that was the main focus of this invention. And so these three disease conditions, whatever is mentioned here, the only option, the final option, is the corneal transplantation. And please remember, there is no, till date, there is no preventive or prophylactic method to prevent scarring followed by an injury. So our hydrogel that we have, as I was telling, we have developed it from two sources: human corneal that is discarded from the eye bank, disqualified corneas, and also from bovine sources. Then we have characterized it and found that there are a lot of cytokines, growth factors, all these things. That is this hydrogel harbors all these cytokines, growth factors, and the entire microenvironment for a cell to maintain its behavior that cannot offered by any other synthetic material. And the eureka moment of this study was this point. Like one day after culturing the cells in the matrix in the corneal derived this hydrogel, when we are observing it under microscope, generally based on my tissue engineering experience, the cell should express like a collagen and all whatever the matrix. But in this case, despite of like repeated experiment, we could not find collagen production. Then we were thinking like why it happened. Then we found that actually it is forming the excessive matrix production that actually leads to scar in the cornea. So actually this hydrogel is preventing the scar formation in the differentiation of corneal keratocyte to myofibroblast that ultimately leads to scar formation. So it is able to maintain the cell behavior. So then we have went for a preclinical study, extensive preclinical studies, and as I was telling, like we have applied for GYTI award with a proposal that this can be applied for keratoconus. Then later we have diversified our area of research, and we found that in the first figure, set of figures you can see here, we have created a corneal injury in the rabbit, and after 3 months, and then immediately after injury, we have applied the hydrogel. And then we found that after 3 months, the cornea was getting like a complete transparent and also with thickness, curvature, and everything. And you can see the extreme right side. If you don't treat this injury, it will like end up with this type of scar formation. Exactly at the center visual acuity, so that leads to blindness. And also the second application is like, so this is the first one is a prophylactic approach. This is for the first time. It is there is a report that after an injury, if you apply this hydrogel, scar can be prevented. And the second study was for the already existing scar. Like we must have seen, like many people have a small opaque area at the center of the cornea or anywhere in the cornea. So that is actually due to the scar that is already exists. So we found that we can easily like with the help of a surgeon, we can remove that scar, excise that scar, and fill this hydrogel at that place. And then over time, the area will become transparent. Means there is no scar formation, and also it can completely get help to get back the vision. And the third like work that we have carried out, that is the keratoconus. Actually, now we are not using the term keratoconus much because some more characterization has to be performed. That actually we are performing that now. And so we have published a paper recently. That is a ectasia model. Like ectasia means it's a thinning of cornea. So that general term only we have used. But we found that for the first time again, like this hydrogel can be injected in a thin cornea, where so that is a condition called ectasia, and means the cornea become very thin. So after injection, the cornea become like this hydrogel is getting integrated with the existing cornea, and it's helping the cornea to gain back its thickness. So a complete recovery could be identified. And here one more work we have carried out, because for this keratoconus or ectasia, there is no model is available in the world. So we have created an animal model, rabbit model, by depleting the corneal stroma and made it thin. And there we have studied the efficacy of our hydrogel. So this work is completely supported by GYTI. And please. Yeah. Then one more application that we found or that we could do with our hydrogel is we have 3D printed a complete cornea. Like in the previous conditions, like we can save the transparent, like no need to use any cadaveric cornea, and we can replace that. But here we were trying to mimic or develop a complete human cornea with the same size exactly what we have. So here we have developed, here you can see this we have prepared a mold, and then on top of that we have printed this hydrogel by using a 3D bioprinter, and we could achieve a cornea with the same size of human cornea with a similar curvature. And also we have performed some ex vivo study to check its implantation potential. So here you can see, like, this is the human cornea from the eye bank, and then we have removed the center portion, and we have placed our hydrogel, our 3D bioprinted cornea with the fibrin glue, and it was exactly like staying at that position, and it is like 90% similar to, or maybe more than that, similar to human cornea. So our next plan is to go for the animal study for this particular project, and yeah. So now we have filed two. Sorry. Yeah, we have applied for US patent along with LV Prasad Eye Institute, and also we applied for Indian patent way back in 2018, before I was awarding this GYTI. And then we have published one paper in ACS Applied Biomaterials recently, just 2 weeks back, on this ectasia model that we have, means ectasia study. And our future plan is to, we are now ahead of human trials. We are trying to getting more fund to conduct the human trials, and also a startup. And our plan is to make a gel like which is available in the market, so that we can immediately after injury, if you go to a doctor, they can apply it and prevent the scarring. And if already scar is there, that can be excised and filled with the hydrogel. And so a kind of product we are planning. And of course, for the cornea ectasia, some more studies we are performing exclusively for keratoconus, because for that there is no treatment available other than complicated surgery and transplantation. And also the most important thing, this keratoconus affects younger groups, like at the age of 10 to 30. So that's why we are more focusing and conducting more studies on the keratoconus. So thank you for all your support, Professor Anil sir and the team GYTI.
Thank you. Thank you.
Thank you, Shibu.
Yeah.
It's great to know that you are moving ahead. You are thinking of a product which could be used maybe in a gel form.
Yes.
Only thing is you will have to look at, what would be the impact of sterilization on the product.
Yes.
Because
Yes.
from the natural origin, that becomes a bottleneck at times.
Yes. 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 True.
Any questions, suggestions from other awardees or anyone else?
Is this hydrogel specific to the cornea, or can it be used elsewhere also?
No, sir. This can be applied only to the cornea.
Because in IIT Delhi, Dr. Ghosh, I think his students have used different kinds of hydrogel, silk,
Yes.
fiber-based, and others. So I was wondering whether there is a cross-domain applicability of this.
Yeah, no, actually these are called, these are tissue specific hydrogels. Like if you're taking this from skin, then it can be applied for skin, like maybe a wound or something. And so here, that's why we are collecting it from the cornea itself, so that it will be having the same.
That means the method
Yes.
that you have developed can be
Yes.
used to create hydrogel, tissue specific hydrogel. Can we say that?
Yes, sir. Actually, people are using detergents and many other chemicals to decellularize. Here we have done it only with the sodium chloride. Only with sodium chloride, no enzyme, no chemicals, nothing. So in that case, we could see as the cells are performing better and cells are more attracted towards the hydrogen.
Why I'm saying this is that in that case, you should file another application on a patent application for generalized application of the method that you have applied, with some examples that you have. Because I think this method that you have developed of using only sodium chloride for decellularizing may be applicable for other tissues also, isn't it?
Yes. It can be used for other tissues also, the same method.
Have you claimed that in your patent application?
No, sir. Actually, we haven't claimed it for other.
There are two choices. Either you file a patent amendment application, which means that you amend your application, because it is not granted as yet, no, isn't it?
Not yet.
So you can amend your application. Either you amend your application and add a claim, or you file another application.
Okay.
Add-on patent would do, right, sir?
Yeah, addition to the patent can be done. Patent application can be done.
Okay.
Another area where you can extend this work is in keratitis. Now specifically because of COVID, many patients are reporting that. So maybe there also it would be useful. Looks like.
Yes, ma'am. Actually, any kind of cornea intervention, like we found that even after surgery, people get some scar formation in the cornea. So any kind of intervention have a risk of this scar development. So we found that it can be applied for all these type of corneal indications. But as you said, when we are using, and also we found that bovine hydrogel is doing better than human hydrogel, human corneal hydrogel. But the challenge is when we are talking about bovine, there is a risk of zoonotic diseases. So actually, long back I wrote to Professor Gupta sir also to connect me with the Virology Institute, and he has done that. But we have a lot of constraints, like I have to go there and work. For that, I have to get trained. So maybe all these things has to be solved in future. Because as Vandana ma'am said, there is a chance of zoonotic diseases, and if it is human hydrogel, of course, the cornea that we are using is already have gone through all these procedures, like all screening procedure, but still we need to prove that it does not have any microorganisms. And so sterilization of a hydrogel is also quite difficult, and everything has to be done in GMP facility. So our next aim is to conduct the toxicology studies by overcoming all these hurdles, and then conduct toxicology studies and prove that this is completely safe. So far we have proved that there is no immunological response, and it is forming efficacy also could be proved. But now the challenge is the toxicology study, and it's a sterilization and all these things, like actual commercialization part that we need to do.
Good, very good.
Good. Thank you, Shibu. And we can move on to the next one. Mani Kuntala from IIT Kharagpur, 2019 GYTI awardee.
Yes, ma'am, I'm here. Am I visible?
Yes, you are.
Yes, you are.
I hear you clearly.
Good afternoon, everyone.
Good afternoon.
Yeah. I'll just start sharing the screen. Is my screen visible?
Yes, it is.
Okay. Okay. So good afternoon, everyone. I am Mani Kuntala Mukhopadhyay, and currently I'm pursuing my doctoral studies in the Department of Chemical Engineering, IIT Kharagpur. I'm in the final year of my PhD right now, and I was awarded GYTI in the year 2019. So my talk for the day is interfacial energy-driven distinctive pattern formation during the drying of blood droplets. So the whole motivation of the work came from a very easy concept, which was drying of fluids. So drying is a ubiquitous phenomena, but the physics of the drying is extremely fascinating and intriguing, because drying depends on several factors like the physical properties of the fluid, the contents of the fluid, the particle size, and the substrate nature as well. So we wanted to see that whether these the physics behind the drying can be used to detect diseases or not in case if we replace a fluid with a drop of blood. So in the literature earlier, some people, very few works have been done and people have started exploring and looking into this domain, that whether dry droplets of blood could be used to detect disease or not, or any other body fluid, let it be plasma as well as urine. So our main motive was first to understand the physics of the process, so as to understand what are the interfacial attributes that actually helps in the formation of the drying pattern, and then identify that whether it could be used for any specific disease, which will have certain differences, so that it will be easier for us to get a vivid description along with something to correlate with, or some factor or some tangible output which can be used to detect a particular disease. So we collaborated with Calcutta Medical College for this work, and during our ongoing discussions, what we came across is thalassemia. So I will talk about both the current market scenario for detection of thalassemia and why we chose thalassemia. So India contributes to around 10% of world's thalassemia major childbirth. And at present, what is done is the government hospitals, they conduct camps and they visit various rural areas and they collect the blood samples, and each of the blood samples undergo HPLC, which is high performance liquid chromatography. And they basically, each of the samples undergoes a gold standard technique in order to identify that whether the sample has thalassemia or not. So per sample costs around 200 rupees. So this is huge for the government hospitals, and also it's a lot of workload on them, because undergoing HPLC for each and every sample is time consuming, and also it requires infrastructure. So what they suggested us is that can we try this thalassemia to begin our experiments with, and if we can devise a method which will reduce the initial load on the government hospital, so that we can say for sure, these are healthy samples and these shouldn't be sent for the gold standard methods, and only these samples can be sent. This was our initial motivation. And looking from the physics perspective, in thalassemia, what happens is there is a reduced synthesis of the beta globin chains. So the alpha chains, which are extra, they try depositing on the red blood membranes of the red blood cells. So this ultimately leads to membrane damage and as well as lipid derangement. So what happens is there are two components in our red blood cell membrane, which are glycoprotein and sialic acid. And ultimately, they get mal-distributed when a person is suffering from thalassemia, which reduces again the spectrin content of the cells. So all of these effects ultimately lead to reduction in the overall negative charge on the surface of the red blood cell. So this was one of the major physicochemical property of the cells of thalassemia, which was different from that of the healthy blood cells. And we thought that we could use this as one of the studies in order to identify that whether it could be highlighted in the drying patterns or not. So I wish to highlight that when we started our work and when I was awarded GYTI, it was completely in the nascent stage. And right now we have performed experiments for over 100 samples over a study time for more than 12 months. And for each of the samples, we have undergone the physical characterization tests as well, where we have measured the beta potential and the MCD as well, which I was saying in my previous slide, which is different for the thalassemia and the healthy samples, which I've highlighted here. So our experimental setup is very simple. What we do is we take a glass slide, just wash it with ethanol and water, and put blood droplets of both the healthy and the diseased samples on our glass slide. And we allow it to dry under the ambient conditions. And once it is dried, it takes around 15 minutes to get dry. And once it's dry, we observe it under the microscope. So what we were observing was that consistently, and it was repeatable for all the samples that we have done for around 100 diseased and healthy samples, that the diseased blood samples had track length which was significantly shorter than that of the healthy blood samples. So this was something which was easily visible to us, and measuring the radial crack length was also quite easy. So then we thought of going for a 99% confidence interval method, where for all the 100 samples that we have done, we had created, or we had measured the radial crack length, and then we came across a threshold upper limit value above which we could classify that, okay, these samples are healthy based on the radial crack length, and the other samples whose crack lengths are less than 800 microns could be sent to the gold standard method. So this basically reduced the load of the samples on the government hospitals. So we did some blind tests with around 50 samples. So initially our main target was to, in order to avoid any false negative results. So right now we are working towards fine tuning of the method for the false positives as well. I'll talk about it towards the end of my presentation. So we performed our experiments with around 50 samples, and we got 0 false negative results. So all our results were again validated with the gold standard method, which is the HPLC results. So I won't go into the physics of the process much, but I will just give a brief description as to why this is happening. So since the zeta potential or the surface potential of the cells are different, so we started looking into the interaction energies of the system, because crack is nothing but it's an essential signature of an energy release. So what did we do was we calculated the interaction energy of the system based on the cell-cell and the cell-substrate interactions. We did lots of HSM of the samples, and it was quite interesting when we realized that when a blood drop dries, these cells, which are looking like pancakes, they stack on top of one another. And the overall interaction energy of the system for the healthy and the disease samples was significantly different. So for the carrier samples, the total interaction energy of the system was less than that of the healthy samples. So the radial cracks which we were seeing, which is nothing but energy release for the carrier samples, was significantly smaller as compared to the healthy ones. So we have done a detailed theoretical model as well, and we have calculated the ratios of the crack length, which we have again matched with our experimental results. So after that, we thought of developing an in-house software, which we have made it right now. We have named it ASAP. So if we click an image and if we upload it into this software, it can automatically calculate the crack length. We don't have to use ImageJ, so it can calculate the crack length, and it can give us the result based on our threshold limit that whether it is a healthy or a disease sample. So we published our patent last year, and towards the end of the last year, this work got published as well. We have also presented our work in Hematocon. Hematocon is a conclave of where all the doctors from all over India come, because we wanted to show our work to them, and our work was selected among one of the 5 best papers in Hematocon. But I feel that these patents and publications are just the beginning of the work, and a lot more work needs to be done. As Professor Anil Gupta was highlighting yesterday, since I am in the final year right now, so a certain part of the work are being planned, which will be carried out by one of my junior colleagues. So first amongst them is we want to fine tune the sensitivity of the entire system. Because right now we were working with 2 ideal samples. One was diseased and another diseased only with thalassemia, and another one was completely healthy. So what right now we have started working on is we have started looking at other common diseases. That what if a person is having thalassemia and some other common disease, or if a person is healthy with some common disease. So will these patterns change? So we have started working with anemia samples to begin with, because low ferritin content is something which is very common in India. So what we have seen is, as of now, for the samples with low ferritin content, the results are all in the healthy range, and they are not interacting with or they're not interrupting with the crack length of the thalassemia one, because again, the zeta potential of the cells or the surface potential doesn't get different in the person who is suffering from anemia. But there can be some other disease which might interact, and it can show a crack length less, even if the person is not suffering from thalassemia. So right now, we are working towards fine-tuning the false positives of the system so that we can reduce the total number of false positives. And because of Corona, some parts of the work have got significantly delayed, but we do have plans to visit the camp, which will be organized by Medical College Kolkata, and will be performing on-site tests with them so that we can get immediate results, and the rest of the samples can be sent for gold standard methods. So as an extension to this work, as a part of my thesis work as well, I'm looking at the plasma patterns as well for detection of a liver disease, which can again have a significant role in the diagnosis of the disease. I would like to thank GYTI for recognizing our work, which at that point of time was in a very nascent stage. I'm thankful to my supervisors and Calcutta Medical College as well for collaborating with us. Thank you.
Wonderful.
Thank you so much, Manikuntala. Very simple yet such an effective tool. I think that's the need of the hour. And I'm sure you already said you are looking at other diseases. So one disease which I would like to suggest would be sickle cell anemia.
Sure, ma'am.
Majorly, there is an issue identifying people, and it would be great if you could extend the work.
Okay, ma'am.
And there are populations which have high incidence of sickle cell anemia presence, but not disease. Now, please understand that there are tribal communities which have the presence of the sickle cell, but do not get affected by it. So therefore, there are two cases now. One is presence of the causal organism, but not causing the disease. Second, where it manifests into disease. Isn't it, Vandana? So therefore, that might be
absolutely, it will be a very interesting study to do to pinpoint, why some of them have and still not show the symptoms. Yeah.
The other thing I was wondering is whether if you dilute the blood with some agent, whichever you think, saline or whatever, can that make it more specific to disease? So false positive, one way you can reduce false positive is by treating the blood with something very simple, right on the spot, just stir it and then take the drop instead of taking the pure blood. Does it, have you tried that? Is it possible?
Yes, sir, one of my earlier work, like before this work, I used to work on studying the morphological alterations of the red blood cells. So that from there, I started this work because there what I saw was when we were doing simple smear on the glass slides. Now there are several diseases like stomatocytosis and spherocytosis, where basically people were looking at the total, they count the total number of cells that are deformed. And all these tests were performed on glass by smear. Now what happens is when we dilute the cell and dilute blood and put it on the glass slide, they undergo deformation. So that is one problem which is predominant. So what I suggested at that point of time was I coated it with a thin layer of protein, because the red blood cell membranes have protein coat, and if they interact with a substrate which is favorable to them and which is not harsh, then the deformations of the red blood cells reduce to a lot extent. I've studied in detail the physics of the process as well there. So diluting the blood samples, if we are using glass slides, it can trigger that problem that it can deform the red blood cells, so which can interact with the pattern. So we can look for maybe some other substrate, if not glass, by looking, because there also the interaction energies will come into play. So instead of glass, if we can use some other substrate and then dilute the sample and then see the results, then it might be effective. So that we can.
For my curiosity, another question. The biopotential, what did you say, biopotential? The physics of
zeta potential of the cell.
So when you look at the beta potential, what are the variables which affect it? Just for my knowledge, which are the factors which affect it?
So zeta potential is basically the charge that is present on the surface of the red blood cell. So this charge is contributed by mainly all the components which are present in the red blood cell membrane, but most likely the glycoprotein, sialic acid, and the spectrin content. These three are one of the major contributors because they are just on the surface. Suppose if you are placing the red blood cell like this, so this part which is coming in contact with the glass substrate, so these three components are present in just the vicinity of the red blood cell membrane. So these three are the major components which will contribute to
basically we have to manage a surface which has a charge, which has a different charge, or which does not neutralize the charge. Is that right?
Yes, sir. So that what I did in my previous work when I was working with the morphology. Once I coated it with protein. So glass has a charge of around minus 60 millivolts. So what did I do was, once I coated it with protein, the charge reduced to minus 8 millivolts. So when the cells were interacting with a protein-coated substrate, the energy was much less and the cells were not getting deformed. So basically, we were seeing the morphology of the cells as it is in the body. And there were no glass-induced deformations on the cells.
Very interesting. Tasleem or anybody else has any question, that would be interesting. I find this work fascinating. This is amazingly interesting.
Thank you so much.
A simple question. I shared your slide, when just a simple question can trigger such a profound research, and you have looked at the physics of the surface, tension, some sense, and
thank you so much.
It's a very interesting work. You should advance it for various kinds of diseases.
Yes,
and you know what? I will tell you something. When homeopathic medicine is used, once I went to Germany and visited a homeopathic medicine plant. You know what they do? They look at a blotting paper, and after they stir the solution, medicine, they put a drop. And the imprint of the medicine on the blotting paper is used to test the effectiveness of the molecule. So those who say that homeopathic medicine has nothing should look at this test, and they will find that different dilutions have a different pattern. And I was so deeply impressed many years ago, 2-3 decades ago when I went there, I noticed that. So what you're telling me is something profound. It has a clue, which, as Dr. Vanna said, it might be useful to look at other diseases as well. But you have already answered a paradoxical question that the anemia does not show that. Difference in the anemia doesn't show that. So obviously there is a category of diseases which only can be diagnosed. Very nice. Thank you.
Thank you.
Thank you so much. Thank you, ma'am.
So now we move on to the last one in the session. Harsh Maheshwari, 2018 GYTI awardee from IIT Madras.
Yes, ma'am. Can you see me, ma'am?
Yes, we can.
So just give me 1 second. I'll present my screen. Where is the PPT? Yeah. So can you all see my PPT?
Yes, not in presentation mode though.
Just 1 second. Yeah, now can you see? Is it in presentation mode or still it is in?
No, it is in presentation mode now.
Yeah, thank you, Vanda ma'am. So hello everyone. My name is Harsh Maheshwari. I am successor of original awardee Kiran Amireddy GYTI 2018. I'm currently working. I'm a MS student in the final year of my graduation. So today's topic is super resolution ultrasound imaging technique, and this work is carried out under the guidance of Professor Prabhuja Gopal at Center for Non-Destructive Evolution (CND) in IIT Madras in Mechanical Department. So before I actually explain about this topic, I'll share my personal experience, which I had few months back only. I was diagnosed with the multiple poly, which is it is kind of a small pimples that is being created in the inside the large intestine area. So if this diameter of that poly size is more than 10 mm, then it need to be cutted out. Otherwise, it will have the serious bleedings problems, leads to serious bleeding and problem. So like when my first diagnosis was found, I was found with the diameter of more than 10 mm. But luckily my doctor was so advanced, and he knows the area, so he asked me to go for another inspection with the higher frequencies. So later it was found it is not one poly, but it is multiple polys which is having the smaller diameter. So I was lucky, and it got cured later. So currently you can see that the problem of the technology that we have in the biomedical is the resolution problem. So we face a day-to-day problem of the resolving the area in the body. It is very difficult. So the work that we have done is to resolve the current existing technology with the better resolution techniques. So as we know that ultrasonic imaging is kind of widely used in the NDE, non-invasive diagnosis, and non-restricted testings. The reason is it has the high depth of penetration as well as versatility, and it is safe. It is kind of safety because it does not have any radiation or something. But yes, the problem with this ultrasonic imaging is that it has some limitations of such called a relay diffraction limit, which is lambda by 2, which that I was talking about, that my poly, which is it was smaller than it was bigger than 10 mm, which was not able to resolve because the frequency that the doctor used, it was lesser than the lambda by 2. So that I can show you this in one of the image that I took from internet. It's like this is unresolved image. Here you cannot see the gap, but here with the high frequency you can see the gaps. So this is called resolving the image with the high frequencies. The problem with the high frequencies is that it will decay with very quickly. It won't pass through for longer distance. Since it was on the surface, this defect this thing was diagnosed. But if it's beyond the surface limit, then it is very difficult to get resolve such an anomaly in the body structure. So one more thing is, like, conventional techniques have the higher instrumental cost as well as the high low penetration in the material. So that's why we come up with the techniques that is used for non-invasive diagnosis, which is used for the super resolution. So before I explain what is super resolution, first let's understand what is super resolution means. I meant, so let's say there is a two anomaly presence in the system or any body or any material. So when it is exposed to any wave for detection purpose, so there it will generate two components. One it is low frequency component, and one is high frequency components. So high frequency components are called evanescent waves, which will decay very quickly from the surface. So if someone is unable to detect this high frequency component, they weren't able to see these two different defects individually. They will see this full defect as a one full defect or any anomaly. So what if we can manage to harvest this high frequency component by some means that can be useful to diagnosis this multiple polys or any other anomaly presence in the body. So there where our technology comes, it is called metamaterial technology. So what is metamaterial? Metamaterial is nothing but it's artificially structured material which has a property which generally do not found in the natural occurring material. So this metamaterial that I am talking about is the resonance metamaterial, which in which the length of the in this metamaterial we have created certain holes, you can see here, and the hole length is corresponding to the Fabry-Petrov resonance. When the length of the metamaterial is equal to the multiples of the lambda that we using, then we can harvest this high frequency component, and then we can go for the detection of these two defects individually. There are other projects which is carried out in our lab, such as the focusing. This is carried out by one of the PhD student in our lab, Manjunath City. So he was able to focus the wave beam in the material using bimetallic lens, and this also was patented later in 2019. Similarly, one of the students from our lab, Sayaditya Kuchi Batla, he was able to manage to manipulate the wave in the bending in opposite direction. You can see like the wave is completely flat, but later it was got bended using the metamaterial concept. And another property of the metamaterial is acoustic bend, which was my another work that I was carried out during my masters. That to stop or eliminate the surface vibration of, or you can say seismic vibration by using the metamaterial concept. So so these are the milestones that till now we have achieved during this project from main awardee, the Kiran Amireddy and me. So earlier he was able to manage to detect the defect which is placed apart lambda by 5. You can see the distance between two defects is lambda by 5, and using the ultrasonic imaging technique with the metamaterial added, he was able to detect the metamaterial, as you can see this simulation result as well as experimental result. Similarly, what we have tried later is, like, since we tried to know, we learned that metamaterial is used for the detecting unresolved image. We try to optimize the parameters of the metamaterial, such as the hole diameter, the pitch diameter, pitch, and the as well as the length of the hole. And doing so, we were able to achieve the lambda by 25 resolution. You can see this one. This is the this is without metamaterial. This is with metamaterial, and the this was also carried out with the X-ray, but it was not able to detect. But using our technology, we were able to detect the defect present in present, which is having the dimensions of lambda by 25. This two work was published in various publications, which is mentioned over here, as well as also it was published in the scientific report. It is one of the highest ranked natural communication. Then later we, since this metamaterial is periodically placed, periodically drilled, instead of periodically drill holes, we tried to dig in the more into the non-periodic metamaterial or non-periodic lenses. What actually benefits we get is in periodic lenses we have limited frequencies which can be resonated and amplified, but here we can see that few time because of the wood anomaly present in the non-periodic lenses, we were able to achieve the resolution of lambda by 36, as we can see here. This is with X-ray, as this is without sorry, this is without MMM. This is with metamaterial. You can clearly see the defect present over here. And this work was published in APL as well as the conference in the ESEMSE in Sweden, 2018. Later, since the resolution that we have achieved in lambda by 5 or 25 or 36 was using the laser as a detection. So as we know, the laser has many problems. It is very sensitive and prone to have a noise in the signal due to the high sensitivity in the high sensitivity and the ambient disturbance. So we try to kind of make this laser to be avoided and make this commercially available transducer with the cone structure, so that we can productize this concept that we have made in the laboratory, the metamaterial with the cone structure. We try to do the same analysis that we did with the lambda by 5, and we were able to achieve the same results that we have achieved using the laser technology. So we have absolutely the laser technology, and we introduced the commercially available transducer. So with this same commercially available transducer, we can do better sonography, or any better material characteristics process with this technology. But the cone technology that we developed was just experimental based. So later we tried to optimize the parameter of the cone, such as the diameter of the cone and the length of the cone, and we found that the 2.75 lambda length of the cone is the better giving results. As you can see, it has a better transmission ratio at 2.75. And so after doing all this analysis, we finally on the product development in the trial phase 2, where we have developed the metamaterial fixer. You can see this metamaterial fixer, where we're gonna put this sample, and we will try to generate the signal from the bottom, and we'll try to capture the signal from the top and do the full analysis. So this technology we have developed for the material characterization, but it also has the applications in the biomedical, that as I told you earlier, in the sonography that I was diagnosed with. And it has so many advantages, such as low cost, radiation free, it is portable, as well as it is easy to implement and handling, and less sensitive to ambient temperature. One more thing I would like to add is, since I didn't have the time to do full PPT, I just saw my publications that over a period of time we published. These are the publications that we have done in this project, as well as this is the awards that we were our team and our professor was awarded. These are the 4 different awards, and they, with this, I would like to conclude my summarize my project, saying that this has a very highly advantage in the biomedical field as well material field, And if this product can be further if it can be further implemented in the biomedical, it will revolutionize the biomedical inspection technology. So with this, I would like to thank you, my professor Prabhaja Gopal, as well as the GYTI award for the funding this project. Thank you.
Wonderful. Wonderful.
Great,
Amazing.
Harsh. Thank you so much. Anyone having any questions or suggestions?
How far is it from, let us say, standardization into transducer fitted into the system? So a kind of a well-packed device, how long it will take to create
Yes. So
device? At the moment, it is all at lab level,
yes,
lab scale.
yes,
But
yes.
when it can be doing pilot testing, when can one think of pilot testing by doctors?
Yes, so currently we are at the trial phase 2. So what our professor has visioned is to make this first in the material characterization development. Then we were thinking to make it in the portable small devices, so which can be used for the doctor purposes also. That is our trial phase 5. So once we will complete the trial phase 2, we will move forward to design for the biomedical applications similar to that we have done for the material characterizations.
How long will it take?
So like we have envisioned with since it was like by material characterization project should have completed by last year itself, but because of corona we were not allowed to go in laboratory and perform experiments. So we were in me and our team envisioned to complete this by 1 year full trial till trial 11 something.
Wonderful. Thank you so much. Thank you.
Thank you so much.
So I'll request both the chair to give any overriding impression on the advice that you have for the students. Tasleem and Vandana, if you have any advice to give to the students.
In fact, I always say, it's been a good learning. Now this is a time, all earlier time we used to teach. Now it's good time to learn. So definitely each one of them is giving their best, and we appeal to them that do not lose hope. Anytime you need anything, definitely from GYTI side, people are there. They are able to connect you to different mentors all across country. Even if needed, advice from overseas can be given to you. But since we are talking about Atmanirbhar Bharat, obviously we do have sufficient expertise within the country to support you in case you falter at any step. So take advantage of this and move ahead. That's what I would like to say. So
Tasleem, are you there?
I can't see him.
Has he left?
At least in the participant list, I am not
able. Okay. Well, all right. So what I would suggest is that I was thinking of an idea. Tasleem is not here, but I was thinking that can we do some crowdsourcing of use cases of these platform technologies? So can we ask people to find as divergent ways of using this from biomedical to material science? Because what you mentioned, Harsh, isn't it? Harsh. Yes. It could even be used for material science research, is it not, Harsh? Harsh?
I can't see Harsh in participant list now.
Sorry, what is the name? I'm sorry. Harsh is left? Harsh is left?
He's left, looks like.
Okay. Yeah. But what I was thinking, Vandana, that if you think that sometime we should brainstorm on the idea of doing crowdsourcing of use cases for platform technology, because not everything will strike to a scholar. Scholar is too focused.
Right.
But for finding unrelated applications, we need to get into the larger group. So sometimes we can think about that.
In fact, I had written few suggestions, and in that one of it was, can we actually pose open innovation challenges?
Yes,
know,
yes, yes,
where awardees also can come together and solve. And
yes.
if not awardees, meaning it can be open to anyone for that matter. And very correctly, sir, you said that many times it so happens is biologists are working in silos, chemists are working in silos. We need someone to who can actually bring all of them on the same platform. So what has been published maybe 10 to 15 years back, it can be put to use if we really study it well, and especially with respect to drug delivery. And can be definitely done. Other thing which I had written few points, if
Please go ahead.
I could, I was going through even our website, and I wanted to probably see, who are the awardees. And when I look at the website, there are no contact IDs given or number given. So it would be a good idea if we can put contact IDs at least of all the awardees. Third thing which I had written is, of course, there were very good suggestions. Yesterday, I happened to be there and heard yourself as well as Dr. Renu Sarup about alumni. But what we can also do it on the Srishti website itself, if we can give, rope in some clinicians and rope in some mentors who can do their hand holding. So they'll be free, if we have an idea and they want to take an opinion before even they start, they can probably go to those people. Not all clinicians have time, but I'm sure someone who is India centric definitely would give time irrespective of these things. So if we can, and as also from industry few people, so because that is some missing links, there are some missing links that way. Plus, I think the last thing which I had written is just like when I was a Bill Melinda Gates grantee, or even Innocentive grantee, they have their own LinkedIn groups where a continuous exchange of idea goes on. So I think maybe we can take a lead from there, and there can be more interaction rather than meeting once a year. They can meet on a daily basis whenever there are any such.
Question is there, somebody has an interesting finding and wants others to react to that.
Absolutely.
We will follow up this. In fact, that is maybe the alumni group can be made on LinkedIn. That's what you're saying.
Absolutely.
So that it will serve both the purposes.
Right.
It's a good idea. Very good idea. We will take it forward. Thank you so much. So I thank Vandana, and of course in absence to Tasleem also, for wonderful opening remarks, your presentation, and the comments that you have given to different GYTI awardees. I hope that they were all found it useful. We will meet at 2 o'clock. We will not change that. So at 2 o'clock, the next session will take place on business development. And then we will have the final discussion, the panel discussion at that time. The Pandita is also going to join us, and Dr. Vipin will join us from INI. Thank you so much, Nanda, and all the best to you and your wonderful work that you're doing.
Thank you, sir.
Positive spirit that
blessings are very important. I think we are lucky to have you here in India, and it's the likes of you and Dr. Mashelkar who are our role models. So I'm sure we'll keep doing our best and moving forward. Thank you.
Thank you so much. Thank you. Thank you. Thank you very much. Good luck. Good morning, friends. Good afternoon, friends. We are now going to begin our post-lunch session at 2 o'clock, and we have several speakers lined up for today. We will wait for Madhusudan if he is joining. Let us hope that he is joining very soon. And then we will also have Dr. Vipin Kumar, Dr. Deepanvita Chattopadhyay. She might join a little late, but I can tell you that she's one of the most knowledgeable, experienced mentor for the incubation and business development in the country. ICICI Knowledge Park was set up much before any other incubator or any other park had come up at the time. And I have visited her facility, and I can tell you that they have done a great job of not only providing incubation support to the students and scholars and startups, but also created a wet lab for biotechnology, life sciences, which students and scholars and startups can use. So the kind of a common lab facility have been created. So I'm sure Dr. Vipin will share with us the experience of incubating innovations in NIF, National Innovation Foundation, which RDB Network had helped in setting up in 2000 by scaling up the model of GYAN. So GYAN was set up in 97 and Srishti was set up in 93, and the network started around 88, 89. So friends, I'm very happy and welcome you all. I'm waiting for some more GYTI awardees to join. I'm waiting for Nitin Basant, Ravindra Backward, and Weber. Weber was here yesterday, but I'm hoping that he will join us today also. We already have Prem, and we have Prem, we have, let's see, Jayesh is also here, yes. And Jayesh, we have. So my suggestion would be that you should also help in giving comments to each other wherever you feel that there is something to be added. So don't feel that the comments have to come only from the mentors or only from the professors or senior colleagues. Alternative networks are far more important, and I will remember, I shake eyes there, and I can tell you that yesterday one of the present, I think it was Ramesh, who was talking about a link he wanted with some neurological experts who are on the clinical side. And that said, I know somebody in Imans and I will connect you, and maybe she has already done that. So this kind of connection that we can forge are a very important part of fostering the ecosystem of collegiality, of solidarity, of mutual help. And I would like to, by the time Madhusudan joins, I would like to share with you a concept of network entrepreneurship. And I mentioned that briefly in the opening remarks, but I would like to share with you a little more details of that concept, because I believe that network entrepreneurship has a long way to go in this regard, and we have not really scratched even the surface of what can be done together. So network entrepreneurship essentially means that entrepreneurs, even if they are in complementary space, then all the more, but even if they are in competitive space, they can still collaborate in certain aspects of their work when their domains, either technological domains or spatial domains or sectoral domains, are non-overlapping. So I'm based in Bangalore, you may be based in Mumbai, somebody is based in Delhi. And let's say we all three have developed a diagnostic tool for TB. Can we cooperate? Yes, we can. We can cooperate. It is possible that our offerings are at different scale or are different complexity, and depending upon the customer, I can say, sir, my technology will not be most appropriate for you if you have a small hospital or a single doctor hospital, because it requires investment of the higher order. Maybe you should go to Vikas Pandey, who has developed a fluorescent microscope in about 60,000 rupees, and that might be very useful for you. So you can promote somebody else's technology which is low cost or has less complexity. You don't lose anything. The customer or the potential buyer gets something very satisfactory, and they would have to do a lot of effort to find out this solution that is existing. So we may have developed solutions of different complexity, different cost, different affordability, and many times it is not possible that every customer will find my offering as most suitable. What should I do? Should I refuse to serve the customer? Or should I refer that customer to somebody else who has a solution which is suiting that person's need or that company's need or that buyer's need? And in that sense, I earn an IOU. So next time when that person will receive a customer who doesn't feel satisfied with his or her offering, they will come to me. He will refer it to me. You know, doctors do a lot among themselves. They refer patients to different specialists and different labs for different tests. Can we not do that? Can we not build up a network system whereby we grow together rather than growing alone? And that would be very, very useful if we did that. It's very important that we learn to exploit it. So what are the elements of network entrepreneurship? Let me discuss 4 or 5 key elements of network entrepreneurship. Number 1, I and you work in different domains. Let us take the simplest case. You work in diagnostic, I work in drug delivery. You have your client, after having used your solution for diagnostic, would obviously like to go for treatment. I have a treatment plan, or I have a platform technology for delivery, say microneedle patch. Dr. Rohit mentioned yesterday that his lab at IIT Bombay has developed microneedle patch. One of his students got GYTI award for that. So now, if delivery system from IIT Bombay, molecule, let us say from IIT Kharagpur or ISC, and diagnostic tool from IIT Delhi or IIT Kanpur or IIT Madras, can we not build a chain? Can we not help each other to grow? So one idea is that we build a complementary value chain and offer to the client, to the potential licensee or to the potential buyer, a basket of opportunities rather than only my solution. So long as I work alone and with limited budget to promote myself, I will not go very far, let me tell you that. But if I become part of a network which promotes each other, my growth will be much higher. This is the first point that I want to. The second point, no less important, is I can make a catalog where I can list technologies, different components, supply chain members, and offer that to the patient. And I can say, look, if you want this particular solution from me, because the half-life of that is a little less, you have to get it every 15 days or 20 days or whatever. Say, for example, we were discussing that a while ago about hydrogel for corneal healing. And assume for a minute that hydrogel has a short shelf life and the company which makes it has to provide it fresh for maximum 15 days or 10 days or 5 days or 2 days, whatever it is. Then you need a supply chain which will deliver it in real time within 24 hours if it is required. Because the patient will need it, it may be accident case or injury case. So we need to have supply chain members, supply chain service providers, transporters, packaging people. Somebody has to put it in a liquid nitrogen container. I don't want to make it myself. Somebody is making it. And I don't want to store it because I don't know when will the need arise. So can I immediately have a very smooth, seamless linkage in such a way that when I need it, I'm able to get it. Welcome, Vipin. I was just talking about network entrepreneurship. So first point I mentioned that when there are technology which are non-competing, we can try to promote each other's technology, not just our own technology. Second is when we want to promote, we can also have a catalog of joint technologies, but not just technology, but also supply chain. So if somebody says, I want Viticode or I want this corneal hydrogel, then the person should not ask the question, how will I get it? The person should know that we have a network of liquid nitrogen containers, we have a network of the transporters, we have a network of of somebody or e-commerce platform where you can pay for it, so that everything is seamless. The customer gets the delivery of the hydrogel because all other actors in the supply chain have been networked and have been put together in the system that we have. Third possibility is that, and this has happened in many cases where the entrepreneurs will like to customize their services. We were talking about a personalized medicine. There was a case where the GYTI awardee had developed a personalized medicine solution. And there, you obviously will like to have tissues, specific tissues for which they have to do a grafting, or they have to do a solution provide. They have to provide a solution. Now, in this case, the matching of the tissue with the graft that is there has to be done in your lab. So you obviously need people who will collect the sample, bring it to your lab. You will then work out its compatibility, find out whether your solution works with it or not. Because there's no point in supplying a solution and then the customer says, look, it's not working at my end. It brings bad name to ourselves. We are selling something which may not be suitable. Why don't we then do the pre-test, compatibility test at our end, with which we have to match the solution? This also happens sometimes in the bone grafting cases and other cases where we try to use various kinds of scaffoldings which are required, and their compatibility has to be tested. So what I'm trying to say is that we have often tried, our entrepreneurs have often tried to go alone. And startups, even during the COVID-19 diagnostic, I was doing interviews with several of the startups. And there are people who are supplying chemicals, there are people who are supplying different components. And the customer is made to go to each one of them. Why can't I link them? Why can't I create either aggregation platform or a catalog so that I can provide a end-to-end solution to the customer? Most customers would not like to go to 5 places to get 5 ingredients to, fulfill their need. They would like to have one point at which they can get all the 5 things delivered, for which our entrepreneurs have to rise to the occasion and think like that. You think yourself as a customer, my dear friends who are online today, GYTI awardees, and see how would you like to get a service delivered to you. So then if you want certain services to be delivered in a particular manner, please remember that you have to deliver the services with equal seamlessness and similar convenience and efficiency. And I don't see any reason why we will not be able to meet that. So that's one part of network entrepreneurship where we promote each other, we become franchisee. That's another issue. We can also become franchisee of each other. So I am having a drug delivery system, you are having a diagnostic, and I can provide your solution at discount, at a commission. Maybe I'll get 20% of the value for generating demand for your product. Why not? Anyway, if you have appointed a distributor or if you have appointed a retailer, you will give 20% margin to the retailer, another 25% or 20% to the distributor. Why can't you give to the fellow innovator? So in that sense, each innovator can, in our own system also, can become distributor, can become franchisee of 5 other products. So if I go to a seed entrepreneur, I will find a catalog hanging on his wall or on his website of 10 other seeds.
Sure.
Now you are promoting your variety, but why can't you let me know of other solutions that are available for other crops for which there is no competition? You have in pigeon pea, I may have in paddy, or I may have in other crops. So we are also wanting that we create a culture where innovators promote each other by becoming franchisee, and there is an incentive for that. I mean, we are not asking just for pro bono. If you want to do pro bono, you are most welcome. But if you want to get a small commission for that service, no harm. I mean, anyway, any commercial partner, any commercial entity would give discounts to the people in the supply chain. I mean, any retailer, any distributor will give that to the fellow innovator. So I would suggest that our innovators also think along that line, that when they go into market, they can help, they can become franchisee of each other. There is also another model. I will give you an analogy. In South Italy, there was an automotive company, and they bid for, let's say, large order, say 1 million headlights or 500,000 headlights for car. Their capacity was only, let's say, 100,000. What would they do? They invited other manufacturers and asked, look, we have got this order. Till the order is completed, you're all like division of my company. Once the order is complete, we start competing again. So we cooperate and compete in cyclical manner.
Sir,
Yes.
sir, sorry to interrupt you. Actually, we had talked to Madhusudan Rao, sir, and somehow he has misplaced that he is having the lecture from 4 o'clock, but we had told him that it is from 2 o'clock. So by mistake, he has seen that it's 4 o'clock. So he'll join in half an hour, sir. He is asking to start the panel discussion, then he'll join.
Who? Madhusudan?
Yes, sir. Sir,
No, but 4 o'clock we cannot have, no?
Yes, that's why I said he's joining in half an hour. So he had asked if you can start the panel discussion session, and meanwhile I will join.
No, but Dipanvita will not join now, so we cannot do that. So I'm sorry. Please convey to him that we can have a short presentation by him at 2:30 or something if he can join in another 20 minutes.
Yes.
All right. Because we can't disturb the entire panel.
Yeah, he'll join in half an hour, sir.
So tell him that if you join at, no, half an hour will be too long. So if you can join at 2:30, please request him. Then he will have 10 minutes to share at least whatever he wants to say.
Yes.
Because I don't want to delay the next session.
Right.
So the session begins at 2:40. I would like that to begin with 2:40. Why should he suffer? Because of somebody else's delay. So please, please, continue. So one is the alternate.
If all the participants, if all the panelists are there, then the session can be, bit early.
But then Dipanvita will come only at 2:40. That's the point.
Oh.
Yeah, so we will have to wait. I think it's okay because our experience, as you know, with Pen is very rich. She was one of the first to set up the ICICI Knowledge Park.
Yeah.
So therefore, we will wait for her, and she would join little late, but once you begin with your introductory remarks, and she might follow after that.
Sure.
If she doesn't join by 2:50, then we will start the panel discussion and presentation by the participants. Okay. So let me go to the next model. One is the alternate model. So one, you have a cycle of cooperation, then you have a cycle of competition. You can do simultaneously competition and cooperation. So I'll give you a historical example. In early 18th century, in Lyon city, France, there were silk loom owners. And they knew that the demand of silk is fashion conscious. So when your demand is going up, demand for my product is going down. Not everybody can succeed at the same time. They were all in the same space with different designs, of course. So they had a challenge that they wanted to train their children as apprentices. And they would like to get the children trained by somebody else. So when it came to learning space, they cooperated. Nobody will say no to each other's daughter or son to be trained. But when it came to market, they were competing. So you can have simultaneously in knowledge space, Silicon Valley, what did they do? People will meet in the evening in a bar and talk about their problems, and they did not hesitate. Which company they are from, they didn't just think about, oh, why don't you try this code or that solution or that solution? And they help each other. That's what I would encourage our innovators to think about, that in learning space, we cooperate. In market space, if you're in the same space, same competition, same product, you can compete. I don't mind that. But let us not assume that if we are competing in the product market, we're also competing in the knowledge market. That's not the way any ecosystem grows. So we can have simultaneously cooperation and competition in different aspects of our life, and we should not hesitate in trying that. The last point that I want to mention, and then we can open it for discussion for some time, that when I want to promote my product or service, very often we focus on product. We forget about service. This is a problem that we find at grassroots innovators level. Majority of the grassroots innovators do not build a team for after-sales service. And the result is that they don't even, and I can tell you personally that there are some innovators with whom I've been pursuing this matter for 2 years or 3 years, maybe more, that make 1 minute video at which point lubrication should be done, at what frequency. No, it's very difficult for mechanical innovations also. So what I'm suggesting is that after-sales service is very difficult for each startup to set up oneself. So can we think of a common company owned by, let's say, 10 startups in same space, which will serve the clients of each one of them? So there's a separate after-sales service agency. You must have seen many companies like Godrej AC and other people. They have a panel of repairing people whom they, even Maruti did that. They will train them. They will ensure that they use original parts. They will give them proper standards and everything else, and then get their work done through them. So either you can have a common service agency, or you can have third-party agency which will service your product, which will test your device, will check whether there's anything, whether the handling is wrong, whether the device is defective, and then provide on-the-spot solution to the customer. So this is another important part, that we are trying to do everything ourselves, and the result is that we are not able to do anything outstandingly well. We are able to get some business. I'll give you an example. There was a student who had young scholar, who had developed an ECG machine, a small card-sized ECG machine, this big, which had a sensor on the side, and there were two sensors to put your thumb if you wanted to do a single lead ECG, and if you want to take at different points, you have to put for 15 seconds that device at different points in the heart, and you will get the reading. Now, that fellow went on changing the design, with the result that holding it or putting some pressure on the sensors became more and more clumsy and difficult, and so on and so forth. Now, to get customer insight about what is the preferred way in which they would like to handle a device is very important. Very often when we design devices, we do not pay attention to the way customers will handle them. And when customers handle them, they would like to have certain conveniences, and those conveniences are very important that we should recognize. I'll give you an example. Yesterday we had Ravi Prakash from NDRI Karnal, who has got many awards. You know that you were part of the BRICS committee, which gave him an award for BRICS for this cooling bucket for milk. Now, he has got many other awards afterwards. Good. Now, he wanted to develop a system where the whole bucket will be hired from the collection center, cooled, charged, and cooled with a phase change material. Milk will be collected, given back, then the milk collection center will have to clean up all the buckets. I asked him a simple question. I said, why can't you make a small device which can be attached to my bucket? Then cleaning the bucket is my responsibility, not yours. You only take care of this landing with the device, or even I can buy the device. So the question is, when you try to make a complicated supply chain, I don't blame him because he's not trained in this discipline. But what I'm trying to say, look at from the customer point of view. And can you handle all the complexity of issuing the bucket, cooling bucket, collecting the milk, cleaning it, then moving it again next day? How many staff members you need, how much of overhead you will have? It will become non-viable. So we should think about, how would customers use my solution? How would they like to use? By the doctor, hospital, patient in this case. And if the patients have to use it, what is the convenient mode? What are they familiar with? Can I do, supposing I have a device like Holter device. Now, many of the patients may have used Holter device for monitoring their heart. If you have a similar solution, many times what people do, they make the whole device to be held where the readings have to be taken. No, you can have sensors here and the device can be in my pocket. That's the lesson we should learn from the Holter. So many times the students, and I'm talking of particularly the life sciences students, because they have not been trained in the customer insight. They have not been provided those tools by which they can do that. So many times they may do mistakes which will affect the delivery or the success of their solution, your solution in the marketplace, for no fault of yours. I don't want to blame you. So therefore, what do? We should either connect to the management students in the management institutes nearby your lab. That's one policy solution. So they can do the customer, they can do market research. What Dr. Vandana Patrawale was mentioning in the earlier session, that look, we have not done market research in many cases because we are not trained in that. We are biologists, we are scientists, we are technologists. So then what's the harm in taking help of those who can do this? But spending time with the potential user is a very important point. I mean, those kind of discussions, I think you cannot delegate to third party. If you do that yourself, you will find it very, very meaningful and helpful, because that will give you the advantage of learning firsthand as to what are the features, form, or function customers want to leverage. Not everything is given the same value. I have seen many startups which try to add too many features. Even you know that some of these innovators try to bring far too many solutions. I mean, I was reading about the 25 devices that in the kitchen, the student that you have awarded this time. Now, it's an interesting idea, very interesting idea, but do I need all of them to be integrated? Do I use them all at the same time? Why integrate them? I mean, surely when I have to grate onion or I have to grate something ginger, that is a 2-minute job. Why should it hang around by rolling pin or chakla melon when I'm doing that thing? So many times we try to, I mean, it is all right for innovator to think like that let me put all the integrated solutions at one place. Which is I don't blame that person. But we have to mentor that. Look, what is essential, what is non-essential, what is frequently used, what is not frequently used. And then try to decompose and reassemble the solution so that the essential part remain at one place. The non-essential part remain a little distant because you need space, you need convenience. So I'm referring to an innovation which Dr. Vikin and his team had given Inspire Award just a few days back, I think on 8th or 6th. And this student had developed a multi-purpose kitchen device where lots of things could be done together. They were all integrated into one, so to say, table. And I appreciate the imagination of that student. That's wonderful. I forget the name of that person, the kitchen thing. There was a girl, I think, who developed that. That inspired body. Yeah. But nevertheless, it was a good idea. But I think too many features were present. So I would like all these startups to think, do we need all features in the version 1? Number 1. Do we need all features for different customers? What doctors can handle, not necessarily what patient can handle. So if I'm making a solution for the patient, keep it simplest. My father, when he was alive, wanted a cell phone, but he will not like to call anybody. He wanted to just have a phone which will receive. And if you want to make a call, he will not make a call. He will just press 1, 2, 3 button. 1 button, elder son. 2nd, middle son. 3rd, younger son. Now, it is our responsibility once we get a signal that we should call back father and talk to him. Can we have such a, do we have such a cell phone? But that's the one which most people, most elderly people of his generation, 90-year-old he was, need it. They don't want to get into the keyboard. They don't want to have calling numbers. They don't want to have to remember anybody. They just want a button for their 3 children. The rest of the thing they should do, and we should do. So I'm saying keep simplicity. If it's an elderly person for which you are developing a solution, that has to be very simple. If it is a young person, maybe a little more complexity. If it's a young child, even more simple than the elderly person. Maybe children can learn, but still complexity affects them. So my suggestion is that we will try to not put too many features in our solution. Too many problems we should not try to solve at the same time. Solve one problem well, develop a manual, develop after-sales service solution, provide a simple poster to the user if it is a customer, if it's a patient level solution, what are the things that patient should do, use color codes rather than only words. This button is for this purpose, this is for that purpose. Simpler things, easy to understand, no confusion will make the product more successful. And I'm sure when you look at you as a consumer, you will find the same thing that I'm talking about. Nothing very new, nothing very different. It's just that we forget many times that when we are designing solutions for the people, we forget that we are also among the users. And what do we prefer? You are scientists, you may handle a lot of complexity. Patient is not a scientist and doctor is not a scientist. They want simpler solutions. So I'll close here and open it for some comments if any one of you has before I hand over. Maybe in 10 minutes, we can spend some general questions that you may have, any one of you, as to how do you really, beginning with the customer insight, how do we talk to the potential user in the beginning of our research, beginning of our solution? What kind of constraints define the domain in which our users are going to use the solution that I'm developing? Depending upon whether it is for direct user, that is patient, is it for paid doctor, whether it is for hospital, whether it is for pathology lab, depending upon for whom am I developing my solution. And what are the current practices they have? How much will be the learning cost for them to use my solution? Is it easy to understand and learn? What are the possible errors that can happen? Have I given them a FAQ for ways in which they can fix these errors? Is there a manual given along with the solution? Who are the vendors who can supply the spares if they are required for my device? Where should they buy it from? Should they search for everything? They should come to me and increase my load of servicing? Do I have a distribution network or supplies after sales service network? So I will stop here. Any one of you have any comment? You can ask those questions. And then we will like to learn from the NIF's experience in encouraging both. They have done a lot of licensing. They've also developed some direct distribution, both open source and commercial. And I don't think we should distinguish, we should restrict ourselves to commercial development of our product. Business doesn't mean only commercial. Business also means social business, where we will provide solution to a large number of people so that they can then diffuse it in their communities. And that's also a good solution. So anybody has any comment or question?
Just I want to share one, the small thing. In fact, the incident happened yesterday itself. In fact, what you are saying exactly the same thing. We were procuring 60 laptops for the Inspire Awards, students, those awardees. And as per the procedure, we have opened the bid in GEM. And now GEM is promoting the startup companies, and one startup from Kerala was the lowest one, in fact. And we had no choice except because it was eligible, product and everything. So we had no choice except giving the order to that person actually. And they were also happy that they got the order of 60 laptops. So the founder of that startup company called immediately. In fact, he said that the cost of one laptop is 58,000 here, but you are the academic institutions or the research institutions. So I do have two options for you. I can revise my quotations. In fact, actually at that time, two options are you are having. One is either you permit us to install Linux, as the Linux is open source software, in fact, and you would be able to save $120, out of it. So your cost will be drastically down. And he has given another option. I said that the Linux may not be, friendly for large number of school students. They need the Microsoft soft-based, system. He said that another option is, if you buy the license from Microsoft, it would be just $15 to you, but for me it is $120. So you would be able to save $105, in fact, on each laptop. So that is also a big cost. I said that's fine, in fact, and he has connected us to the Microsoft person, and they agreed to give us 60 license at 15 rupees per license. So we would be able to save approximately $6,000
roughly.
Roughly $6,000. So the thing is that there was no effect on him, in fact, but they want to cater the need of customer in a better way, in fact, and they're giving the option so that the customer should remain satisfied. So with that gesture of that person, in fact, I'm happy. Then definitely I'll share that story to many people, and others will also include that in their
mention the name of that company. No harm in mentioning the name of the company.
So the company,
that everybody know about that.
It's Coconix actually.
Coconix, NIX.
Yeah.
Well, I would like all the startups here to really learn a lesson. Beautiful example.
PPP model of Kerala government and some private, and they're the startup company actually.
Very
so
nice.
Yeah.
So this shows, what you have just illustrated shows that when you have a empathy for the buyer, when you understand the needs of the buyer, when you understand the characteristics of the buyer, and you can offer some better deal to them, don't hesitate, because, for example, we know that GST for R&D institution is 5% on many solutions, whereas for others it is 18%. Now, if I am a part of the R&D system, and if I can get that lesser GST, tell me that. I don't know that. Don't take advantage of my ignorance. So I think this is a good point, that when you are serving customers, the education of the customer about solution, building their capacity, helping them to get advantages of the various policies that government has or country has, is a good thing, so that they can get a better deal than what they may be getting otherwise. I really find this example wonderful example of developing a customer-centric, customer-oriented, and customer-first approach in our business, way that we can help them to understand. But many times you must understand that customers may not know about this. So and these decisions may not be taken at the purchase officer. So I'm sure the concerned colleague arranged a dialogue between Dr. Vipin and the company CEO, That's why this deal could work out. So many times when you in your company, in your startup, somebody is not able to handle that. Dipanvita is here, so I'm very happy. Dipanvita, welcome. I was wondering whether you were going to get late, but I'm happy that you are here. I was telling everybody
Thank you so much.
I was telling everybody that you were the pioneer. I had visited your facility long time ago, many decades ago, and you were the first one which provided a common facility of wet lab to these startups. I mean, the startup movement had not even begun. The word startup didn't exist at that time. They were only simple terms that people were using about enterprise and so on. So I am very happy that you have been able to build this whole ecosystem. And I remember when we met again, when you were developing a report for the ecosystem strengthening, actually. That was the project that you were doing at that time. And many, many years ago, we met in Ireland. So I think all these students are very privileged. And we would like to hear you. And Dr. Vipin is from NIF. You know each other in some respect. You know NIF. So I would like you to begin. Dr. Vipin, shall we request her to begin?
Let Dr. Dipanvita begin.
And then you can proceed.
Yeah.
Dipanvita, floor is yours.
Okay.
And tell our students about some nuggets of your wisdom for how to deal with the business development issues for life.
Thank you. Thank you, Professor Gupta. I have to tell you, I had awards committee meeting of CSIR in the morning, 11:30 to 2:30. I had given my feedback, but then what happened around 11:30, I'm also moving home. And this is all about, I'm not telling you any story, I'm telling you about business development and what all you need to do. So what happened is we had contacted a vendor for moving, I'm moving houses. Part of my luggage is going to Kolkata because I belong to Kolkata. And some of it is moving to another house in Hyderabad. And there was a problem with the vendor that was packing. It turned out to be an issue. And I had to ultimately cancel that meeting. I wrote to the chair of that meeting, Professor Shivram from NCL, earlier director. I told the Professor Shivram, there's an emergency at home, I have to attend to that. And I will not be able to do the meeting. But I've sent all my recommendations and things. And I was hoping against hope that at least the problem gets sorted out before that 2:30, so that I can join this meeting at 2:30, which I had promised Professor Gupta. And why it is important for a person who's an entrepreneur at heart to be nimble and to be able to do things as and when required. You cannot have plan A and not have a plan B and think that everything will go the way you have planned. Today, I had planned my day in such a manner that I will first attend to some of the packers, and then I will do the CSIR meeting, then this meeting, but it didn't go that way. There was some trouble. And so 2 things that I would like to tell the entrepreneurs, the young entrepreneurs here, and this is a very, very live example. One is you should be nimble enough to be able to think of plan B. You should do planning well enough so as to able to handle any crisis that comes. So I didn't know what the crisis would be, but it was a new vendor. So I made sure that I send all my recommendations to the chair of the committee by yesterday evening, so that just in case something goes wrong, the committee doesn't suffer. So it is about planning, pre-planning, even if you don't know. So there are some disciplines that's required when you are getting into something which you're doing it for the first time. Maybe you're doing it, the world has not seen it, or you're doing it in your geography for the first time. So there's an innovation component in it. And you are also kind of exploring. If you are at heart an explorer, my dear friends, if you are an innovator, then you need to be nimble. Keep in mind that things may go wrong, not fit in the kind of planning that you have done, and be able to juggle with multiple things at a time. And the fourth is not to panic. I was actually very worried about this session because after 2 days, I actually confirmed Professor Gupta because I knew I won't be able to come at 2 o'clock. And I could have started panicking that, okay, I canceled the CSIR meeting. Now, should I cancel this? I said, no, time has not come. First, complete whatever, if you can complete. And my sixth sense said, I'll be able to solve the problem quickly. I actually delegated the problem. The other thing is you have to know how to delegate. So I delegated part of the work that I was doing. And because by nature, we have to be multitaskers. By nature, we have to attend to several things at a time. So I can't relax and say, one, I'll do everything myself. And two is I'll get nervous if I cannot handle it. Both are not tenable. So what you have to do is you have to have people on your side. If you have to have friends on your side, you have to have people you can depend on. So in a business that you're driving, in your daily work, in Hindi we say, in Bengali we say log bal, in Hindi it will be log bal. So you have to have people behind you to do it. Even if you're doing akla chalore, even if you're doing it alone, the idea maybe you're pursuing alone, but you should have people around you to back you up when in need. There will be ups and downs in anything that you are pursuing, and not this a simple thing like what I did today. It may be there are valleys of deaths, there are ups and downs in the lives of entrepreneurs. The most important thing is being able to keep your cool, think ahead, plan, and do your work. I'll stop at that.
And be ready for crisis. Because
Yes.
I remember when Bipin was organizing, he was doing the most work. I was hardly doing anything. Festival of Innovation at Rashtrapati Bhavan, and there was a rain in the night. And the whole pandal went away. Now, 8 o'clock, Secretary to the President will come for inspection to check, and they will have to issue the security certificate at 8:15 so that President can come at 9:30. 1 hour before, they have to issue the certificate that it is safe. Can you imagine what would have been done in that day and the night? But he managed everything. I remember he managed everything. Everything was perfect. Morning, the president, honorable president came and everything was done. Certificate was issued by the security staff. All the things held in order. But as you said rightly, one had to be ready. You can't panic. You can't lose your nerve at that moment because things are seemingly going out of hand when the whole thing is disintegrating at around midnight. And what do you do now? The time is limited. Options are limited. But the contractor was very good and everything worked out. I will never forget that morning and the way he managed the whole thing. So your advice is very, very meaningful that there's nothing gained by panicking after all. Things will go wrong.
Things may go wrong. I'm not saying it will go wrong. It may go wrong. And you have to have kind of a plan B in mind. You need to have friends who are, and if you are sincere, honest, and do your things, you're diligent, people will understand. And that is what.
Thank you. You have related, only you could have done this to link the tension on your head, and still you are here. We really appreciate all the GYTI awardees. Appreciate Bipin, floor is yours.
Sir, thank you very much. Dr. Dipanvita, you have given a very beautiful message. In fact, you mentioned that you need not to be panic. Life is always in uncertainty. So nothing is certain in the life, but it doesn't mean that you will be panic. But when the people are behind you, then definitely you get a different confidence. And like the professor sir has mentioned about that Rashtrapati Bhavan's festival of innovation and entrepreneurship. Always, it is not me. In fact, it is all the colleagues and the collaborators. When I asked them that, "Ho jayega?" They said that where is the problem? In fact, by 4 o'clock it would be ready. So if it is not at 4 o'clock, by 6:30 it was ready. In fact, and at 8 o'clock when the visit from President House was there, and they said that, oh, last night we were thinking that whether it would be ready or not. But when the determination was there of the people, in fact, and all night people were working, they were dedicated, they were committed, and definitely it was not surprise for us also. Thank you very much thank you very much, Srishti and Bayrek, for giving me this opportunity, in fact, to share my views here. In fact, I know that GYTI, the journey of GYTI, also came from uncertainty. How much pain Professor Gupta has taken, in fact, how many times he has done the advocacy for the support to the individual. In government, it is very difficult to support the individual, but in GYTI, in fact, he was fighting that why not a good idea get the incentive in terms of funds, in terms of grant and the mentoring support. So what exactly we did in GYTI? So I am the witness of every step of GYTI, and I'm happy that lots of startups are coming out from the GYTI awardees, and they're the cutting edge technologies. That time we supported the college students, the university students, and mostly Professor Gupta was not supporting to the faculties. His more intention was to support to the students. So now those students have become the companies, and those companies are doing good. In fact, we are happy with that. So if you see that how many startups in India, if you see the last statement in Parliament, so 42,733 startup companies are registered under DPITT. And if you, in one document of CII, I found that one startup company has generated the job for 12 people. They have generated 12 jobs. So 4.6 lakh jobs startup companies have generated till today, and out of that 4.24 lakh jobs were generated during last 3 years. I'm happy to share that it is not some states, but all the states and union territories are having the startup today. So it might be the policy, it might be the the support, it might be the people like Professor Gupta, or different other faculties or different other people, in fact, who are taking this initiative. And these startups are there in 590 districts, and 45% startups are there in tier 2 and tier 3 cities. So which is also the achievement. It is not that the startups are there in metros. In tier 2 and tier 3 cities, startups are also there. So though we have done the startup investment of 5,089 crore under that fund of funds, but that is not sufficient. In fact, more and more funding is required, in fact, at every stage, from the from government sector, from different schemes, from different banks, so that many startups companies should not go to Chinese investors, or to those investors which are not reliable, and those investors which will acquire the companies, in fact, in future. So we want the startups should also grow. They should also be in in better clubs, and but it should not be that the person, in fact, who started that venture should get obsolete after few years. In fact, we need both the things, in fact, that we have to be careful. So today we will listen the experience of different people So there are 4 people, in fact, in this session. Basically, we came to listen them. One is Vaibhav, Jayesh, Prem, Daxin, and Nitin Vasant. So we'll start. We'll request them.
And Ravindra also.
Okay, Ravindra also, because there was
Ravindra Gaikwad. Just add that.
Gaikwad from IIT Madras. Yeah. So now we'll invite the first speaker, who is Vaishnav Shinde, in fact.
Vaibhav Sitole.
Vaibhav Sitole. Vaibhav is from Naipar, Ahmedabad. So Vaibhav, now the floor is yours.
Yes, thank you, sir. Good afternoon to all. So I will start my screen. I think my screen is visible to all.
Yes. Hello. So good afternoon, everyone. I'm Vaibhav Sitole. I'm the GYTI awardee, GYTI 2020 awardee, and I'm the CEO and co-founder at IOTA Design and Innovation Lab. I would like to thank GYTI and BIRAC, which who have provided that grant support. That's why I have formed this venture, and I'm able to start this my idea to get translated in for the society. So the GYTI has awarded me GYTI has awarded for the invention of a user-friendly do-it-yourself blood microsampling device for diagnostic and pharmaceutical application. So this is the schematic of this device we have developed in our laboratory. So this is a blood microsampling device that will collect 2 drops of the blood from the remote location. We can say that the home sampling, and after that sampling, we can transport or ship this device to the centralized lab. We can say that the pathological lab or CRO for the specific analytical monitoring. Okay, so what? Slide is changing. No.
Please go ahead. Changing. Yeah.
Yeah, I think some issue with some.
Slide is not moving.
Okay.
You are on second slide.
Just wait, I will share this. Okay.
If you have a difficulty, we will call somebody else. Go upward. Maybe that's it.
Yeah. Vaibhav, it is there. You are on 4th, now 2nd. Move.
Yeah, I think some issue with that. Yeah, I will just orally explain this, what you have is that problem. So yeah, blood sampling is needed for means any primal monitoring, like you can say for that health monitoring or health monitoring. So whatever is the current sampling are available in the current setting, like venipuncture, people are taking 3 to 4 ml of the blood from the venous puncture. Okay, so in that there are many people are terrified with that sampling because phlebotomist will collect 2 to 3 ml of the blood from this venous puncture, and it will be hectic for newborns, some of the pediatric and geriatric patients. And WHO have come up with the guidelines. So for means people have to collect only 3% of the whole blood from the for the diagnostic and various clinical application. But when we go for the frequent sampling, we can say for some clinical research, some in therapeutic, some in new drug discovery and development stages, that in that stages we require frequent biofluid sampling for that pharmacokinetic and toxicokinetic studies. So in that case, that whatever is the current method are available, it will be hectic for that patient. Also, in current method, we require cold environment for the transport or shipment of the sample from remote location to the centralized lab or pathological lab. So to overcome this problem, dried blood spotting on paper card, it is excellent alternative to the conventional blood sampling. But it required only 2 drops of the blood in one paper card. It having stability, we can preserve that sample for long term. We can say that monthly or yearly, you can preserve sample at a room temperature. but it having its own limitation. Like when we go for that quantification of any drug or quantification of any bioanalyte, it will be need some technical processing for that quantification, and also some kind of this cross-contamination biohazard issue will be happening with the DBS method. So here we have come up with that user-friendly dried blood sampling device based on capillary blood sample collection, which is from the finger pricking. So this is a video. I will just. I think video is also not able to see. So just we have developed that device in our laboratory. Right now, prototype is ready, and we have tested with some of the pharmaceutical drug and some endogenous bioanalytic monitoring. We have got some promising result with some pharmaceutical drug. We have tested for the caffeine monitoring, naproxen monitoring, and right now we are moving with the metronidazole monitoring of this drug. And we have also tested this device for some endogenous bioanalytic monitoring like cholesterol, GYTI, creatinine, and blood sugar. And right now that our product at the prototyping stage, and in the next 1 to 2 months, we are moving for the clinical validation of this product. And after that, we can market this product for that. Initially, I'm expecting this product to market for the domestic purpose, like for the clinical operation, like in CROs and pharmaceutical industry. That device, that kind of the things is required for the pharmacokinetic and toxicokinetic study. And this device will require only 30 microliter of the blood for that. That will collect the blood from the patient at a remote location, and we can send ship this device to the clinic centralized lab, or we can say that pathological lab, for the monitoring of this whatever is that bioanalyte. And that device having its characteristics that it can preserve that collected sample for long term. We can say that monthly or for years also. So we can use this device also for the genetic sample preservation also. So I have prepared that slides, but it is not visible to me. So this is just.
Okay.
Yeah. So these are some recognition I have awarded, that GYTI 2020 award, and some are the publication who have published with that related to this project. So yeah, this is from my end. I would like to thank GYTI and BIRAC because for securing this grant. With this helpful support of this GYTI and BIRAC, I'm able to form this company and able to develop this product up to at a prototyping stage. And I would like to thank Startup India for recognizing these are the startups, and I'm incubated at a Cradle EDI Ahmedabad. So I would like to thank to Cradle also for supporting us as for incubation support.
Thank you very much, Vaibhav, for wonderful presentation. So is it already in the market, or it is at the product development stage?
Yeah, it is at the product development stage. So
Okay.
right now we can say that these are TRL level 4 to 5.
4 to 5?
Yeah.
Oh, for how long we have to wait for this technology?
4 to 5. We have to clinically validate this technology because it is considered as an in vitro medical device. Okay, so for that we have to validate this device with enable accredited lab.
Okay.
Yeah, so we have tested this device with LM College of Pharmacy here in Ahmedabad for some pharmacokinetic drug monitoring, and after that get promising that data, we can move for the clinical validation with some enable accredited lab.
Okay, so have you already identified the lab where you will, go for the clinical validation?
Yeah, I'm just negotiating with some of the CROs here. So one of the CRO is available here in Ahmedabad, Vida Clinical Research.
Okay.
And he has also tied with the BioNits Bangalore. So they are dealing with the clinical validation and testing of medical device.
Okay, thank you very much. Any other question? Dr. Dipanvita, you want to?
No, thank you. It's a very nice presentation, Vaibhav. So just wanted to tell you, this is a very important area, and there may be mid-size medical device companies already in India who may be interested, and they can even do part of your clinical validation. Have you contacted anyone?
No, ma'am. Right now, I'm just focusing on that. Right now, we have developed that design development. We are developing that prototype only. Right now, I'm exploring this device for exploring in the market. I'm started that explore.
Okay, and because what happens at times is while your need identification has been done well, some nuances and fine tuning are essential, and that would come from people who are actually in manufacturing similar devices. And in case you want, we can have a separate discussion. I can connect you to some people with Professor Gupta's permission.
Yes, sure, ma'am. Sure, ma'am. I would like to.
By all means, that will be most helpful.
Yes. Thank you.
Yes.
Can I add something here also?
Yes, sir.
I was just wondering, since this is a kind of the COVID time and you're collecting the blood sample,
yes.
So you may want to look at its the collected samples compatibility with antibody detection. Right now you're looking at pharmaceutical kind of a thing and also some blood parameters.
Yes.
And you said also SGPT, SGOT kind of a thing, which means that your sample collection safeguards the proteins inside the sample. That means probably the antibodies also can be safeguarded. So probably you can also look at its the sample, that micro sampling methods suitability to antibody, the extent of antibodies in a sample. So in addition to the parameters that you're looking, you can also keep looking at how you can make it compatible for that application also.
Yes, sure. Because sir, initially I'm focusing for that pharmaceutical application, but it has means wide an application for diagnostic, some kind of the genetic sample. We can also extract DNA, RNA from the DBS spot also. Okay, so we can diversify this technology further for further biomedical application. So, but it has to be needed some clinical validation for that specific application.
Yeah, if you need any help there, we can do that, because we are. I should introduce myself. I'm from CCMB in Hyderabad.
Okay.
We could help you in, at least getting your beta testing of the sample, because we have a lot of collection of these samples over a period of time. So it's easy for us to give you a report on the efficacy of the samples.
Okay, sure, sir. I will connect to you offline. Okay, maybe you are saying that device, that this is the device, whole device that will collect the whole sample, and you can see that this is the stuff. Okay, we can reuse this device for multiple sampling, and that when you are collecting that sample, that stub is collecting only 30 microliters of the blood. Okay, and after that collection, we can transport this stuff to this transport cartridge. Okay.
Understand? It looks very clean way of
exactly. So
hands-free kind of a thing.
It gives some kind of the flexibility and convinces during that handling of that sampling also. Whatever is the cross contamination biohazard issue, it is also avoiding in that aspect.
Great.
Yeah.
Very good. Yeah. Thank you.
We'll have to manage the time because.
Yes, sure.
Thank you. You are unmute, Vipin, unmute yourself.
Oh, sorry. I'm so sorry. So now I request JS Kumar from ICT Mumbai. Hear your presentation, JS.
Yes, sir. Sorry, because sir, I was associated with Coast Guard and there is a issues of oil spill, so I didn't get time to make a presentations and request to the GYTI people also. But I have the story where when I have started some sustainable businesses and I'm working with multiple people, I will tell you the story.
Okay. So JS, you please finish it in within 10 minutes.
Yeah, sir, definitely.
Because we have to start another session also. Yeah.
In 2015 or 16, that initially we wanted to start our entrepreneurship in the areas where we wanted to work for the agriculture also. So Professor Anil Gupta talk I heard in 2014, and I know that the impact of innovation is very really matters. So we work in North Gujarat and the ground level, grassroots level. We identify that most of the potato is wasted out on the roads, and the people are facing lots of the issues, and farmer is also not getting the right price of that. So we took the motivation out of it, and we have started working on the company. And right now we have the running company of 150 tons per day of the potato starch production in Gujarat, where more than 4,000 farmers is getting benefited out of this company. And again, we have identified some issues at the ground level, that the more than 30% people will be affected by the water scarcity by 2013. And most of the areas of the North Gujarat, where the water irrigation's problem is again the bigger issues there because of short rainfall. So we are now converting the starch into the super adsorbent, and we have developed the super adsorbent is having the water adsorption capacity is around 200 times. And its the cost is around 4 times less as compared to the market product, because market product UPL and all the product cost is 800 rupees, and our product is cost is 150 rupees, and we wanted to support the farmers as we can. And we have again we get the complaint from the farmers as that they again that they are excessively using the chemical fertilizer, where we now develop we utilize the waste from our company as well as local areas, and started commissioning the plant of 100 tons per day of biogas plant, where we utilize the waste in converting to the liquid fertilizers. And that liquid fertilizer we are selling is to that cheaper cost, around 5 to 10 rupees per liter to farmers, where farmer will get the benefit of the organic fertilizers, and they will partially fractions of their potato final product we are taking as organic potatoes, and that is also the benefit for our company also, and they are also selling the organic potato. This is the business model that we are working at the ground level. And I have done the work in PhD and also that scale up version of GYTI supported project of the human extraction basically. So I have the waste here, we have the collected and converted into the using the advanced oxidation technology and converted into liquid fertilizer, and protein, keratin protein having the cosmetic applications also. So this liquid fertilizer, we are mixing with that, develop the organic fertilizer, and we are again the cost breakdown from the 200, you can see that amino acid hydrolyzed, the cost is around 150 to 250 rupees, but we have to develop the technology, and I thankful to Professor Pandit and the ICT that we have successfully developed the technology, and we are going to scale around the 500 kg per day, that the cost of the final product in entire technology that reduction from the 150 rupees to 20 rupees only. So again, this is the innovation part and the need of the market that we are fulfilling through the technological innovations, where this is the entire business model we are I'm working with many people also. And this is something kind of Professor Anil Gupta sir said that our network entrepreneurship, we are the clubbing of 6 to 7 startup as of now and one big company also. So we are working together and try to resolve the problem at the scratch level. And I'm also the founder of the two companies, the Revoltech Technologies Private Limited and the Gilmon Research, and I'm associated with technical advisor of 6 startup only. So another segments that again this scratch level, the we have to identify the problem and resolve the issues of the milk stabilization during the transportations. So again, with support of ICT, we have the developed mechanical device using the cavitation technology, and right now the pilot scale trial is going in the Nasik, where inline partial pasteurization using the mechanical devices, where the refrigeration cost as well as transportation milk stability is definitely going to be higher. So we have tested at the lab scale, and we are now scaling it to a higher scale around the 1,000 liter trial and try to implement as soon as possible. And healthcare sectors, we are working on the smart gadgets like that smartphone. We're modifying the smartphone as a diagnosis tools, and that product is something like microscopic kind of things that we are going to launch in couple of the month, for the accessible for that diagnosis application also. And we have the successfully diagnosis dengue as well as malaria using only on the smartphone. And now moving in that area for the fluorescence microscopy to make it affordable, and targeting cost is around the less than 20 to 30,000 because, and now the ISC Bangalore, some prof is associated, we have the collaborator and we are working together on this invention. And I'm really thankful to Professor Anil Gupta, sir, ICT. And
amazing, amazing. I'm so thrilled to hear you, Jayesh. I have seen your journey over this time.
Yeah.
You have created you have become an incubator yourself, actually. You're helping so many other people.
Yes, sir.
It is magnificent. We have to meet sometime.
Yes,
and this is a marvelous story.
And sir, basically, I contact to Indian US-based Rockwell companies, that diagnosis scientists, Dr. Arvind Virmani, Dr. Lalit Vora, and there's lots of the outside people. They are the Indian researchers, but they are working outside. And I try to contact them, and right now we are working together. And thank you for supporting COVID-19 detection kit. Unfortunately, we didn't complete that invention, but right now we have in our hand where we can detect the virus within a 5 to 10 minutes and on site, and we are working on that technology.
Wow, that's wonderful.
Very nice, very nice. Thank you, Jess, for sharing the wonderful story. And you know, we are so happy to get that you have become the incubator. In fact, you are not just a startup company; you are incubating others, and this kind of hand-holding, you know, to the startups are required, because at that time they are new in that area, and you know, might be they need more than money, they need the mentoring support, and the people like you are the ambassador of a startup movement of the country in true sense. Just one query. In fact, you are utilizing the waste, the household waste or the industrial waste?
Sir, currently we are using our company waste, 3 lakh liter per day that we are generating the waste, and also we are utilizing household waste as well as cow dung and also human waste.
In the waste, there might be some toxic elements. So you are segregating before?
Yes, yes, segregating each and everything, because if going into land, it will be the bigger problem. So we are making surety that this waste is very good for our plant. So that's the thing that we are currently doing. And sir, we are inviting on their inaugurations our plant of biogas 100 ton per day. In future of around 8 to 9 months, we are going to start that.
Very nice, wonderful, wonderful, and congratulations to your initiatives. Yeah, thank you very much. So, Dr. Dipanvita, you want to add something? If not, then let me move to...
No, nothing in particular, except that, again, I have to repeat what both of you said. Congratulations. If I get a chance, when things become okay, I'll visit you.
Thank you, ma'am.
Thank you, thank you. So now from BITS Pilani, you know, Prem Dakshin. So Prem,
yes, sir. Here? I'm here, sir. A very good evening to everyone here.
Thank you.
Yes, sir. Sir, I'm having some net issues over here, so I'll just share my screen. I may not be able to turn on my video because you know, that may affect the audio. You're able to hear me, right, sir?
Yes, we can hear you.
Yes.
Yes, sir. Yes. So, yeah. So, yeah. Once again, a very good evening to everyone. So my name is Prem Dakshin, and I'm a GYTI 2020 award winner. And the project that I'll be presenting today is custom cam in medical ventilators. So, I mean, I just pulled in this presentation in the last moment because I really wanted to share this journey with you, because mine being a ventilator and the recent pandemic has given a lot of change to what we're doing. And we have literally redefined our goals, that what we have for the future from the learnings that we had to the pandemic. So saying that, I'll just start with the presentation. So it all started in 2019 when we and my mentor, we decided to work on our medical instruments and try to figure out if there is any existing gaps over there. So ventilators were one of the instruments we were working on, and whenever we try to gather, I mean, ventilators have seen a drastic growth in the past. So there's a lot of features available that helps a lot of technician to identify major things during the treatment. But one constant complaint that we received from different stakeholders was, they kept saying the ventilators were more bulky and more expensive. So this expensive part was not, giving a chance for ventilators to enter, hospitals in rural India, underdeveloped places where people can't actually afford to buy this high-end ventilators. And the second part is, and apart from these two, there was another third part which we understood is coming up with a rigid ventilator. So when we spoke with some biotech engineers, we figured out that ventilators are very sensitive instruments. It's not something that you can move from one block to another block or one floor to another floor every day that can affect the calibration. But we understood that if you could come up with one ventilator which is super rigid, like say for example you're in the middle of a disaster zone or in the middle of a warfare where you literally need to carry this instrument in your hand and you need to jump, climb and crawl, and you can still use it on someone who's injured in places like that. And it may not have all the features of a full-scale ventilator, but at least it would maintain the minimum PEP value and keep the O2 saturation in a safe zone till we can transport the injured person to a proper hospital. So when we looked at the opportunity, everything led back to the expensive part and the bulkiness part, and how people below poverty line can't get access to this expensive ventilators because they are not available in most of the government hospitals. So keeping all this in mind, I came up with this new mechanism that I call as the frustum cam mechanism. So if you see here, I just given a basic simple introduction, which is the hardcore mechanical concept. So this concept is similar to be more frank, I initially designed this mechanism for automobiles where we have this eco mode and power mode. So if you look over here, this circular part over here is called as the cam. So it has a center offsetted. So when it rotates, it hits the valve up and down, and the valve comes back to its position when the cam goes down due to spring action. So this diameter of this circle, it will determine like how long this valve will travel, which is called as the valve travel or valve length. So what happens over here is that, the part 100 in figure 3 is what is called as the frustum cam. So when we rotate this thing, this frustum cam is going to push the valve down, and that in turn is going to open up and, makes the air to flow towards the patient. So, but by providing linear motion to this entire mechanism, we can keep this point, we can move this point and place it at different levels over here. So this valve lift is defined by the diameter of the circle. Greater diameter is going to be greater lift, and obviously you're going to have greater flow. Lesser the diameter, we're going to have lesser valve lift and lesser airflow. So we use this in the same concept in automobile, but I modified the design and came up with this part over here that we are using in the ventilators. Except in automobile, we are controlling the air fuel mixture, and here we are controlling the tidal volume. So of course you can ask me, there are a lot of other things when it comes to a ventilator. So I do have another photo where I have shown how I'm going to integrate this to a ventilator. But since there's a time limit, once I finish, if you guys still want to see it, I can show you that. But another important part about this mechanism is that, this was actually derived from an automobile concept, which can actually run in, 8,000, 9,000, 10,000 RPM in different kinds of temperatures. So again, coming to the rigidity part, the ability to have a handheld mission with which you can literally move it, drop it, and still be able to use it on a person in an uncertain environment. So this mechanism is going to be a key part in help us to achieve the rigidity. So I proposed this solution for GYTI 2020, and I received the grant. But exactly after that, as we know, the pandemic came in. So after the pandemic, we initially knew that, we needed compact movable ventilators, but the pandemic clearly highlighted this to the entire world. So during the pandemic, I also got in touch with Bioness. I got in touch with a oxygen concentration manufacturing company here in Chennai, and I tried to speed up the process, hoping that I could come up with some system that could save lives. But unfortunately, I was not able to finish it on time. But of course, the pandemic has definitely gave us a lot of things to consider, and this literally, made us to change our views of how we are going to take it up. So after having a lot of brief discussion with our stakeholders, the first thing we thought is, the market is now highly saturated, because even yesterday, Dr. Renu sir was saying that, all the automobile companies and the defense all pulled up together to make ventilators. So unless and until that this ventilator is going to have more features, more than what I just told you, it's going to have a stand, it will never have a stand in the market. So we made a quick visit to a hospital, the SRM Group Hospital, multi-specialty hospital here in Chennai, which also has an attached MBBS college. So I had an opportunity to interact with different stakeholders like researchers, doctors, clinicians, deans, the people from management. And one new need that I discovered over there was, data collection and data management. So, let us say if there is one particular person who has a specific respiratory disease and he goes under a ventilator for 3 or 4 weeks. So the breathing pattern that he has in this 3 or 4 weeks could be a very valuable information for researchers who are currently working to find a solution for that particular disease. And at the same time, when I spoke with the doctors and the management, I understood they needed data like they really wanted to know the clinicians who are working on different ventilators, at what time it was switched on, when it was switched on which setting was it run, who was the patient it was used on. So these kind of details, they felt would be very much helpful if there's any product that could come up with. So that's when we decided to upgrade our system. Like apart from ventilator, apart from the features that I just told you, we decided to make it an IoT enabled device where this ventilator, apart from doing its regular purpose, it can also transmit data and store it. So I came up with a brief plan, and I know I had a brief explanation with the doctors. The first step was to go with a 1D data transfer, which is basically, collect the data which the researchers and the management wanted and store it in a remote cloud location, which can be accessed by the required stakeholders. And the future development that we had in mind was to come with a two-way data transfer, where the ventilator, if it notices there is some sort of breathing abnormality in a patient, it immediately notifies the concerned clinician or doctor in their handheld devices. So again, when I spoke to them about the future development plan, there was a big no-no from their side, because of course, I do understand there's a big cybersecurity involved in this thing, because we don't want a third party to hinder in the data transmission and tangle the data. And at the same time, here we are actually working on devices that's saving human lives. So trying to bring a huge change and bringing new technology into it, I personally found it was very difficult to convince the doctors. So one advice that I got from them is to get some data and work on them. Maybe get some prediction model or an ML model, deploy on it, and get some results. So that could be like an initial level proof to sort the problem. So now, I had the GYTI grant, the pandemic happened, I had the new set of goals. So again, after a lot of brainstorming sessions, we decided to go with our first prototype. Again, this being the first prototype, we decided to have some checkpoints that this needs to satisfy. And first and foremost is to act as a proof of concept, because the mechanism that I told you, I do have a lot of designs and simulations, but I don't have a complete working model with control systems. So that could be the first thing. And secondly, again, coming to a ventilator, there are a lot of features like PEP, O2 saturation, trigger values. So after consulting with some doctors, they told me to take the initial parameters of breath per minute and tidal volume and achieve these using these mechanisms. So once this is achieved, I can keep adding the other features to it. And the third big change that we had was to swap our control unit. We swapped the Arduino boards and got in Raspberry Pi, because we decided to make this an IoT-enabled device. And for this particular prototype, it's not going to be an IoT, but we still use the Raspberry Pi device, because in the future, when we are like converting this to an IoT device, we can just merge the algorithm or modify the existing algorithm. So if it is an Arduino device, then we need to redo the whole thing. So after speaking with IoT experts, we got into this Raspberry Pi. So considering all these things, you can see on the right side the different designs which we worked out. This was the first one, which was very huge. We had some brainstorming sessions and we fine-tuned it to this one over here. And further, we had more of fine-tuning sessions. And as we were developing the design, we came up with new ideas on how to mount the cam with the camshaft. And if you notice, there's a lot of rubbing happening at this point. So if this part gets worn out, we again came up with an innovation of making this a brass part with threading, so that if this part gets worn out, now you can just screw it out and make a new one and push it in. So after doing all these things, finally we came out with our first prototype design that you're seeing on the screen. So we have the entire frustum cam mechanism that is controlled by two servo motors here. One, which will be rotating the frustum cam and controlling the breaths per minute. And the other servo motor, which is attached to a threaded cam, will move this entire structure to the right and left with the help of this railing. So that will help us achieve the tidal volume requirements. So I do have the design, the basic dimension. Of course, due to allowances, there is minute differences here and there. And the entire control unit, including Raspberry Pi, the GPIO extension, the hat for servo motors, everything will be going inside this block. And another idea that we came up recently, but we're not sure if we'll be able to do it, is to make this as a desktop model. So if you can notice, we can remove the side panels off and people can actually see what's happening over here. So we are planning to put glass over here, 3 sides of metal and 1 side of glass for the valve chamber, so that people can actually see the movement of the valve. So this could actually be as a POC as well as a desktop model for people to understand. But however, we are still working on it and not sure how it's going to work out. And finally, this is the fabrication that has happened so far. The cam has been done, the main frame. This is the camshaft with the sleeve. This is the stepper mounter molds and the molds for the cams. The main mold for the cam, and here I just kept everything inside the main case just to give you an idea of how it's going to look. So when this photo was taken, the mounts were not ready, so I was not able to do it. So most probably by this weekend we will have the lower half assembly, which is basically this portion, will be done by this or by the maximum by the end of next week, and then we'll be working on the upper half assembly, which is basically the inlet and outlet and the valve chamber, and then we already start working on the coding part of the Raspberry Pi for the control unit. So that's it on my side, and thank you so much. I just want to thank GYTI and everyone from BIRAC and Bionest who really helped me out, in achieving this. So thank you so much.
Thank you, Prem. We understood that it is a prototype stage and you have to go a long way, in fact, for the validation of this device, and you have to get lots of approval. So best wishes to you for your future.
Yes, sir. Thank you. Thank you very much.
Dr. Dipanvita, you want to mention something?
Nothing in particular. Again, sir. Maybe when we wrap up, there is time, we can have a few questions.
Okay, fine. So now the next speaker.
We have 2 more speakers.
We have 2 more speakers.
Yeah, so I thought we'll just finish hearing them.
So Nitin Vasant is from Cochin University of Science and Technology. Nitin?
Request them to reduce the time of instead of 10 minutes, try to do it in 6 minutes.
Nitin, are you there?
Nitin, unmute yourself. Nitin is not able to listen, so then you get Ravindra.
So now we'll move to Ravindra. Let us see that if Nitin comes back. Ravindra is from IIT Madras. Ravindra?
Hello. Can you hear me?
Yeah, we can hear you.
Yeah, I'll share my slides.
And Ravindra, please, finish it within 6 minutes.
Yeah, I'll try to finish it within 10 minutes. So, can you see my slides?
Yeah, we can see.
Okay. So, good afternoon. So, myself, Ravindra Ekwar. So, I have received this GYTI Sitara Award last year. So, I'm working under Professor Ashish Kumar at IIT Madras. So our main problem is to diagnose the sepsis. So as we all know, sepsis is a life-threatening condition when body's immune response to an infection damages its own tissues and organs. So if you see, anybody can lead to a sepsis condition. So there are like 4 common infections that basically leads to the sepsis, which are lung infections, urinary tract infection, gut infections, or skin infections. So these sepsis has different stages. So first is like sepsis. If it is not treated on time, it can lead to a severe sepsis. And also it can lead to a septic shock and then ultimately the multiple organ failures and the patient may die. So it's very important. So our main objective is to build an instrument prototype for accurate and reproducible time point of care measurement of gasotransmitters. For validating the prototype for sepsis and cyst model in animal as well as in ICU patients. So if you see the motivation, if you see the statistics globally, so there are around like 27 million people every year that develop sepsis, and out of 27 million people, 8 million people die because of this sepsis. Not only that, if you see the maternal deaths, so the main cause of the maternal death is again sepsis. So that's why it is a rising global and socioeconomic burden because of the hospitalization or maybe the ICU cost. Not only globally, if you see the India as concerned, like 1 in 3 patients who die in a hospital, that is because of sepsis. And this number is every year it's increasing. So you can see there are some reports which says like there is like 10% rise in sepsis cases in every year. So if you see the main concern is here is like out of all the sepsis deaths, 80% deaths can be prevented by early diagnosis and therapy. Not only that, if the current pandemic, which is caused by this coronavirus, if you see these viruses attack our lungs, and because of that, the infection happens in the lungs, and that can lead to a sepsis condition. So the literature suggests there's a strong link between the death from the COVID-19 and the sepsis. So this is again another motivation why we have chosen this problem. And then this is the need for rapid diagnosis. So in sepsis, if you see, the time is very important. So we can see here in the graph, the y-axis shows the patient survival rate. So as the time passes, the patient survival rate decreases. So almost like if you see the statistics, there are 10% increase in the death, the risk of death up like every hour gone. So it's very important to detect the markers, whatever we are targeting, that should be detected at the early stages of the sepsis. But if you see the current technologies, which are based on different markers, that is IL-6 or PCT or CRP, so those markers at the early stages of sepsis, they are at very low concentration. When the sepsis develops, at the latter stage only, we can say that there is a increment in these biomarkers. And if you see here, there are like the current technologies which are based on these biomarkers. So basically, they misses this critical period. So if you use these markers, if you use these technologies, they give the results after this critical period. So we'll miss the golden period, and then the patient survival may get reduced. So it's really important to have real-time and in situ monitoring of reliable sepsis biomarkers, which can save lives of many patients. So here we are proposing gasotransmitters as a biomarker for sepsis. As the gasotransmitters are small signaling molecules which are generated inside our body by different enzymes. So these are like H2S, NO, or CO. So basically, there is a strong literature which suggests these gasotransmitters participate in systematic inflammatory response syndrome, that is sepsis, and could be used as an important indicator for sepsis. So as an example, you can see here as an example, you can see here the H2S. As the H2S decreases or increases, there is a physiological changes inside our body happens. So as there is a normal level of H2S, as it increases, the patient may develop a septic shock, or as the H2S decreases, there might be a chronic heart failures. So it's important to have these biomarkers at the normal level. So by detecting those, definitely we can predict the sepsis, and it can be used for a prognosis of sepsis as well. So here we are proposing a microfluidic-based technology. So those who don't know what is microfluidics, so basically microfluidics is a technology where we deal with nanoliters or picoliters of liquid or fluids in microchannels or microstructures. So basically what we are targeting is a bedside system where we can continuously monitor this gasotransmitter from the patient's blood. So you can see here, if a patient is going through an heart surgery, so there will be an ECMO machine. This is just an example. So the blood coming from the blood circulating within the ECMO machine, that can be given to this system, which is at the bedside. And then in the system, we have different modules. The first module is blood plasma separation module, where we'll separate out the plasma and the blood cells. So as we know, we are targeting these gasotransmitters in the plasma. So it's important to remove the blood cells. So here we have a first module, which is based on acoustic-based blood plasma separation. So basically here we have a microchannels where we have attached the piezoelectric material. So by actuating that, we can separate out the plasma and cells. So once the plasma is taken out, which is given to a second module, which is mixing and incubation module, where different chemical probes will react selectively to these gasotransmitter, which are present in the plasma. And after reaction and incubation, they produces a fluorescent signal, a fluorescent compound that will be given to a next module, which is optical detection module. So where we have incorporated different optical components, such as the optical fibers, lasers, or detectors. So based on
Ravindra, you have to move faster.
I'm just about to complete. So this is Once we have detected these gastrotransmitters, it will be desled. So basically, this is really important to continuous monitoring, and this enables us to like detect these gastrotransmitters in continuation and real time. So this is just experimental setup, or can you say the experimental images what we have in our lab. So basically, if we talk about the current status of the technology, till now we have detected H2S, H2O2, and in our device. As we see here, this is the blood plasma separation model. So as the plasma, as the whole blood is covered within the channel, when the acoustic is off, when acoustic signal is on, or we actuate this piezoelectric material. So because of these acoustic standing waves, we can focus these cells, and then plasma will be taken out from the different outlet, and then cells will be taken out at different outlets. So basically, till now we have, as I said, we have figured out, like we have detected H2O2, H2S, and in the blood plasma. So the next step, already we have filed a patent on this, and next step is currently we have a collaboration with Apollo Hospitals Chennai. The doctor Paul Ramesh is having a collaboration with us, so we are working with him. And side by side, we are having clinical trials. So the phase 1 clinical trial is already done. So we are focusing on the phase 2 trial. In phase 1 trial, we have screened 20 ICU patients. And then in the next phase, we are targeting more number of patients, as well as we are targeting the animal models also to establish the theory and to get the more data to. And side by side, parallelly, we are also developing the prototype for market. Thank you.
Thank you very much, Indra, for excellent presentation. And I'm sure that your clinical trials will get successful, and our best wishes to you.
Yeah, thank you.
Nitin will not be there due to some emergency. He will not be able to speak today. So we have to conclude here with this for speakers. So you can unshare your.
Yeah, sure.
So with these speakers, in fact, one thing is very clear, that what Professor Gupta was sharing, that networking at this stage is most important. Many of these people are stuck. In fact, they do not probably at this stage need more funding for the business, but they need the funding for prototype or the product development. More than that, they need the mentoring support from you people. So maybe the startups, maybe the experts, maybe some other people in their network, if they could be able to help these guys, then definitely they can be the technology of tomorrow. As the age of technology is very small, in fact, if this technology is stuck for 2-3 years in these processes, then the chances of their survival in the market, will be, bit dim. So we have to move with the time. In fact, time has to be, we have to take care of that, So one thing is networking, and second thing, in fact, when we are doing the research, when we are developing the technology, it should also not be at the research stage. Initially, what Professor Gupta was mentioning, that we have to stick to essentials. So what is essential? Let me first finish that, but it should have the expandable feature. So tomorrow, some more, work I want to achieve with that technology. So it should be properly expandable. But at this time, in fact, 3-4 things we require. One is, completing the project. We should first understand at what TRL level we do have at this stage, and where we are moving. We do have a very clear plan. And then the networking with the funding agencies. At this time, the funds are available. If you see the, the problem of funding agencies, in fact, in all the task force, people are looking for good technology. Many times we are not able to explain properly, in fact, what we are looking for the fund. So we have to be very clear, in fact, and we have to move from, upward level toward the TRL, in fact, so that it should reach to the market. for clinical trial, in fact, what sort of clinical trial and what sort of other, trials we are looking for, we have to have the clarity. If not, then we have to do the networking. And at this time, GYTI is having a very good network. Sir, I request, in fact, if like the Fab Lab network, we could have the GYTI network. If somebody has stuck somewhere, so the people could, share, and some other people in the group can help that people. In fact, what will get
you are saying is very relevant. Yesterday, Renu Swarup mentioned this in her opening remark. The time has come for two things, she said. One, aggregation product platform for GYTI products and services, and second, alumni network. One of the suggestions was that maybe we should create a LinkedIn network, LinkedIn group of all these GYTI winners, so that they can have a real-time discussion, and maybe other people can also participate as and when need arises. So I agree with you, completely agree with you, that we need to create much more communication and coordination among the GYTI awardees themselves, because we have a huge range, and from 2012 onwards, if we put all of them together, engineering and non-engineering, because sometimes they're dealing with an engineering problem that may be solved by a non-life science person as well. What we just saw. So we should probably bring them all together. Let us see.
In fact, the fortnightly or monthly, there can be a meeting, online meeting, where the people could flag the issues. Issues can be technical issues can be financial issues. In fact, and if the people are getting interest, then they can have the offline meetings also. So more and more products should reach to the masses. One good thing, in fact, with this GYTI network is, you do have the Professor Gupta, in fact, who can push your technology more than you. You cannot push that much what Professor Gupta can push, in fact. And wherever you have stuck, please share it with us. Please share it with Professor Gupta, and might be we would be able to help you. In fact, we means all the experts here. All other members. Like, we need the more and more people like JS. So if you have reached up to certain level, please try to do the hand holding for others. Like JS is incubating some other companies, you should also incubate some other startups. So thank you very much. In fact, now I would request Dr. Dipanvita for her comments here. Yeah.
Thank you. When I was hearing our GYTI awardees and very promising startups, two things I wanted to see in their presentation and I didn't see, and it was mainly they were talking about their technology. I didn't see anything on their competition and the market. It will be very important because, when you are designing your product, it is very important to be able to design for manufacturing. Is DFM being considered? Are you actually looking at what are the features of your competitor products and how are you different? So those kind of things would be very important. So work on that. I'm sure you have some slides or some thoughts on that. But what I would love to hear from all of you, can each one of you list out 2 challenges that you have in what you are doing? And can we all try and hear and see whether those challenges can be sorted in a network mode? So there are 2 ways of doing solving a challenge. One is it can be a technology challenge, it can be a business challenge, it can be a regulatory challenge. And what Professor Gupta said, what Vipin said, everyone is alluding to the same thing. You have a strong network to back you up. Leverage that, use that. Don't work in silos. And if you can put up 2 challenges, and can we see whether these are solvable or not? So I just leave it at that.
Madhusudan, if you have any suggestion, any advice to give them with your experience from CCMB?
Yeah, I mean, I must say the 4 presentations, the energy level of their presentation is really, really impressive. I think that struck me most, and I think they know what they're doing very well. But as Deepanvita was mentioning about the challenges, and like most of them are into a kind of ex vivo or in vivo kind of products, the regulatory path is going to be that much more difficult. And I think in your network, you should be able to provide them very early advice. Otherwise, they may go into a direction and then, they may be left hanging in terms of where to go. And many of these devices that I saw are multi-purpose ones. So they are presently maybe working with one application, but they also should look sideways so that, like for example, the separation of cells from plasma can be applied in many other contexts. So similarly, they should not, of course, their focus is on one particular application, but it may be useful elsewhere too. So that is what I thought they should look for, because in business development, the only thing is how quickly you adapt to the customer requirements. It's less important what you're doing. It is more important what people want. So that you always have, it's a kind of a reverse engineering, if I can say so. I mean, you reverse engineer the customer's needs. So my advice is constantly have to look at the demand in the market, and of course what Deepanvita said is competitors. Ultimately you have to put it along with the other competitors on the shelf, and where do you stand? And these are the things. I'm sure entrepreneur journey is a very multi-hat activity, but that's a challenge. That's the fun part of it. I'm sure that's why I said the energy levels of all the 4 speakers is very, very satisfactory and impressive. I wish all of them very best. Thank you.
Yes, and if I can add, Madhu, since this is a session on business development, I would love to see whether they have spoken to enough customers. Do they know who their customer is? So, Professor Gupta, I remember there was session that we had done with a professor from Berkeley, and he said, get out of the room. Sitting in the room, you cannot solve a problem. You cannot bring a product to the market sitting in the lab, get out of your room, talk to people, talk to customers, find out who your customers are, who the saboteurs are, who will not want your technology. So I think that's a very important thing. You should know who wants your technology, who are the real customers, who seem to be the beneficiaries. Your beneficiaries may not be your customers, and who are the saboteurs, who will actually try very subtly not to have your product.
In fact, Deepanvita, so nicely you have mentioned this. In fact, very surprisingly, sometimes I'm Professor Chakravarti from IIT Bombay, and it is so critical when you're looking at business model incubation, because sometimes we just design and we design for our imaginary customer. There's nobody there who wants it, and then we have the wrong, the wrong pitches also. We have the wrong list of requirements too, and this can happen with professors also like me. So get out of the room, talk to users. Very nicely said. Thank you so much. Yeah.
Thank you, Chakku. You had given us I have attended your class on this example of the post letterbox.
Yeah,
and
thank you,
showed
sir.
how many times were you stunned by the rejection by the
users
before
right,
they
sir.
accepted your final design, and they took it all over. But before they accepted, there were cycles of rejection, repeated rejection.
Very true.
And one has to have a heart strong enough to bear with those rejections, because
exactly. We have to stand for a cause.
This is part of life. I mean,
very true. Entrepreneurship is like a cause, no? So we have to, make our things work. So, we have to leave no stone unturned to take things forward.
So I would like to thank Deepanvita. Despite such a hectic engagement at home, I don't know, how did you figure out to be so peaceful and so kind to the GYTI awardees, students? You are really privileged to have her today, because today anybody else in that situation of moving house should be there actually, because small things can be misplaced, and then she will spend all her time next year to locate them because they have been put in a wrong box. Now, this can be very irritating for a lady or for that matter, anybody else who's moving the house. So I understand that. I hope that the delegation that you have done is well deserved and well competent, because they will then make sure that if something is misplaced, they will make a list of that box number thing and tell you later on that, look, we decided to put it here. There was some space. We didn't want to leave it blank.
No, the problem was even worse. The problem was there's a very famous, reasonably famous vendor called GYTI in Hyderabad. And my office had contacted GYTI. They came and they were reluctant to give me the insurance papers. And then my office people just went to them and found out it's not a registered vendor of GYTI. They're not from GYTI. And they had come and done 1/3 of the packing. I cancelled it, and that was like, I have to tell you, and this is again for everyone here. So I suddenly remembered, and Madhu, you remember, Dr. Sheila Bhide, who was the principal secretary when IKP was set up. I remember ma'am was ma'am is on the board of GYTI. I just called her up. I said, ma'am, I want this help. There's some problem. Can you actually, who should I contact in GYTI to sort this out? Then she was absolutely prompt. She said, Deepanita, don't let anything move out of the room. Whatever has happened has happened. And by this time, the money has come back to my account. I had transferred credits. That was not a GYTI vendor, not a registered vendor. They said, if you opt, we'll make sure. But so they suggested somebody else. They have come and finished the survey. Whatever that vendor had taken money, it has come back to my account. So I think we have to also, that is a part of network. I just thought I'm not going to take it. I just called Sheila Bhide. I said, ma'am, you have to help me.
Yeah. So, the same thing is happening with all the Agarwal packers and movers vendors.
Oh my God.
You won't believe it, the number of people who are mushroomed as Agarwal packer movers and they also have vendors because they become big, right? So they have to distribute. And
so GYTI said actually, and the name was GYTI Packers and Movers. So we thought that is the only, but we found out with the insurance thing, there they bungled up.
Yeah.
So thank you very much, Deepanvita. Thank you, Vipin, for being with us, and thank you so much, Madhusudan. Your comments are very helpful. You offered to help in 1 or 2 cases. That is very much appreciated. Deepanvita also offered to help in 1 or 2 cases. Actually, this is the power of the mentors. And you know what? You will struggle for months and years. Our mentors can solve it in a minute for you. They can open any gate in the country. And I think that's the power of our network. I'm so grateful of Honeybee Network and Srishti and all our goodwill emerges from the goodwill of. So thank you so much, Deepanvita, despite all the hassle at home. You have been very kind. You have been very kind. Thank you, Vipin, for a short notice, but still you did a good job. Shared with us all your stories and the fact that the startup movement has reached the tier 2 and tier 3, as you were mentioning. But the challenges are also increasing now because the support system in those places is very weak. Mentors also not too many in tier 2, tier 3. So our responsibility, all of us together, our responsibility increases manifold as these startups will emerge from grassroots level, from tier 2, tier 3 level. They will not be able to articulate in English, or sometimes they will not be able to share a business plan. And yet they have fire in their belly, which we'll have to really address. So thank you so much. We will move to the next session. Thank you so much, Deepanvita.
Thank you so much.
Again, for being so nice, and we will get back to you. We have next session on roundtable discussion, open house discussion from 4 to 4:30. This is Dr. Anil Wali. Has he joined? Anil Wali, has he joined?
I think we are a little bit ahead of time, so.
We are ahead of the time. We are 7 minutes ahead. So I can understand. So
but I joined early because I want to know a little bit what's happening. I was always curious.
So Chaku, maybe you can use this time. You can use this time. You have always enough stories to tell. So you can use this time to tell us some lessons from design, because most of them are designing solutions, but they are never thinking how designers think. So maybe some tips on they are all life sciences. So they are doing microfluidic device, they're working, and you have dealt with some of the life sciences innovations for whom they do develop the solutions. You remember that those students who were developing air temperature sensor for the livestock.
We had them build the prototype.
Yes. So could you share some of your thoughts and then?
Sure, sir. In fact, I kept one these in the innovation valleys as well as a little bit of the introduction. Maybe I'll show that first, and as soon as I get the idea, I'll show them the design because that is now ready. I just pulled it out, so let me share that quickly, sir. Yeah, here we go. So like just to, in fact, as I was going through all the discussions, let me, is it started sharing? Not it. It is sharing now, no sir? Is it sharing?
Yes, sir, we can see your screen.
Yeah. So like, again, extremely, glad to be, with all of you, and, you're all on this journey of taking your ideas to the market. And it's the very most challenging part of it. In fact, I keep telling all my students that the idea is only 10% of the total journey. We always think that agar mere paas idea hai to sab kuch ho jayega. Nahi hota hai. You need every discipline, you need every strength, and every, in an enterprise, there is, from manufacturing to product development to vendor development. So all these things are very well oiled. So, I'll just show you some examples, and, and each of them has a role in your entrepreneurship journey. So that's very, very important. So let me, share an interesting, I took 9 years to do my PhD. And I'm very happy to inform you that Professor Anil Gupta was my jury. He was my, examiner for my PhD. And I took a tough path. I said, I'm going to do PhD by doing real case studies, that is with industry. Nobody attempts that because it can take long, and it did really take me very long to come up with. But my solution, which came up in the end, is very simple, and that's I call.
And I must tell you, I became his fan ever since I was examiner. I've been his admirer, fan. I love his work. I love him. I love his approach.
Thank you so much, sir. Very kind of you, sir. So, and in that same way, I've been always, looking forward to Professor Gupta for his advice and, to take things forward. So here we are, a very interesting challenge of when we take things from idea to market, collaboration is the key. And how do we collaborate? How do we talk to people? How do we get things done? Let us, quickly see this very simple model. And of course, a little bit of the journey of how the research went. In fact, I came from a company called Larsen & Toubro. I worked for 6 years after my Master of Design in IIT Bombay. And I learned what nobody can teach, outside. What I learned was when you get into companies which are well oiled, their efficiency model, they're very efficient in all their processes. So when they brought me in as a new design, they didn't have the design department at that time, and they had the engineering departments, very well oiled, from ranging from, product planning, ranging from product development, customer, the centric design, customer, feedback department, marketing department, everything was phenomenally well done. Whereas design wasn't at nascent stage. I was the only designer who joined the company. And when I joined the company, they were just looking for this one design. Just imagine students and colleagues who are from industry, when you come up with this design and you have this whole streamlined facility for you taking your idea to the market, you can really be you'll be very happy. And that's exactly what happened to me. I came up with this fascinating petrol pump design in 1988, and the company took it to production. And it went away into production and became a runaway success. The company did so well that, they were, the number one in the market. And of course, we designed a lot of other products for Whirlpool and, for other projects. But let me show you these small case studies, for giving you the idea how to reach from idea to prototype to, pilot production to mass production. The sequence is very, very clear. And what we do in all these cases. So in this case, for example, I like to continue the petrol pump study. So because I had synergy in petrol pumps, the competitor came to me promptly when I went and joined IIT Delhi. IIT Delhi, I became a professor there. So they said, now that you're a professor, you design another petrol pump for us. They didn't say you beat your Z-line petrol pump, but you design another petrol pump. Our formula for design, Professor Gupta was telling, I should tell you the formula. Formula is very, very clear. When you're doing innovation, you are coming up by using contemporary technology. All your technologies are contemporary, what you're using, right? So you don't take technology which will mature after 10 years. If you do that, your product will take that much time to come into the market. So that is one very important thing. We also looked at very, very closely creative ideation. I must tell you that creativity is very, very important when we do entrepreneurship incubation and taking our ideas to the prototype level. Because here, for example, how do we increase our creativity is a very big question. And, that can be done, of course, all of us are under pressure to build things. So we can always take analogies. There are a lot of interesting, lectures on that. I'll also share a lecture with you later on my way we use analogies. For example, students and colleagues, please see this interesting analogy. In 1994, people were using mobile phones which are the size of cordless phones. All of you have seen cordless phones, right? The big ones, Nokia ones. And at that time, our inspiration for our petrol pump was the cordless Nokia phone. Does it look logical? No. That is the whole beauty. Creativity flows when you take analogies from fields which are, ahead. You could choose any analogy which is working well, where people are aspiring for. So now look at this one analogy which changed the course of my new design, which completely wiped out my earlier product. The Z-line petrol pump was no longer to be seen. The bullet petrol pump became the runaway success. Look at the sketch on the left-hand side. It's exactly like that Nokia phone, with that antenna over there, with that large display, and look at the doodle. We call it doodles. If you can write in any language, you can sketch doodles. Don't think that sketching is only for designers. No. Every entrepreneur should have a sketchbook and they should doodle. And I, for the everlasting credit of my professor in Germany, I did my PhD, part of it, 1 year in, Karlsruhe, University of Karlsruhe, as part of an exchange student. And he would tell that sketching is the mother of all, development and all build of ideas. So here you look at the sketch, which is very big, bad doodle, but it makes you talk to your mind. So here I'm just wondering, oh, this is a Nokia phone. What would be the display? Oh, the display is oval and big. It's most prominent. So we need to have a very prominent display. What is the antenna? Oh, what will be the antenna equal to in the petrol pump? Wow, the antenna should be my place where my fuel should come from. There is no logical reasoning for that. In my phone, the antenna is using for communication. In my petrol pump, the fuel is, of course, pumped. But these illogical, these creative insights can lead to tremendous amount of engineering and product aesthetic, insights which can take you to big heights. So please be very crazy. Today we will have a small assignment, maybe while we have time to go. I don't know how much time we have, but we will quickly finish that. So now look at those advantages. So I took that. We quickly made multiple, CAD models. Look at the CAD model now. The display is large. The hose is coming from the top. The petrol is flowing from the top, and it's very short. And I have all these detailed case studies available online. So I'll share the Swayam platform with you. It's a full lecture and a full course on how these are done. And here we have now created a modular petrol pump with a lot of advantages over the large company, which is a 10,000, which is a 3,000 crore company, and this Midco is just not even 50 lakhs at that time. So now you can imagine how you could beat a large, and that's what is happening with all your biotech innovations. You're going to be the biggest in the world because you're contemporary, you're new, you're coming up with new technology and new thought processes, which can change the course of, multiple things. So here, for example, then it became a runaway success. Oil companies started only giving orders to this because it had all the ingredients of contemporary requirements of manufacturing, modular design. And look at that hose coming from the top. My God, phenomenal advantages. The hose came from the top. The person who's filling petrol doesn't get hand pain by evening. And that is a very small advantage. Now look at the other big advantage. This petrol pump was giving 65 liters per minute because the flow was very, very smooth from these pipes, because the pipes are round. Look at the big round on the top. And this is a bonus from the creative analogy, which is unrelated. So because of that curvature, now the fuel is coming at 65 liters a minute. And in a day, you can service some 14 to 15 cars more than the people who are owning the Z-line petrol pump. And it's business again. Look at what Deepanvita was saying. Who will value your product? And now all the oil companies, the stations want to use the bullet pump because you can service more cars and you can make more money. So this is the whole beauty in which we, take our creative analogies. And of course, now my major, focus was to show you the collaborative model for innovation. Colleagues and students, the major model is very simple. The core team, which is working on innovation and entrepreneurship and idea generation, has to be 150% involved in this activity. Whoever is your partner has to be 100% involved. If there is no 100% commitment, there is no innovation or entrepreneurship. 100 or 150%, I keep saying this. That is a very small team, 3 to 4 people. Then you have something called the support team or the enterprise-wide team, which gives you 10 to 30% support. You have to build this team and you have to build them partnership. You have to give them royalty. You say that you join our company and I will give you this 1% of my share. You please help us grow. And I'm telling you, even in IITs, for example, my professors have such great research. Because they're not ready to share anything with anybody, their research is all lying in their books and in the mock-ups or prototypes, and it's never reached the market. So please remember, make collaborations, make contracts with your support team where they're putting 20 to 30% of their time to take your project to the fore. You can never take your product to the market alone. And then comes the networked external team. They are those support teams who are available. For example, educational institutes whose professors are not asking for asking for being on your panel or board or ask for royalty, but they can be given, they can be hired, or you have consultants, you have NGOs, your user groups. So all these groups from the network external team. So these 3 teams, when you create these 3 teams, and of course they multiply, 3 core team, 3 into 3 support team, and much 3 to 3 into 3 and more than that as the network team. And you have to build this team day one. As not day one, your team grows while you grow. Depending upon your idea, the way it grows, you build this team forward, and there is a lot of success. I'll share all the details with you, and I hope I'm still having time, and we'll maybe Professor Gupta, I can come back again if Anil Walia has joined and you want to give a little bit of overview.
You have 4 more minutes because Anil is not
I will
there.
continue. Yeah,
so
sure.
He has contributed his time to you.
Oh, sure. So so here we have the collaborative model for, innovation here. Very simple model. In fact, I was a consultant to McKinsey. McKinsey was a consultant to oil companies, and they wanted somebody to work on petrol pumps. So McKinsey came to me, being the only guy in the country working on petrol pumps. And they said, Chakravarti, this model is very simple. I said, simple models are the best models to implement. And we need to implement very simple models. And of course, we have we have divided the stages of how the collaboration should happen, how your product should, how your concept should grow from a concept to a mock-up to a prototype. All that happens in stages. At every stage, you need to collaborate. And of course, what are the functions of each team, very clearly, marked as part of the thing. We will share that, through Prasanna Gupta to all of you, the whole model. And, of course, the composition, how we build the core team, how we build the, who will form your support team. And in case if it's an industry, these are all the support team. We did this workshop with Ashok Leland, and they came up with fabulous results by using this team. When I did this, did an innovation program for them for 6 months, and we got very good results. So from here we now move to and of course the final collaborative model. And again, thanks to Deepavali and Prasanna Gupta, keep always, and of course Vipin, you have to keep your user network loop from day one. When you show your idea day one to your customer, he may say, "Ye to nahi chalega, mujhe nahi chahiye." Doesn't matter. Take all the inputs from him. Or somebody else may say, "Arey, ye bahut acha hai, isme ye bhi kar do." Take inputs from him. Take inputs from the concept stage to the mock-up stage, and every stage with all your feedback. And this feedback will grow the perception among your customers, perception among our users, as well as it grows within yourself on what exactly the market needs. So the network loop has to grow across. The user network group is the most important group in this design. And of course, after that, we set up the Design Innovation Center in IDC. We have done a lot of projects for the industry. Some of these projects are all available on the Swayam platform. We will share the links with you. They're all open source. They're all available for everybody to go through. You can use it. My colleagues in other academic institutes can use it for their case studies, and students can benefit from the from getting credits also by writing an exam in the end through NPTEL. And like then we worked for the postbox which Prasad Gupta was working. And students, when I made the pilot production of 200 numbers, this postbox failed because we failed to go and survey in the rural areas and in the in semi-urban areas, and the maximum population was in those areas, not in the cities. So I moved out of my room, but I didn't move out of my city. And we failed miserably. And then after that, we redesigned the product. And now that product, the Prime Minister has launched it. So we're taking it forward again. But that is the whole lesson that we need to learn, that we need to be with the target people. But this is a social project. So you need the target as a whole population. Everybody should be able to recognize your post box. And this failed because of lack of the recognition. And I'm a professor in form and aesthetics. And how can such a professor make a mistake? मुझे बहुत डांट मिली ministry में, because ministry is not going to see whether you're a professor or not. You'd be asked you to make a product and it failed. And I take with all humility, you have to take those critics, and then we of course made things better now, and it's like there, and the whole case study is also available for you. And then we designed, Professor Ram Gopal Rao is the director of IIT Delhi, and we designed this very, very interesting nano based cardiac diagnostic device which can detect your, for example, 2 years before you get a heart attack. If it tells you that you're going to get a heart attack, Chakravarti, aap sambhal lo, to main sambhal jaunga, right? And this is the level of nanotechnology which now is happening. And that small hole is where you drop your blood, and that whole equipment was built. And when we conceived this product, within 6 months, the funding increased by 80 crores. Just because the funding agencies saw the scope. And now this is a company which is incubated in our incubation center, and like the product is really up there for further work. And these are the interesting things which design, when you conceive the prototype. Before you conceive the prototype, you can make a working rig. This is the working rig on the right hand side. But then when you make it like a prototype, and what did we use? We use mobile phone parts. Most of them are mobile phone parts. And of course the sensors and other things. And we built this prototype within a year. So that's the whole beauty of how you can take things forward. And of course, my petrol pump case today told you how it became a runaway success. My PhD topic, of doing action research projects, resulted in also a lot of riches for me. In fact, and riches for IIT Bombay, because IIT Bombay takes 30% of our royalty. And that's the whole beauty of how you can result in better systems. And of course, we did some more projects like the Bean Tracer, which are also
I
available.
should stop you here because this is something very interesting. The President of India remembered this when he went around the exhibition in the Rashtrapati Bhavan. And next day morning, when he was with his physician during the walk, he said, "Do we have that in our dispensary?" He said, "No, we have never seen that before." So he said, "Get it." So immediately I contacted Chakravarti and I said, "Look, President's office needs your rain detector, because many times it has to put here and there." And that's how it reached, this solution reached President's house, President of India's house, thanks to the fact that this innovation was so simple and yet so important. And it got the attention of the President of our country. So this is very important. Chakra, we could leave some time for these participants to ask.
Sure. Now we're through, sir. So thank you so much, sir. Yeah, we'll now go to the discussion. Yeah. We'll stop the sharing now. Yeah.
May I go first?
Yes.
Hi, sir. My name is Nitin. So I really like your model, but in that, this mostly suits an enterprise scale of a company, when we are a startup company and we have one product designer and one user researcher. So this sort of, a tired approach, I mean, what would you recommend for a startup?
No, the same thing. For the startup, we call it the support team. So the startup, if they don't collaborate with the current small scale industries around them very early and make them work for you as partners in your growth, how the software company came up, why is the software doing so well? Because they actually collaborate and they network and they share resources and they share profits. So being an entrepreneur, you should be able to talk to your vendor and say, "Aare, ye ban jayega, to main abhi aapko paise nahi dunga." When we do well, tomorrow I'll give you 1% of my royalty, for example. So those will be your support team, and then he will work in a different scale altogether for you. So you have to build the same team, but we call them the support team. And your support team will have...
And Chaku, another thing they can do is to sometime, you can have a class of 5 or 6 startup where they can pose their challenge to the class.
Why
and your
not?
students can do an exercise for, let's say, half an hour. And in the end of the class, they can make a presentation. I do that in my class many times at IMA. And Nitin, this is a very effective way. There are 2 advantages. My students get to know what are the struggles involved in startup life, real startup ventures. The startup gets to know what these young people who are going to be the future managers or designers of the country, what do they have to contribute? So it works mutually advantageous. We need in our class real life example, and the class students need interaction with the real life startup. So I think you should interact with the faculty, whether of IDC or other management schools or design schools, and involve them. You don't have one person, you have the whole class with you working on your problem. One class, one session in a one course, how much would it bother? I mean, we can easily arrange that. That's not a difficult. Anybody else? Any other? Yati?
Sir, I have a question, sir.
Yeah, go ahead.
Sir, it is beneficial to work parallel in multiple ideas for an entrepreneur, started.
Siddharth, again, very good question. You please put your video on if it's bandwidth.
Yes.
Yeah, so it's very good to see a person in front of you. Yeah, that very good question. Again, we always say that when a lot of people who come into incubators come with one idea and they actually have become a big company with some other idea, because they identified the real value and they identified the real customer who wants a different value. Let me give you a quick example. One of my students created this wonderful, portable operation theater, because in villages and all, you need to have, what do you call that? Disinfected, what do you call that? Isolated, in operation theaters, you have this, completely, sterile environment.
Sterile chamber, yeah.
Yes. So he created this and he worked for 1 year. It was a student project, MDes student project. Then I made him join BETIC program with Professor B. Ravi in IIT Bombay, which is synergistic with doctors. So a lot of doctors keep coming there, and then they all said, "Acha hai, kar do." So 2 साल लगाया उसने पूरी prototype बनाई, all the flexible systems, pumps, motors, everything, right? फिर बाद में he got selected in the ideas program, and in the ideas program they said you deep dive and find out who's going to buy your product. तो तब निकला बाहर वो 2 साल के बाद निकल के, and he married a doctor, and then both of them traveled to various locations trying to still, अरे कौन खरीदेगा मेरा ये sterile, portable operation theater. And then, for example, the breakthrough happened where they were in a burn ward, and the burn ward doctor said, यार ये तो portable नहीं चाहिए, हमको तो यहीं पर चाहिए, हमारे सारे burn victim मर जाते हैं because उनके पास sterile, हमारे पास sterile environment नहीं है. So his whole project idea changed from a sterile operation theater to a sterile burn ward individual hospital bed for burn victims. So he completely modified his whole project, made it more sleeker, more cost effective, and for the burn victims. And now, and he's been incubated now in Coimbatore, in Kerala, in one of the incubators in Cochin. And now, the product will come to light. So that is what happens. That you may think of something and start with something, but when you keep going forward, you may land up, and you should be ready to change, because, if you're not ready to change, sometimes you will lose the game.
Okay. Sir, one more specific question. Actually, I have just registered my startup. I am a student in IIT Kanpur here. So, sir, want to know that currently I am working on 2 medical devices, and parallelly I am thinking, since it will take at least 1 year to get commercialized, so parallelly I can do something for 1 year or 2 years so that I can also do some earning. So, as an entrepreneur, what you will suggest, that we should working on this or only focus on these 2-3 ideas?
But I will take Professor Anil Gupta's advice on this. No, Professor Anil Gupta, we should work on one, because see, एक ही बहुत है ना. Wherever there is a very, very strong customer, where value for the customer is there, value for your potential user is there, check that out right away.
I would say that make a no-frill version of your solution. I mean, you have a solution which has 15 features. Strip 10 of them. With only 5 as most essential feature, you work out version 1 And maybe the takers of version 1, which will cost less and which will be less sophisticated, easy to manage, are many more customers right away. So you earn your money by a no-frill version rather than by waiting for 1 year and getting all the features integrated, tested them, and then somebody says, no, I want one more. So you add 16th feature, and the journey never ends.
Updation.
If you keep on responding to every customer need, this is that I'm going to be making now a paradoxical statement.
In fact, I will add to
listen to everybody, but don't incorporate every suggestion.
Exactly.
You should know what to accept and what not to accept. Saying no to things is as important as saying yes. And many times we say yes to everything, and in the process we lose identity of where we began. We become a potato. Potato can mix with every vegetable. Doesn't have its identity, no? Potato नहीं बनना है.
और एक बात बोल रहे हैं, आपको एक वर्किंग रिग बनानी है. वो वर्किंग रिग, अभी आपका प्रोडक्ट अगर इतना हो, वर्किंग रिग आपका एक सूटकेस हो जाए चलेगा, पर वो तुम्हारा सॉल्यूशन काम करनी चाहिए. और वर्किंग रिग आराम से बन जाता है क्योंकि हम लोग 4-5 प्रोडक्ट को कैनिबलाइज करके काट वाट के लगा के हम लोग एक वर्किंग रिग बना लेते हैं अपना सिस्टम का. दैट फंक्शनल रिग विल बिकम योर very good source for doing your
test bed. It's a test bed. If you give you the simulation, yeah, you can do test beds. Use it as a test bed.
Okay.
And then get to the solutions. The features can be less, of course. Yeah.
Okay, sir. A small session. Will you please take a small lecture of us all if possible?
Yeah, why not? No, all of them are available online. I'll share with you. All of them are available online. You'll like it. I will immediately post my SWAYAM platform link. You have a Google account. It's free. It's available. And you please see all the case studies. We'll show you. And you'll get a lot of insights by seeing all of them. It's a very small 8-hour, 4 hours of video watching. And you'll be sharing whatever knowledge is available now. Thanks to Professor Gupta's drive. हमारा अभी अपलोड रेशियो और हमारा ज्यादा है डाउनलोड से. आजकल मैं डाउनलोड करना बंद कर दिया सर, अपलोड ही कर रहा हूं सर.
वाह. Siddhant and all the
जो भी knowledge है अपलोड करो.
Please listen to what we are discussing. He has mentioned. I have always talked about this in all my talks. What is your upload to download ratio? You don't upload much. Now, because he has uploaded all of that, you can access it without any problem, without any cost, without any hassle, without any password almost. You can just go to NPTEL platform and will see his course, Design, Technology and Innovation. Is that one you're referring to?
No, design or innovation by design भी है, दोनों भी.
Innovation by design, innovation by design.
और design technology, actually मैं NPTEL का नहीं देता हूँ, इनको सबको बैक करने ना हमारे पास और नंबर नहीं मिलती, फिर मिनिस्ट्री मुझे पकड़ लेता है नंबर के लिए. तो मैं स्वयं का लिंक देता हूँ आपको. तो स्वयं में आप जाना, स्वयं पोर्टल में, write my name, Professor Chakravarti, and design, technology, innovation. You'll get all those courses. Then you go to Swayam platform, and then type innovation by design. And then, my name, and you'll get all the innovation by design lectures and a lot of additional content and notes. And, and there's a self-page. A whiteboard is a self-page and there's credit. So you go to the self-page so you can finish all of them, in a Zippy. And there are people who are students who want to get credit. They go on a credit mode, which is timely based. So, we'll share all that email with Professor Gupta. So he'll share with all of you.
Chat में डाल दो
chat में डाल दूंगा सर सीधा. Yeah.
Anybody else, please?
Can I ask one more question, sir?
Go ahead.
Yeah. So I think startups should always talk to users, take feedback, and we have done close to 200 different surveys. But when a part of your audience is also outside India or some people that you don't meet in your daily life, how do you do that? So let's say our market is, let's say in Europe or in US. I mean, we can send them gift cards and take interviews, but how would you define that process to be done?
I would request my colleague from IIT Delhi to talk about that. Hello, sir. Are you there?
Yeah, for some reason, must have been last minute emergency.
Oh, so yeah. No issue. So please repeat your question in short, Nitin.
So talking to users is very important, but when you don't have that users with you physically, like in India, what do you do? So you're building something.
No, you mean your users are not in India at all?
Yeah. What if like that?
If they're not there, then you contact either some academics who have connect to those users, or you connect to some, depending upon what kind of user is it. If it is a hospital condition where you have certain patients, then you have to write to those hospitals and get them interested in what you're doing, and then request them to arrange a discussion with those patients. So it is possible. I mean, there are so many people who are helpful in this word. That's why this word is so beautiful. So we often hesitate in reaching out. I mean, let me tell you that you will be surprised by the generosity that society has. Nitin, write 10 mails tonight to 10 possible partners around the world, and I will not be surprised if you might hear at least from 6 sooner or later.
अनुगुप्ता सर 10 बोल रहे हैं, आईएसबी में तो 5 सिखाते हैं एंटरप्रेन्योरशिप कोर्स में। 5 लोगों को पूछो आपका काम हो जाएगा।
हाँ।
इंडियन स्कूल ऑफ बिजनेस में। So this is the whole thing.
लेकिन क्योंकि हम लोग मतलब मांगने में भी हिचकिचाते हैं ना, देने में हिचकिचाते हैं, मांगने में हिचकिचाते हैं। क्या करें फिर उनका? तो I mean, you should be generous in giving and generous in taking. मांगो लोगों से ना हेल्प। मना ही कर देंगे ना, ज्यादा से ज्यादा क्या होगा, जवाब नहीं देंगे या मना कर देंगे तो क्या हो गया, फिर और किसी को लिखेंगे। सोचना नहीं है बहुत ज्यादा। Don't think too long, just write. And this word I can tell you, Dipandita offered to help in so many cases today, isn't it?
Very true.
And she has probably one of the largest network of both companies and institutions. आप मांग के तो देखो उनसे या चक्कू से किसी से भी, they will help. कोशिश तो करो। Anybody else? Anybody else, please? Any other? I see so many of you here, so please do not hesitate and raise any question that you have. I mean, yesterday I will again remember that, Shweta, for that matter, that when Dr. Patrawale's student wanted some help, she immediately offered, मैं जानती हूँ किसी को निमंस में मैं आपको कनेक्ट कर देती हूँ। So many times the peers help, not only the mentors, peers can also be helpful to each other. हम सबका social network है, हर एक का। हम उसको converge करेंगे तो हर एक का network बढ़ जाएगा। That's how it happens. Anybody else? Shibu, Shivraj, Chita, Siddhant, Tejaswini, Vaibhav, Sumanta. Any one of you has any suggestion, comment, question? Before we close, we have 5 minutes more, and we can really dig deep into Chaku's huge repertoire of cases and examples. And he has worked with all disciplines in IIT Bombay: electronics, solar, life sciences, whatnot. They all go to him actually because he's so helpful. So
sometimes the professors don't come, so I go to the lab. बोलते आपका क्या नया है, फिर हम देख के उनको बना के देते, फिर वो समझ जाते हैं कि ऐसे होना है।
That's another way to do it. Very nice, very nice. Don't wait for people to come. You can also go to them. That's right. You're right. Absolutely right. Absolutely right. So anybody else has any question about designing solutions? And don't think design is only for artifact. Design is also for process.
Yeah.
Chaku, maybe you can say a few things about process design because people think it is only for products, but
or I'll just show the values of death support. They don't really know where their project is at what time, sir.
There are different projects. Different people.
Oh, I see. Sure.
For example, Jayesh has now set up several companies.
Oh, lovely.
So he's at a very advanced stage.
Oh, lovely. So, like
early stage, we invited him because he can inspire others.
Yeah, sure.
How he has grown. He has 100 ton per day capacity to make fertilizer and other things for potato and so on. So we have people with huge capacities created over the time, and there are some who are still at the prototype or proof of concept stage. So we have all kinds of startup, all kinds of GYTI awardees here, but they're all unique in some respect because they have been selected through a very rigorous screen. You know, out of few thousand people who apply, get only about 15 in a year. 15 on life science and about 10 to 15 in the other disciplines of engineering. Do you remember some of your students got GYTI award?
Very nice.
So, very good.
So in fact, again, like what Nitin was saying, some of the people use university contacts, go to student bodies in the universities, enroll them to do studies abroad. In fact, we were quite surprised that the MIT professors were in touch with my own student, my own TAs, and like for understanding the local market when they were designing, eye testing equipment. So so nothing can stop you from going to
Ramesh Daskar.
Ramesh Daskar, sir. Fabulous hackathons he was doing, and he was in touch with all, and they were, they in fact bunked classes and took the entrepreneurship that hackathon course, and we didn't complain because it's a good thing. But I'm just saying that Ramesh Daskar did that.
Yes, you're right. You're right. So there's no harm in offering some. Yeah, Deval, please go ahead. Deval, please go ahead.
Hello, sir. Can you hear me?
Yes, sure.
Yes, we can hear you.
Yes, sir. I have a question pertaining to, so particularly for medical devices, sometimes, I mean, we cannot give it to the user or the patient to use it directly, right? Because either there are ethical concerns or there's safety concern. So for those situations, how do you evaluate the user's feedback? I mean, is there any way, alternate we should approach?
For example, he's not your user then. For example, if you're using a prosthesis which is going into a body of a person, the people who are going to decision makers are the doctors, the caregivers, or the hospital administration who will buy this equipment, right?
Yes, sir.
So you have to take feedback from them. Sir, if time permits, should I show that BETIC projects where we did this, large prosthesis, जहाँ पर हमारा collaboration हुआ था?
We have 1 minute.
हाँ, 1 minute ना, तो वो वीडियो में आपको like
लिंक डाल दो
या ठीक है, मैं लिंक दे दूंगा आपको YouTube का लिंक, मैं आज उसको अपलोड करके, मेरे पास वो एक बहुत इंटरेस्टिंग, just let me explain to you in 1 minute. Sure. We were with the principal scientific advisor of the Prime Minister Chidambaram's office, and he gave this project to 3 institutes: IIT Bombay, Tata Memorial Hospital, and NFTDC, National Ferris Development Corporation in Kanchanbag, which is a non-governmental organization, and Balasubramaniam, famous guy who can make anything. For example, he can also make sponge aluminum, whatever you want in his lab. So these 3 institutes now built a mega prosthesis for children who have cancer in the knee. The kids who have cancer in the knee, the cancer grows from the knee to the femur and the bottom parts. So you cut off some parts of those bones also. So the prosthesis is very different from the knee operation. So we built this. It took 3 years to build it. Complete collaborative, effort between 3 institutes, and now it's at the clinical trials. It's at the 3rd level and 4th level of clinical trials. And I was involved in the armamentarium, making tools for the doctors to implement this in the body. So in fact, it is possible that you need to talk to people who are going to be the decision makers, talk to people who are experts, talk to doctors who are going to operate, and very, very established method of doing that.
Okay, like basically show them the prototype and then get the feedback.
Exactly. Showing mock-up models, showing CAD models, showing a prototype. You have to show that and take all types of feedback wherever possible, from those people, including nurses, doctors, hospital and potential costs involved, current competitors. All those are very critical. Yeah, we are done, sir.
Some of the doctors themselves may be patients. So, I mean, we don't have to necessarily go to patient in this case, but we can take help of doctors, even students, MBBS student. So they can also be. So thank you so much, Chaku, for being,
welcome, sir.
As always, so open to help. And at some stage, we will bring some of the awardees to your class.
Sure. Most welcome, sir.
So and that,
I will share everything on the chat, sir.
Yeah, please do that. Please do
share
that.
all the platform links so that they can join.
You shared what Siddhant was asking from IIT Kanpur, and there are other students from ISC and IIT Bombay and ICT. All over the country, we have students here. So they will all be benefited if you can kindly mention.
Sure, so I'll put that
So we will move to the last session today, a valedictory session. And I'm so happy that this conclave has given us so many ideas about what we should have been doing for all this while. I must say that it's a learning lesson. There are a lot of lessons for the GYTI team at Srishti and Honeybee Network. We realized that we should have such interactions more often. And also we would like to form a group now so that every month we will have a few hours to discuss any question by anybody, and not necessarily that we will find the answer, but we will find the questions. Once we find the questions, we will be able to connect you to the right people who might answer this question. So we have a huge reach around the country and around the world where we can find scholars and we can find entrepreneurs and we can find mentors who will be able to connect. We have one such mentor today with us. And Ketan Patel, main director of the company Troika, and the chairperson of the board of governors, board of directors of NIPER Ahmedabad, is a very dear friend. Ketan, you have joined, I suppose. I'm just trying to confirm. Yes. So he is an amazing fellow. He's a gold medalist, first of all.
Yes, I'm here.
Yeah, thank you, Ketan. So I was trying to introduce, but I like to begin with an incident. Rather than reading your biodata, I must tell the day we met, and he reminded me the other day that we met in a flight.
Yes.
We were both in a meeting. I didn't remember that part. And then I said, why don't you come and let us talk about it? And we met in my office in Wing 13 at that time in Ahmedabad. And the idea was that he had come out with a breakthrough, which is the diclofenac injection actually is very painful injection. So it's a paradox that an injection given to remove the pain, it causes pain itself. And through his R&D, through his research, he discovered a solution which the large number of companies dealing with this problem around the world had missed. And he found that if you change the particle size, then it disperses very rapidly in the muscle and the pain is not there. So it was a very interesting physics and of course the pharmacokinetics and all of that along with it, which made this option. He had not filed a patent at that time. So we said, you must file a patent. That was the first patent he filed. I remember that. And we had a long discussion. And after that, how many patents have you filed?
Another 15.
15, that's wonderful. So at least, our R&D sector, our pharma sector is blamed for being just me too.
Yes.
And I agree that the large number of products that his company may have, maybe generics, maybe general, they may not be unique, but in addition to whatever we are doing for our cash flows, there's always scope of identifying that unmet need.
Unmet need, correct.
And what he had done was he identified an unmet need of all the people who needed this pain relieving injection, that they were having pain of the injection itself. And the solution was found. So I'm very proud that my dear friend and a very distinguished entrepreneur, he has done many other things. I remember that one of our products which we licensed to Ketan, Herbavate, was the first time. And what is interesting is that was a cream for skin disease, and he invited all the community representatives. There were six community representatives sitting in his office when the launch was done. Very often companies forget about the people. But I remember that photograph, and I still show it in my presentation, that all the 6 community representatives, elders, were invited to the launch function so that they could all take pride how their knowledge, when pulled together by Srishti Lab, was going to the market. And I remember that went to Brazil and Malaysia and wherever. So Ketan has had both kinds of achievements, one in his own discipline, in his own company, but also in linking up with the grassroots knowledge systems, innovations, and see wherever they can go further. And of course, we need to work more together. But nevertheless, there was a good model of benefit sharing. So whatever royalty came, we sent it to all the communities by money orders every year, 100% went to the people. So in that sense, it was a very good benefit sharing model, which was working well and has been working well for such a long time. So this is another aspect of his life. And I would say before I hand over all of you to him, to inspire and instigate, both. Instigate because having been in NIPER, he has talked to the students, he has realized that many times we don't look at the obvious. So other day, I was talking to him and we were both together. He was the chair of the session and I was there to talk to the incoming batch of NIPER. And I asked them, all of us know the way some ointment, the nutmeg was rubbed and given to the child, the infant, through the navel root. Is there any drug which is delivered through the navel root? And there was none. So the question then arose that why don't we work on new, for the science it is new, but for us it is traditional drug delivery route. So many times it is so obvious, we have all seen it and ignored it. हम सब ने अपने घर में देखा है नाभि के ऊपर हींग मलते हुए, हींग का पानी चम्मच बोलकर या जायफल को घिस कर लगाते हुए, सबने देखा है. But when it came to developing scientific basis and adding value to that, I don't know which are the ointment which will be delivered through that route faster, but there must be some. And that's a live question that we are opening. So I'm very grateful to you, Dr. Patel, Ketan Patel, for being here with us. And these are all GYTI awardees. There are equal number of people on YouTube who are listening to you and all around the country, not just the GYTI awardee, but everybody else is also listening to you from around the country. And these are students who got selected for GYTI award. And we only select 15 people in a year out of thousands who applied. And they are from IITs, ISA, Institute of Capital Technology, JNCSR, and top institutes of the country. Because competition is so tough that the second-tier institutions are not able to make. But mind you, NIPER, there was a student from NIPER Ahmedabad who also got GYTI awards. NIPER Mohali got one GYTI award some years ago. So other NIPERs have not got, but NIPER Ahmedabad and NIPER Mohali have got GYTI awards. This is also a matter of great distinction, that the institution's age and resources don't matter. What matters is the fire in the belly. So thank you so much for being with us. Floor is yours.
Yes, thanks, Anil, for the introduction. And in fact, I've gone through the books of the year-wise awards which have been conferred. And actually, it's heartening to note that there's so much innovation happening in this generation, at least. Our generation, possibly majority of us were not really inclined to innovation. The times were different, the infrastructure was not there, and the funding was also not there. Today, everything is available. And the 6 books which you have sent me, I couldn't go through all of them, but I went through some select innovations which interested me. And I found that there are several. One which offhand comes to my mind is a diabetic diagnostic kit made by some students. So the test kits which we are using right now from US and Switzerland are very expensive because they are all disposable and the chips are very expensive. So, and I'm sure there are many more such innovations, and I hope that the innovators also become entrepreneurs and give birth to products, because that is what the new India is all about. In this last session, what I would like to share with you is what I have learned during the process of innovation, that what are the facets and what are the triggers to innovation. So I have compiled a kind of a poster session for you all to understand my perception of how one can innovate. I'll just share the screen. Give me a second. Just desktop down.
There's a share screen button, green button at the bottom. Share screen.
Yeah. I'm just accessing it. One second.
Sometimes if you have not opened the presentation, then it will not be visible. First,
it has been opened. It's just that...
use Alt tab then. Use Alt tab.
Downloads.
Close other windows.
Record. Record nahi kari. That's okay. I'll just sign out and sign in.
No. Your window is open, then you close other windows.
No. You'll see there's a permission being asked, so I need to log out. Zoom will not be able to record the contents of your screen until...
No, we are screening, we are recording. We will share the recording.
Okay, yeah, exactly. Okay, so now downloads. Okay. Share. Now open.
No, you have shared the whole screen. Don't share the whole screen. Go to the window option. Go out again. Share. When we go to the share, there is a board that will come. So if you go to the window again and you will select the presentation.
Whiteboard. Is it the whiteboard?
No, not whiteboard. You have your presentation is visible in the among the windows.
No, I have not opened it. I'll open it.
So first you open the presentation.
Okay.
Now you share the button. It will be visible.
पहले वाला कैंसिल करके फिर से करना।
अब ये ओपन तो हो गया है।
दिख रहा है वो?
हाँ. Now what?
You press the presentation. Wherever the presentation is visible, that you just press that button. That one. आ गया presentation का बटन वो विंडो आ रही है?
हाँ आ गई।
उसको प्रेस कर दीजिए। And उसको बस हाँ. Now it is coming. You can make a full screen.
Okay.
That's right. Perfect.
So as an innovator, I am struggling with this innovation.
All right, doesn't matter.
Office में से होता है तो the people do it, so you tend to not learn.
Yeah, no, I realize that, but it's okay. It's working all right now.
Okay, so this is a kind of a handbook which I have made, and it's predominantly visuals. And so the one thing which I think comes in the way of innovation is this obsession with reverse engineering. Because reverse engineering somehow in India, because of the large population and various business strategies available, including low pricing and high penetration customer relation, reverse engineering tends to give enough profits, and that becomes a disincentive for companies to risk out into innovation. जब धंधा चलता ही है reverse engineering से, तो क्यों करें innovation? Because at the end of the day, innovation definitely entails a risk, and quite a substantial risk. And as there's a conflict between the entrepreneur and the innovator. Entrepreneur wants to have profits, and the innovator wants to do new things which will erode on the profits for the time being. So that's what we have decided at Troika, that we don't reverse engineer. We raise questions actually on various things which we observe. We observe that there is a certain gap, and we ask a question, why this gap? And therefore, then we are able to imagine, innovate, and excel at creating novel drug delivery. So that's the fundamental philosophy which has driven us. ये वापस hang हो गया. किसी
ये करो.
चलो ये इससे चल रहा है ना. Yeah. With the arrow it's working. So I won't read the whole thing. Let's go to the visuals. So the bottom line is that it is only those who innovate, they will lead, the rest will simply follow. And this doesn't, I mean, the size of the company, etc., doesn't matter at all. All of you know the story of Apple, actually, which is the most classic, where Steve Jobs had been actually removed from Apple because he took the company into a complete a loss hole. But then he had to be called back because he had some ideas which the board got convinced, and they allowed him. And then the rest is, of course, history. And today they lead, and how? The reserves that they have created are massive. So likewise, what Anil mentioned about our Dynapar AQ injection itself has put us in the leadership position. See, I'll just show you the visual of the product. This is the product which we made. And the one is ampoule, the other is a pre-filled syringe. Pre-filled syringes are used to administer an injection very quickly, as well as without any chances of contamination. So we've given that premium range also to the customers. But before this was launched, ironically, every diclofenac injection, including the one from the original inventor Novartis, they used to cause pain at site of injection. So it was ironical. A product which is supposed to alleviate pain was causing pain at site of injection. And that was the question which we asked ourselves, that ye kyun ho raha hai itna pain? Because the drug itself is known for very powerful pain analgesic anti-inflammatory action. And that question led to this product. And the product became so powerful that when we entered the market, we were not leading. The leadership was, the red queen was Novartis. They had almost 80% market share. And our brand at that point when we entered, of course, with a 0%, and there were around another 45 companies in the rest of the 20% market. But the superiority of the product ultimately brought us into leadership. And it eclipsed the sales of Novartis. The product brand was Wolveran injection. And it was a 3 ml injection. It was causing pain. This one didn't cause pain. Therefore, there were other multiple advantages of pharmacokinetics, etc. The brand kept on growing. It eclipsed the sales of Wolveran. And a day came when Novartis copied our product. And we sued them, and we blocked their sales. And today we are in a situation where we have at least 65% market share. Balance 35% is all to companies who have copied our product, because the patent litigation has been lingering since last 7-8 years. So although we hold a patent, there is gross and rampant patent, evasion of our intellectual property. So we lose about 50 to 80 crores of revenue or more per year since last 7-8 years. But that doesn't bother us. What is important for us is that we got the leadership position. We got recognition from the medical profession, because initially as an Indian company, we were questioned, how come you have innovated? Because Indian companies are not supposed to innovate. So they said, agar karna hi tha to Novartis ne kara hota, aapne kaise kar diya? So that ultimately we came up with clinical trials, etc., and that drove us into leadership positions. So if you innovate, then gradually you enter into that leadership position. Sometimes you can get into leadership through turnovers, but then also there's another very special position where you lead in spite of having a lesser turnover, but a turnover of creatively and innovatively made products.
वो product में भी change था या खाली वो syringe was the innovation, or the product also had a change?
No, the drug is diclofenac, as you can see in the generic name on
Generic
top.
name, yeah.
So the drug is exactly the same. The change was in the formula. There were factors. I'll come to that when I'm going to explain another aspect of how these things come to your mind, actually, because it's all about visualization. Innovation is very close to linked to, at least I feel it's very closely linked to how much you can imagine. That is why the punchline of our company is imagine, innovate. Only if you can imagine and create visuals in your mind of what you want to do, and what are the options you have, and which visual leads you to believe that if I chase this track. So I'll come to that. There were changes made in the formulation, and one of the major change was the reduction of volume. Because if you visualize anything, if you put say 1 liter of a solution into a rubber balloon, तो उसका extension एक certain extent का होगा. If you put a 3 liter volume in the same balloon, that balloon will be extended much more.
Exactly.
So the same thing is happening in the muscles. When you push 1 ml, the muscles undergo a lesser distension. When you put 3 ml, they undergo 3 times distension. Distension is always going to cause pain and even some muscle tear. So that was one. Then there were solvents, and the particle size which he mentioned, they were also causing irritation. So there are on the skin, you know that if you put glycerin, it is soothing. If you put a caustic, then it's going to burn. So surprisingly, Wolveran had put a solvent which was causing a lot of irritation. And I'm very happy to share that one of our papers, not our paper, it's a paper published by a doctor from Apollo, Ahmedabad. They have injected 1 ml Dynapar AQ, and they have injected 3 ml of the conventional diclofenac injection, and then taken MRI pictures on each of the patients to see the extent of muscle damage being caused. You know, the muscle damage caused by Dynapar AQ is negligible, whereas that caused by Wolveran is very, very highly significant. There's a large volume of muscle. It continues from 0 hour to almost 2 days. The muscle volume which is getting damaged keeps on increasing. So this is going to be published, I think, in the next month or so. So, but this, I mean, MRI is again visualization. You can see, but at that point, my ideas were generating in my mind that what are the things that are causing this muscle damage? If I can visualize, then I thought that volume was possibly one of them. And that proved to be correct, actually. Today, the entire 3 ml has disappeared. Now all the brands on the market are 1 ml. So actually we have created a new category. It's not the matter that they are all infringing our patent, but when the suits ultimately come to fruition, I'm sure we're going to claim heavy damages from these companies. And they include some of the big companies. See, that's another thing in our country, that companies which have a marketing muscle, they're not reluctant to copy your product. There's nothing about a self-esteem or a kind of a philosophy of the company. We will not copy a patented innovation. These are the restrictions which we don't keep with ourselves. Then those companies don't tend to innovate, because ye rasta open hai to kyun innovate karenge. This is another thing which I chose at that point of time as a very small, not very small, I would say micro. When I used to meet Anil, he knows that it was a micro enterprise actually, with me as a CEO, come general manager, come finance guy, and some 2 or 4 chemists and some 15 employees. So, but if you want to come up with an original idea, avoid the path which has been chucked by all of them. So you have to think different and you have to do something different, because otherwise it doesn't give you recognition. I being a B Farm, I have so many people, so many of my colleagues who were, who have been all pharmacists and who had set up companies. And at that point of time, they used to ask me, because I used to introduce different kinds of products, not innovations, but even then different. Like antidotes, if you talk, those days India had no antidotes available. Poisoning तो India में जितनी होती है शायद कोई country में नहीं होती है, because pesticide poisoning is rampant in India even today. In spite of so much modernization, it still continues, because principally earlier it used to happen because of agriculture and unprotected feet, but today that may have minimized, but attempted suicide is another way. People consume Baygon and TIC20, etc. No antidote was available. It used to come from Japan. And we made that antidote, and that made this company actually, because itna shortage mein rehta tha, and it was so expensive. Those days when the injections, average dexamethasone injection used to cost 3.5 rupees, wo time pe this antidote used to cost 120 rupees, one dose. And the total dosing schedule was 6 doses. So we introduced it at some 35 rupees. And that led to our presence. Within 6 months, our presence was in every hospital across the country, because all the medical colleges, they essentially need to keep this, because these cases most often end up in medical college hospitals. They don't go to private sector. Those days, in any case, there was no private sector. So private clinic mein to ye case manage bhi nahi hote, people would die. And people used to die. In fact, that is how I came to know. A friend of mine one day told me that you guys, ये सारे Indian companies cough syrup वगैरह बनाती है. आज एक patient मर गया, तो मैं बोला patient तो मरते ही होंगे. तो बोलता नहीं, मरा इसलिए दवाई नहीं मिल रही थी. बोला ऐसी कौन सी दवाई है? So he named this thing called pralidoxime, aldoxime, methodide. Now I was a gold medalist in my education. I'd never heard the generic name of that product. Never. Because वो भी कॉलेज में भी नहीं सिखाते थे, तो पता ही नहीं था कैसा कोई एंटीडोट है. So that first of all hit my self-esteem. यार नाम तक मालूम नहीं है, चलो दवाई नहीं मिलती अलग बात है. So then I went to the college library, and those days I had access to the library because I just completed my studies, and in the first 2-3 years, you know how it is. You go back to your alma mater, and the librarian allows you to. So I checked up the molecule. I looked at its structure. Then I tried to source it. It was not available because it's a very, very, very niche product. Only 2 companies in the world were making it. One was a company called IS in the US, and second was a Japanese company called Sumitomo. And also there was a company in Europe making it, because these drugs, the drug pralidoxime in some modified form is also an antidote to nerve gases. So they were making it for that purpose. And we needed it for this pesticide thing. So ultimately, then I went to the Atira library. Again, I had access, and I found out the synthesis route, and I synthesized it in the lab personally. The reaction used to had to be done at night because it was a photosensitive product. There was no sodium vapor lamps those days for us. At least my company, as I said, was micro. If I had a sodium vapor, I could do it in the daytime, but I didn't have sodium vapor. So I used to run the reaction at night with the help of a battery. And while the reaction would be on, I would keep the chamber dark. From time to time, would just give a shot of light and check it out whether it's working out or not. And finally, I think 6-8 months it took me to make the bulk active pralidoxime. And then another 6 months I made the injection. But then that was that taught me actually कि अलग करने से overnight you make a quantum jump, and more than that you earn the recognition. The medical fraternity, we kind of entered the minds of all the doctors that here is a company which has served a need for the nation.
तुम्हें वो अवार्ड जो मिला था उसके बारे में नहीं बताया? NSTDP से जो मिला था अवार्ड.
हाँ, awards का बाद में बताएंगे. Let me talk about innovation. No, awards तो काफी मिले हैं, but I think
that was the first award at national level.
Yeah, that was for Dynapar AQ. That was for this product. That was given at the hands of Dr. Abdul Kalam.
Correct.
You know my history very well.
I've seen you growing. I've seen you growing.
Yes, yes, you have. You have been actually for the first patent, as he said very rightly, I can't forget. He had forgotten, but how can I forget that meeting? Because he was on a flight and we had not met actually, but we were. I was also going to give an address at a MBA college, and he was also there. And then chronologically, I gave the address, and then he. I think first I don't know the chronology, but he sat and heard me possibly because when in the flight, he told me that your address was very good, Ketan. तो आई वाज शॉक्ड यार ये आईआईएम के प्रोफेसर को याद भी रह जाता है इतना एंड देन वी टॉक ऑल द वे फ्रॉम बैंगलोर टू अहमदाबाद एंड देन व्हेन आई हैड टू राइट द पेटेंट आई केम टू यू एंड आई सेड कि क्या करूं मैं तो यू गेव मी द नेम ऑफ डॉक्टर गांगुली इफ यू रिमेंबर
yes
दैट एसोसिएशन स्टिल कंटिन्यूज So here is what we did in the recent past as a I mean, when you want to think different, what is the different thinking? B12, vitamin B12. This is another huge product which is in the making. Selling innovations, another thing I should tell you, all innovators, is not easy. It is very difficult actually, because you're going to change the habit. You're going to change the beliefs of the users. So initially they will resist because India is not known for innovation. Apple कुछ भी लेके आएगा आप खरीद लोगे, Samsung कुछ भी लेके आएगा आप खरीद लोगे, इंडियन कंपनी को कुछ लेके आएगी छोटी सी बेचारी तो उसका कोई नहीं मानेगा. So the same situation happens, and in medical world it is even more so because it's directly being used on patients. So vitamin B12 deficiency is rampant in India, rampant. Highest percentage and largest number of people in the world are in India who suffer from this B12 deficiency. One is thanks to our diet, vegetarianism, and even the non-veg that we eat is cooked so much and overcooked so much that vitamin B12 being thermolabile, it gets destroyed. So dietary sources of it of B12 are very poor, and therefore we have this B12 deficiency. The funny thing about B12 is that supplementation by the oral route is not predictable. Some people can absorb the B12 from their gut, others can't. Others can absorb it here and there, and that is why even if you take a multivitamin tablet containing B12, तो आपका B12 शायद नहीं बढ़ेगा. And if you get your B12 tested, if from today's population which is on the net, I can bet on it that at least 35 to 60% का deficiency confirm हो जाएगा. Because when I got my own family tested, everybody was B12 deficient, although we eat quite healthy food at home. So the only short way is the injection. And the right side shows the ampule of methylcobalamin B complex injection. And that is the only way. And the B complex injections are extremely painful. So we thought that what could we do which could be very different, which could be an original idea. So we experimented with a sublingual, a B12 which could go through if it's put below the tongue. Ran so many versions of formulations. We make usually a study design protocol. So simultaneously, several versions are tested, and all of them failed. None of the formulations could ensure adequate absorption of B12 from below the tongue. So next we went to the nasal membrane. We said, let's try out the nasal. Again, a protocol of formulations was made and lots of them were tried out, and one hit the bullseye. We found out that if the pH of the solution was kept in a certain band, there was an immense absorption of B12 from the nose. And when we ran the pharmacokinetic studies, we found that the Cmax achieved was equal to the injection.
Wow.
Published value of the injection जो था, उतना Cmax achieve इससे हो रहा था.
Wow.
So, and you know, in India, the biggest embarrassment for our country is
I've been using it. I've been using it since you gave me.
Yes, I think I had come personally to show it to you. आपको तो सारे पहुंचा ही देता हूं मैं. Whatever is usable, I have to send them to you. So, India, the biggest problem is malformed children. See, in the pregnancy stage, if the mother is deficient in B12, then she will, even if she takes iron, the hemoglobin doesn't go up. B12 is a compulsory partner And because the partner is not reliable, ab tablet kitni khilao, some women may benefit, others may not. So there is a defect called the neural tube defect. The spinal cord, when it is being formed, is called the neural tube. And the neural tube defect results in malformed children with the spines coming out of the back and all, very ugly and very, and there's a very bad statistics that we have, and this is
We have lost your voice. We lost your voice. Ketan, we are not able to hear you. Unmute yourself, Ketan. Unmute yourself. तुम्हारा mute हो गया है, Ketan, mute हो गया. और तुम्हारा presentation भी चला गया. Ketan, we lost you. You have to reconnect. You are connected, but your presentation is to be re-shown. दोबारा से share कर दो presentation को और अपने को unmute कर लो. Your voice is muted.
Yes. And now can you see the presentation?
Now we can. No, presentation is not there. Please share it again.
Oh, achha. One sec. Now?
Yeah, it has come.
Okay, fine. I think connection टूट गया बीच में.
कोई बात, अब आ गया. So you were talking about the malfunction in the children, malformed children.
Yeah, malformed children. The spinal cord in the fetus stage is called the neural tube. And B12 deficiency causes neural tube defects.
Oh.
And the largest number of neural tube defect children are born in India. So right now we have entered into a public-private partnership. We are sponsoring a huge study initially on adolescent girls and women, because they are the ones who will become pregnant ultimately. So that study is being done by National Institute of Nutrition and ICMR. And we have been involved, but since the institute would not like to take money directly from us, the funding, we will be generating funding through BIRAC. So I saw that your SISTI is associated with BIRAC. So this is good news. This study agar aayega, to I can assure you this malformed children will be, the chapter will end.
Wonderful.
Yeah, then those ladies workers who are there in all the villages and all, they can,
Asha workers.
Bus, exactly. They can put a puff of this Neso B12. For the first week, you have to give alternate day. That means 3 doses. And then 1 dose once a week for the next 4 weeks. The ladies' blood B12 levels will go above 700. So this is a major product which has been launched. Hello?
Yeah.
Unfortunately, COVID, it has affected, but it's picking up now. A lot of doctors have started prescribing it because it's pain-free. You don't feel anything in the nose. You just take a puff, 1 in each nostril, and that's the end of it. And we are now getting it registered in almost 75 countries, because the rest of the world, other than the developed world, also has the same problem, but lesser than India. But you'll be surprised, even Southeast Asian nations which eat so much of marine food, even they suffer from B12 deficiency.
Surprising, very surprising.
Yeah. Another thing now is, of course, the visual is, we have purchased it from somewhere. It's not created by us. But the sentence is important. Essential trait required to travel the distance between an idea and an innovation is self-belief. So self-belief, I think I have always believed all along that it's very important for an innovator to have, because you're going to think out of the box, you're going to think against the current. Here I'll cite the example of this product of ours, which is a patented spray for pain. Anil, I think I've sent this to you as well.
Yes, you did.
This has become a huge product again. And in fact, it's selling across the world now.
Oh.
Yeah, because see, the problem was that I read in the medical textbooks that whenever you apply anything topically for pain, all these brands like Move and Volani, 1% of the drug goes through the skin, because the skin has a massive barrier. And that is natural. If the barrier was not there, obviously, anything which falls on the skin would go through into the system and we would die. So there is a barrier called the stratum corneum, which prevents everything from going in. So then the visualization part again, could we create very minute nano pores through some kind of an additive and make sure that for the time being the drug goes in? Then of course the natural process will close the pores again. And that work, this has got about 5 to 8x penetration as compared to the normal brands which are on the market, the tubes and the gels. They essentially work by creating heat. The drug really doesn't go through, but aapko burning hota hai, so you feel something has been applied and it is working. But within 1 hour or 1.5 hours, the effect wears off and you start getting the pain. Gradually, it starts increasing. This one gives you a pain relief for at least 6 hours. And it is so targeted as such a small amount of drug goes in as compared to the oral diclofenac tablet. Agar aap lete ho aur ye lete ho, the amount of drug that goes through is just 1/50th of the oral dose. So 50 mg ka dose hota hai, the 1 mg jata hai, but 1 mg goes to the site of pain. And therefore, it gives tremendous pain relief. Therefore, the product has become extremely successful. All the top gynecologists, it is the #1 prescribed topical painkiller in the country. I am very happy to let you all know also that we have now taken Neeraj Chopra as an icon, because I want to support all the gold medalists. Neeraj, as well as all the Paralympic gold medalists, we are going to sponsor them and we're going to use them to promote this brand also.
Wonderful.
This is again a visual created through my imagination. As you can see, the oblique lines are the effort and these are the falls. So with each fall, I believe that we need to increase the intensity of our effort. And that would definitely lead to products like Dynapar AQ or QPS or Nasal B12. Right now, to talk of chemistry, we now I'm venturing into the domain of medicinal chemistry. So we are trying to make a very important drug. I cannot disclose the name, but there is a fluorination reaction which is happening, but not happening to the extent it should react theoretically. So now we have possibly taken till date in the last 3 months more than 60 trials, and we are still continuing. So each time we are failing and each time we are continuing. Maybe we'll succeed or maybe we'll have to accept the lesser normal, because we are not satisfied with what we are getting. We are getting a product, but I believe the yields could be much more. So some kind of an innovation at the molecular level, maybe some catalysts, we have tried several catalysts, they're not working. But that is how I think innovation can happen. Each time you fall, you stand up and attack with enhanced or double vigor. And this is about the questions someone asked: vivid imagination powers innovation. I believe that imagination gives you the ability to create visuals, and the visuals actually lead to seeing the problem at whatever level you see it in your mind. Why Nasal B12 should work through the nose, or why the drug should go through the skin. As I said, creating micropoles, all that is a visual game. And first, if you visualize, then you can build up a strategy to support your visuals or in line with your visuals. And that could possibly then lead to an innovation. There's no certainty, but the beginning is surely imagination. Therefore, our caption, our punchline is imagine, innovate. I give a lot of importance to visualization. And I tell my team also the same thing, that keep thinking. अगर आप कुछ कर रहे हो तो सोचो कि अंदर क्या हो रहा है. Just try to imagine even at a molecular level. This is another visual very close to my heart. I have created it. So as you all know, that the passion resides in the heart and the ideating, creativity resides in the brain. So the need is for the heart to constantly be after the brain to find answers to questions which you have identified and raised, maybe a big question, small questions. And this should be relentless. So fuel your innovative endeavors with relentless passion. And then you're bound to get somewhere, you're going to get breakthroughs, somewhere you may not, but at every step of the way, you'll be creating dots of knowledge, which ultimately will enrich you. So creativity of an individual or an organization will shine through its innovations. And that is what, as I said in the beginning, no matter the size of the company, today, if you ask any good doctor about what is Troika, we take a perception mapping also through agencies, and we come to know that they hold us in a very huge, possibly in a much bigger size than what even I know of the companies. They put us at par with multinationals actually, because of the kind of products we have given, the kind of packaging we give, the kind of quality we give, and the quality of, and the kind of support that we give. But the helm is innovation. We register a mark in their minds because we are meeting something which was unmet. It was an unmet need and we have satisfied it. So these are the innovations. I've shown you some photographs. Now, this one is a very simple innovation. And actually, once you create a kind of an identity for yourself through innovations, even a small but meaningful innovation becomes a big product. Now, Citra Zinc SR is a sustained release vitamin C. If you know about vitamin C, that moment you take it, it goes into the stomach, gets absorbed, goes in the bloodstream, gets filtered in the kidneys, and comes out in the urine. 2-3 घंटे में विटामिन सी सिस्टम से बाहर निकल जाता है, जो काम करना एंटीऑक्सीडेंट का करके निकलता है. So if you could give it in bits and parts, sustained release, then it will last longer. Another thing I read about was that if you flood, many people take 1 gram vitamin C. एक तो acidity हो जाता है because it's ascorbic acid, and secondly, it is proven that flooding vitamin C, the body cannot absorb that much amount. तो उतना ज़्यादा यूरिन में waste होता है. So if you give it in slow release, then 90% of the consumed amount is absorbed, no acidity, and most importantly, longer duration of antioxidant action. And then there is zinc, which is known to work in COVID. Selenium also a very strong antioxidant. So ये COVID के time में हमने launch किया था. It has become a very successful product, at least in India, but in rest of the world, especially the African bloc and Southeast Asia, this has done $1 million business. So sometimes meaningful innovations may not be rocket science, but you can still get good business out of it and good recognition out of it because there's nothing like this on the market. The others I've already told you, there's this one small stick of creativity. This is a painkiller, a very strong narcotic painkiller for patients in cancer pain. It's like a lollipop. This part is to be rolled. This is the tablet. Isko roll karo in the buccal cavity and the drug gets absorbed to the cheek. And the patient feels pain relief. It's self-administered. Moment he gets enough pain relief, he can remove it, keep the balanced dose for taking it again. So I think I have shared with you what I have learned through my journey. If there are any questions, I can take now or later.
No, we will have it now. Ketan, thank you so much. For each case, you did not hesitate in sharing the failures and the struggle that you have gone through, which makes your presentation far more credible and authentic, because many times the students who are struggling, most of them are students.
Yeah.
So they are struggling at the moment, and they get sometimes frustrated when the results do not come up to their expectation. And when they hear someone like you, who has reached a much higher level of market penetration, in at least 2 or 3 products, you are both leader in the country and in the globe.
Yes.
So their confidence, their ability to withstand failures will increase, I think.
Absolutely.
That's what I feel.
That
so anybody has any
question about that?
Yes, I think this is the one that I thought was very pivotal, because this is the one where you have used every failure
to stand up again.
Yeah,
And
whether B12, whether it was
and
the
for the continuous journey, this is what has kept me going.
yes,
रात को भी सोते सोते कभी एकदम ख्याल आ जाता है. So it comes from the heart and then it attacks.
right, right. And I must tell everybody that Ketan has a good laugh. So he laughs a lot when there is a problem. You know, and that's one way to keep your stress levels low. Because unless you laugh loudly and take away the stress out, how do you deal with the ups and downs in the life? I mean, there are moments when we don't find things to our liking.
बिल्कुल सर, ये आपकी ऑब्जर्वेशन बहुत अच्छी है सर. थैंक यू प्रोफेसर गुप्ता. दैट्स सो इम्पोर्टेंट इन द जर्नी ऑफ इनोवेशन टू बी आल्सो हैप्पी एंड लाफिंग.
Yes, Chaku, you are actually Chaku is Ketan Chaku is Professor Chakrabarti. DK Chakrabarti is from IDC Industrial Design Center, IIT Bombay.
Oh wow!
And he shared in his presentation how he failed repeatedly in a design, and he mentioned कि मुझे बहुत मारा लोगों ने, बहुत शर्मिंदा किया, और मैं शर्म से जमीन में गड़ गया, लेकिन फिर मैंने छोड़ा नहीं हिम्मत जब तक मैंने उनको अपना डिजाइन स्वीकार नहीं करा लिया. So this kind of admission helps the young startups to realize यार बड़े-बड़े प्रोफेसर, बड़े-बड़े एंटरप्रेन्योर्स भी फेल होते हैं जिंदगी में, तो हम हो गए तो क्या हो गया, फिर दोबारा कोशिश करते हैं ना.
नहीं, आप तो आज मुझे तो एंड प्रोडक्ट देख रहे हो ना.
मैंने देखा है तुमको चलते हुए.
आपने देखा है कि क्या थे.
And I've also seen you when your anxiety level goes up, तुम्हारा अस्थमा भी बढ़ जाता है ना. तो वो मैंने देखा है कि sometime when you are under a lot of stress that helps, but most of the time you have maintained yourself very light-heartedly. And I think that is one personality trait I thought I wanted to bring out, that you carry yourself lightly, और उससे बड़ा फर्क पड़ता है. बहुत सारी प्रॉब्लम जिंदगी में सहज होकर खत्म हो जाती हैं, कम हो जाती हैं.
True.
So anybody would like to raise an issue, any of the GYTI awardees who are online, and also if somebody is on the chat, they can point out. Yes, go ahead.
Sir, I need a suggestion from sir. So may I proceed?
Go ahead, Shivraj.
Sir, I am Shivraj Naik from ICT Mumbai, and I am also working in formulation development. So what would you suggest to PhD scholars like us who are developing new products with respect to
अपने बारे में बताओ, थोड़ा अपने प्रोडक्ट के बारे में बताओ, एक दो लाइन.
Okay sir, sure sir. So sir, here in our lab we have developed intranasal spray formulation for the treatment of Alzheimer's disease, and in your presentation you have shown the naso product for B12.
Yes.
So sir, what would you suggest to us with respect to regulatory considerations, how we should proceed with our product for various filings or regulatory issues?
That's a very, very pertinent question, which used to bother me also at one time a lot. So let me tell you one thing. First and foremost, you or the company where you work, or if it's your own company, you need to decide which is the regulatory market you want to get into. Because suppose you want to get into the US with a nasal, though it needs tremendous tenacity and a very large amount of money to reach and ultimately give birth to your product. That's the way US is. The same goes for Europe actually. If you want to do it for India and rest of the world, then the regulatory pathway is much less complex. And I would not say that the regulatory pathway of US gives maybe say 200% superior product than ours. No. Dynapar AQ आज भी US में approve हो सकता है, but US को narcotics use करना है. Now, of course, there is a opioid kind of epidemic in the US. I don't know how many of you are you aware of it? There is a opioid epidemic in the US. वो एक company जो उनकी थी, मैं नाम भूल गया. They have
Purdue
Pharmaceuticals and that oxycodone या
Oxycontin sir.
इतना खिला दिया है जैसे हम यहाँ पैरासिटामोल खाते हैं. तो एक तो मेरा ये है कि I really don't admire the US much. And secondly, the needs of the US are at a level of luxury. India में costs also matter. Rest of the world costs matter. Even Europe, the costs matter. Just check out how many products are approved in the US which are approved and which are being used and approved in EU. No, because in Europe the governments pay for the medicines. In US it's the insurance racket. तो जो दवाई 30 रुपए की बन सकती है उसका $300 लगा सकते हैं ये लोग. There is a product called Pensade. Check it on the net. Pensade is a topical painkiller which is in competition with my Dynapar QPS. Pensade penetration is 40% lesser than Dynapar QPS. But a 60 ml bottle costs $280. और यहाँ हम 250 रुपये में Dynapar क्यों? So where do you want to go sell your innovation? That is one very important thing. That will then enable you to decide about the regulatory roadmap. And it is not that कि आप पहले से ही यूरोप का यूएस का डेवलप करो. हम लोग आज today my files of Dynapar AQ are submitted in the EU, and very soon we are also going to submit Dynapar QPS in the EU. So we have to go stage wise. कि भाई एक के बाद एक करेंगे, तब तक rest of the world में हम बेच रहे हैं. So if you are doing, another thing is what are the disease you are choosing. Alzheimer is a big thing for US and Westerners. For us who consume so many spices or whatever may be the reason, the turmeric, Alzheimer and dementia बहुत कम है इंडिया में.
Yes sir, yes sir.
So मैंने भी एक Alzheimer का बनाया था बीच में, not a nasal spray, but a long acting formulation applied topically kind of a thing. But then I realized कि किसको बेचेंगे ये? यहाँ पे तो 90 साल तक तो किसी को Alzheimer होता नहीं है. और 90 के बाद कोई जीता नहीं.
सही है, सही है. बहुत ज्यादा याददाश्त रहती है लोगों की बात.
हाँ, हमारा in any case, I have traveled more than 90 countries. The most intelligent race on the planet is Indians. By a very wide margin. Intuitivity का तो कोई मतलब there is no comparison. So I would answer it that way, that you choose your disease as well. Unmet need को पकड़ो. Unmet need, and are you going to innovate for India or for the West? वो भी एक decide कर लो.
Okay sir. Okay sir.
Right?
Okay. Thank you sir. Actually your session was very inspiring for us, especially for formulation point of view, sir.
Yes, my whole journey has been in formulations. It is also a quirk of destiny. I became a medicinal chemistry guy. Had a fantastic thesis on synthesis of new compounds, new chemical entities.
Yes,
But
yes sir.
luck and journey of destiny, it took me into formulations. I didn't do my master's in pharmaceutical technology. तो फिर भी
Okay,
technology सीख लिया.
Anybody else?
Thank you, sir. Thank you.
Yes, sir. Yes, sir. Good evening, sir. This is Vaibhav.
Vaibhav, go ahead.
Yeah, I am from the graduated from Naipur, Ahmedabad, sir. I have a startup here, IOTA Design in Ahmedabad. So just this is a wonderful talk and motivation, sir. I just need one input from you.
Yes.
So, sir, like you have displayed that product you have developed in your Trica Pharmaceuticals. So, like the like this kind of the innovation as happening in some institute also, like when you're seeing that the Naipur or the small pharmaceutical institute. So why these are not translated in industry? Is there any barrier? Because you are also chairperson in Naipur. Is there any policy issue?
Yes, there's no policy issue.
Yeah.
There is a very big issue of incomplete work.
Okay.
I am giving you an open challenge in front of all of these people. मेरे पास एक project ऐसा complete करके मुझे दिखाओ कि as a research scientist, if I ask you questions, you'll have all the answers. Next day, I'll take it up for marketing.
Okay, okay. Because you are saying that in Naipur, Ahmedabad, there are 100 to 200 people, students are working in a different project, and they having had high potential to translate.
Potential is one thing, and realizing the I don't know whether you know about it. थोड़ा derail हो गया COVID की वजह से मेरा tenure. But when I became the chairman, the first thing I had introduced was that in every board meeting, I want someone who is working on translational research to make a presentation कि भाई मैंने ये embark किया हुआ है और ये commercial इसका ये potential है. आपको शायद मालूम होगा कि वो एक ocular वो dry eyes के लिए एक product कोई develop कर रहा है आपके वहाँ से. हाँ, उनका एक presentation हुआ. उसके बाद मैंने बोला था कि भाई आपका progress भी होता है तो I would be very keen to know. आपको कोई brainstorming भी करनी है तो मुझे बताना. आपको कोई doubts है तो बताना. I myself checked the products which are there on the market for dry eyes, and I told him that these are the products. Maybe you could even think ahead of these products, because when you are innovating, actually, ये मेरा जो Dynapar AQ 1 ml था, when I was innovating from 3 ml, I saw on the market 2 ml.
Okay.
तो मैंने सोचा मैं 1 ml बनाता हूँ. तो इस तरह मैंने उनको बोला था. उसके बाद क्या हुआ मुझे पता नहीं है, कोविड भी आ गया है. But there has been no formal request for translation research on the board. This board meeting again is going to be there soon on 25th or 20th. Again, I'm going to re-emphasize it. चलो है तो आप लेके आओ कि भाई ये मैं बना रहा हूँ और ये इसका मेरा ये लॉजिक है, ये अनमेट नीड है और इसलिए इसकी वजह से ये होगा. होगा नहीं होगा मुझे उसकी चिंता नहीं है, बट कुछ प्रूफ ऑफ कॉन्सेप्ट तो हो.
Yes, sure. Right now, I'm graduated from NIPA.
No, you graduated, but whenever in life you do something, see, we are in the healthcare business. Let me tell you, our innovation challenges are with a huge multiplier as compared to anything in engineering and any other domain. We have to ensure safety in humans. We have to get proof of concept in humans. Read about the company called Theranos, USA.
Yeah, I know that.
Yeah, 1 drop of blood and 600 tests and 200 tests and all.
Yeah, I know
She's
that.
facing conviction.
Yes.
तो हमारा इतना बड़ा चैलेंज अगर है तो हमको इतना तो मेटिकुलस एंड रिस्पॉन्सिबल रहना पड़ेगा दैट वी हैव टू प्रूव दैट मैंने जो सोचा है सही है, वो जो अक्षय जी ने सोचा है वो तो सही है मेरे हिसाब से. अब आगे वो कितना जा सकते हैं उनकी ताकत होनी चाहिए. बाकी डू यू थिंक एन इंडस्ट्रियलाइज लाइक मी हु हैज टेकन सो मेनी रिस्क्स, यार आई हैव अवॉइडेड रिवर्स इंजीनियरिंग, मैं क्यों नहीं लूंगा? मैं आई गेट सो मेनी लेटर्स फ्रॉम डिफरेंट कॉलेजेस एंड स्टूडेंट्स, मेरा ये प्रोजेक्ट है, पढ़ते हैं देखते तो देयर हाफ ऑफ इट इज इरेशनल. अनदर थिंग आई ऑल एडवाइस फॉर्मुलेटर्स इज फार्माकोलॉजी पढ़िए यार. आप खाली फार्मास्यूटिकल टेक्नोलॉजी में चढ़ गए तो आप फार्माकोलॉजी नहीं पढ़ोगे तो नहीं बनेगा सही.
Yes, sure.
बिकॉज इट्स इंटरलिंक्ड. फार्मास्यूटिकल टेक्नोलॉजी के बाद आता है उसका रिजल्ट तो फार्माकोकाइनेटिक्स में आएगा, क्लिनिकल ट्रायल्स में आएगा. सो इंटरडिसिप्लिनरी नॉलेज ऑल्सो शुड बी गेन. ठीक है. सो मेरा चैलेंज है नॉट ओनली टू यू, टू एवरीबडी, लाइए कोई ऐसा प्रोजेक्ट जो वायबल है तो हम इमीडिएट ऊपर उठाएंगे इसको.
Yes, sure.
Thank you. Anybody else? Professor Anil Wali is still with us. So Anil, would you like to suggest anything, any immediate thought that comes to your mind after listening to Ketan, which you would like to share? Anil heads the FIIT, one of the oldest, one of the largest, one of the most helpful technology incubation institution in IT Delhi. Anil, just any reaction?
Thank you, sir. I think I have had the pleasure of interacting with Ketan Bhai early on.
Correct.
I must compliment him for deciding to actually work out one of our iontophoresis technology.
Yes.
Just didn't work out, but I think I have to compliment him for
Anil, I am very sorry.
Academic research.
Let me add to that. This you triggered a memory for me, and which is very much linked to the immediate preceding question. I am a Patel, same village as Sardar Patel. So बोले बिना मैं नहीं रह पाऊंगा. You know, when that we paid the royalty, I met you. You were much younger then. So and we signed, and I was thrilled actually because I was looking up for iontophoresis from the US. And then I read this thesis of some student of yours, आपकी जो biotech की जो biomedical device की जो मैडम थी कोई,
Veena Kaul जी. तो कॉल मैडम ने हमको बताया, शी वाज आल्सो एक्साइटेड कि यार फाइनली हमने कुछ बनाया तो जाएगा इंडस्ट्री में. फिर जो प्रोटोटाइप आए थे, अनिल जी आपको शायद मालूम होगा, याद है, मुझे नहीं मालूम है, उसमें एलईडी इंडिकेटर नहीं था, ऑन कहाँ से होता है ऑफ कहाँ से होता है. तो पहले वो ड्रग जा नहीं रहा था स्किन में से जो जाना चाहिए एक्चुअली. तो हमको ये लगा कि भाई हम शायद गलत करें, पता नहीं, तो एक बार एलईडी तो डलवा दे. हमने लेटर डाला आपको कि भाई एलईडी डालो. आपने बोला 2.5 लाख और लगेगा. मैंने बोला यार कोई डिवाइस एलईडी के बिना आपने दे दी तो हम तो थोड़े कोई इंजीनियर है, हमको पता होना चाहिए. अच्छा, फिर एलईडी बना के भेजी आपने डिवाइस. फिर हमने रन किया, नहीं गया ड्रग अंदर. ड्रग आई वाज ट्राइंग टू पुश ड्रग्स थ्रू द स्किन. दिस इज अबाउट 10 years ago. Absolutely, yes.
ये अगर वर्कआउट हुआ होता तो इट कुड हैव. अच्छा, बाद में आप लोग ने जवाब मेरे को दिया, वीना जी ने, कि हमारा तो भाई सही है, आपका फॉर्मुलेशन गलत होगा. ठीक है भाई, हमारा फॉर्मुलेशन गलत होगा. मैंने एक डिवाइस इंपोर्ट किया यूएस से 3.5 लाख रुपए में, अभी पड़ा है कहीं सड़ रहा होगा. उसमें मेरा सोल्यूशन गया अंदर. तो ये होता है व्हेन यू डोंट गिव अस अ फिनिश प्रोडक्ट. तो आई एम लाइक अ पर्सन हु हैज बर्न्ट माय लिप्स विथ हॉट मिल्क. तो अभी मैं छाछ फूंक के पीता हूँ फिर.
Absolutely. So this is the lesson for all this is that yes, there is many a step between cup and the lip. You are very right. And all technologies, unless they reach a certain threshold, जिसको की भाषा में भी बोलते हैं.
आपने दिया था ना मुझे. Sorry, I missed your name. It's
Anil Wali. Dr. Anil Wali.
He's also Anil, ना?
जी.
हाँ, मेरे को इजी रहेगा. So Anil जी, if madam had showed me the willingness to walk with me ना, तो भी मुझे प्रॉब्लम नहीं था. बट आप बोल देते हो आपका सोल्यूशन गलत है. फिर हमने लेटर डाला कि भाई इंपोर्टर से जा रहा है, साइलेंस. मेरा तो लाइसेंस फी गया, कोई बात नहीं उसका मुझे.
बट दुख ये हुआ कि IIT दिल्ली में अगर ये होता है तो ये नाइपर वाले बच्चे मुझे पूछ रहे हैं कि भाई आप क्यों कुछ करते नहीं हो, क्या करें भाई। इंटेग्रिटी तो रिसर्चर के हेड की होनी चाहिए। वो मैडम को इतना सेल्फ एस्टीम, अगेन ऐसे खुददारी इतनी होनी चाहिए कि मैंने कुछ बनाया है तो कमर्शियली क्यों नहीं बनेगा, या फिर आप क्लासरूम के लिए ही बनाते हो।
It was actually a very good lesson. It was a very good lesson, but I think subsequent उसके बाद जो टेक्नोलॉजी हुई हमारी, एक शायद आप जानते होंगे कंपनी है जिसने कि वो हीमोग्लोबिनोमीटर बनाया, वो which is doing very well I think in the market.
No, I am not saying, see वो even
but ये है कि it was a good learning experience both for
you. बन गया होता, लेकिन आज Indians would have been beneficiaries of a very wonderful device which could have added so many ideas on that.
But Anil, I think what the point is that वो जो साथ चलने वाली बात है ना जर्नी में, वो जरूरी है. Researcher will also learn something, industry will also, give and take, साथ co-learning, co-creation होता है यार. ये फीडबैक आया, उसको फर्दर काम कराओ, एक स्टूडेंट लगा दो, फिर उसको बोलो तुम जा उनके पास, उनकी फैक्ट्री में जा और जाके देख वहां पर कि कहां पर दिक्कत आ रही है, फ्लो क्यों नहीं हो रहा है. एग्जैक्टली यहाँ समझे आकर, यहाँ से फिर वापस जाए दिल्ली और फिर वहां जाके बताएगा मैडम को, मैडम हमारा ना यहाँ पर कहीं ब्लॉकेज है, यहाँ पर
ब्लॉकेज है. एग्जैक्टली
वो ठीक करेंगे तो प्रॉब्लम सॉल्व हो जाएगी. मतलब मुझे कहना ये है कि ये को क्रिएशन का जमाना है, इसमें हम अकेले नहीं कर सकते काम, ना इंडस्ट्री अकेले कर सकती है, ना
ओपन इनोवेशन. जो केतन भाई ने बोला ओपन इनोवेशन इज, आई एंटायरली एग्री विद हिम.
अभी भी अनिल भाई कोई नया आ गया होगा अब तो आपके बायोमेडिकल में, तो कोई एंटरप्राइज बनाता है वापस शुरू करते हैं, मैं तो बोलता हूँ.
Look at this, I mean he's still open.
Yeah, absolutely. So this is actually the reason I triggered this is because I actually liked this his spirit at that time also.
Very nice.
मैं अनिल वही बोल रहा हूँ, ये 15 साल 12-13 साल के पहले की बात कर रहा हूँ जब मेरी कंपनी बहुत छोटी थी. I have grown by quantum, you know that. तो धीरे-धीरे नहीं बढ़ा, एकदम बड़ा हूँ. तो पहले तो 10 साल पहले तो कुछ नहीं था. Turnover was hardly possibly 150 crores.
Maybe, but certainly I will be certainly presenting before you or maybe suggesting to you some more things which you can have a look at. And
अभी वीडियो कॉन्फ्रेंसिंग हो गया, बहुत आसान है. आप मेरा ईमेल ले लेना, अनिल से मुझे भेजना. It will be a pleasure. I am always open to getting new things. बिकॉज एक फियर्स पेट्रियोटिज्म का स्ट्रीक है मेरा. देखो मेरा रिसर्च आप देखो सारा नेजो B12 अमेरिका में नहीं चलेगा, कोई बात नहीं, मेरे देश में बहुत जरूरत है इसकी.
इंडिया इज अ बिग मार्केट.
इंडिया इज अ बिग मार्केट एंड वी हैव डिस्टिंक्ट नीड्स. ये जो न्यूरल ट्यूब डिफेक्ट है अमेरिका यूरोप में कहीं है ही नहीं. हमारे देश में इतना एम्बेरसमेंट हो रहा है, और ये अपने प्राइम मिनिस्टर इसीलिए बोल रहे हैं पोषण अभियान. क्या करो पोषण अभियान से, B12 नहीं आने वाला है कहीं से.
सही कह रहे हो, सही कह रहे हो. Deval, you had a question, the last question. Deval?
Yes, sir. Can you hear me?
Please, go ahead.
Yes, sir. I have a slightly unrelated question. So, since sir has been in the industry from so long and started almost from scratch for the pharmacy side, I wanted to understand his perspective on the medical device side, if he has any on taking an innovation to market. Particularly a device innovation, if there's any perspective from the industry that he has.
Yes, medical device, again, there's an immense scope. Once again, the needs of medical devices in India can also be very different from the medical devices that are available in the West. So an ideal situation would be where you take feedback, you meet surgeons who use different things during surgery, ask them, find the gaps, or you even re-engineer and reinvent the things which are successfully sold by companies like Siemens and large companies at Johnson & Johnson, exorbitant prices.
You know, I abandoned Johnson & Johnson's glucometer in favor of Vito, which had a small device with a microfluidic chip and attached to your phone. So it kept record of my observations, it gave me the trend analysis. And sometime when they were the observation high, the doctor called up from the company, sir, आज की रीडिंग आपकी हाई थी, क्या बात है, कुछ आपको कंसल्टेशन करना है क्या? इंडियन स्टार्टअप.
हाँ, वही
मैंने जॉनसन को छोड़ दिया.
नहीं, वही है कि ये जो इनका क्वेश्चन बहुत वैलिड है. आप बना सकते हो, आप कंपनी मार्कआउट करिए, आप प्रोडक्ट्स मार्कआउट करिए जो आपके विद इन रीच हो, भले आप लो लेवल से स्टार्ट करिए, बट प्रोडक्ट इतना अच्छा बनाइए कि धीरे धीरे, ओवरनाइट कुछ नहीं होगा. देखो ये ब्लॉकबस्टर टेक्नोलॉजी वो सब यूएस सिलिकॉन वैली में होती है, क्योंकि वहां पे $100 मिलियन $200 मिलियन इन्वेस्ट करने वाले, आइडिया पे इन्वेस्ट करने वाले. सो इट में टेक
So if you come out with a good product, then slowly acceptance badegi, naam badega, and then you can keep on growing. You identify the product first, which is within your reach and which you think you can spread and survive on that basis. मैंने वही किया था ना, पहले एंटीडोट लिया था, हिंदुस्तान में कोई बनाता ही नहीं था एंटीडोट.
Exactly, unmet need ले लिया.
Unmet need लेके आ गए.
Ketan, if you have one more minute, I will take one more question from Nitin, and then we will wind up. Nitin, go ahead.
Hi, sir. A very interesting session. I had a question about patent. Any patent that gets filed in India takes at least 2 years. US patent is like 4 years, a lot of money. Like for a startup, it's a huge amount of money. So how do you as a business do that? Like the moment you file a patent, it takes 5 years for that to mature. So is it that the same way that your company is doing, or?
What is the question?
पेटेंट में जो टाइम लगता है,
हाँ, वो तो एब्सर्ड है. वो तो ये अनिल कुछ कर सकता है तो ही हो सकता है.
नहीं, लेकिन उसका एक तरीका है. हमारे एक इनोवेटर ने, नहीं, मैं बताता हूँ. एक जहांगीर है, गणेश जहांगीर, जयपुर में, उसने 1 साल और 3 महीने के अंदर पेटेंट ग्रांट करवाया. कैसे कराया? उसने पेटेंट फाइल किया, अर्ली रिक्वेस्ट फॉर एग्जामिनेशन फाइल कर दी, फॉरेन फीस पे करके अर्ली एग्जामिनेशन करा दिया. अर्ली एग्जाम अपने इंडियन पेटेंट ऑफिस में.
तो उससे क्या होता है?
तो अर्ली एग्जामिनेशन से आपकी एग्जामिनेशन रिपोर्ट जब आ गई, यू कैन से अर्ली पब्लिकेशन. तो आपने 18 महीने तक वेट नहीं किया, आपने अपनी एप्लीकेशन जो है जल्दी प्रिंट एप्लीकेशन को पब्लिश करा दिया. अब वो पब्लिश कराने के बाद जो टाइम है 1 साल का, जो ऑब्जेक्शन मंगवाते हैं जिसके अंदर ओपन, वो टाइम तो लगेगा, लेकिन बाकी जो टाइम 18 महीने का था वो बच गया. तो मतलब कि there are mechanism by which you can speed up.
There is no mechanism, Anil. I'll tell you what. You are not reaching the final stage. ये अर्ली एग्जामिनेशन सब हो गया. मेरा एक अर्ली एग्जामिनेशन होने के बाद 3 साल से पड़ा है, awaiting an order.
ये तो गलत बात है.
It is all at the whim and fancy of the examining officer. If you get a good examining officer, he will process it and he'll grant it fast to you. डायनापेरिक क्यों हमारा बड़ा जल्दी ग्रांट हो गया था, QPS बड़ा जल्दी ग्रांट हो गया था. पीछे 3-4 पेटेंट ऐसे पड़े हुए हैं. अच्छा, पहली बार एक तो इन्होंने एग्जामिनेशन करके हमको रिपोर्ट दिया रिजेक्ट करते हुए पेटेंट को, उसमें सारे प्रायर इंजीनियरिंग के लिख दिए थे. बताइए. अब
I think there is
एग्जामिनेशन रिपोर्ट में तो सारे फार्मा के प्रायर आर्ट थे, वो भी इरेलेवेंट थे सारे. बट दोबारा जब रिजेक्शन ऑर्डर दिया तो बेसिस बनाया सारे इंजीनियरिंग. फिर हमने उनको लिखा कि ये क्या है भाई, इंजीनियरिंग कहाँ से आ गया इसके अंदर. तो फिर गलती तो मानते नहीं है ये लोग, तो बोलते नहीं आप दोबारा हाजिर होइए. हम दोबारा हाजिर हो क्या है, उसको भी 2 साल हो गए आज. ये तो मैं भगवान पे छोड़ दिया है. ऑल्सो कॉन्टेक्टेड मिस्टर रत्नू एट दिल्ली, हु इज एक्चुअली द IAS ऑफिसर. बट ही बीइंग IAS, ही कैन नॉट टेल एनीथिंग टू दिस इंटेलेक्चुअल्स जो पेटेंट प्रोसेस वाले हैं. ये मरने का ही धंधा है भाई. आपका नसीब जोर करे तो करना, इनोवेशन करना है, नहीं तो ये सब रिवॉर्ड्स मत एक्सपेक्ट करना. पेटेंट होने के बाद लोग इन्फ्रिंज करने वाले. आज हमारे डायनापार QPS के 2-3 क्लोन ऑलरेडी आ गए. बट गुड थिंग इज के मेरा सेल फिर भी, बट आई सेड व्हेन यू इनोवेट यू लीड. वो पीछे जो आएगा वो मीटू ही आने वाला है. A big company introduced a copycat of Dynapar QPS, Anil. Pan India, big blast. 3 months there to withdraw. इतना फेल हो गया. बिकॉज मेरा पेटेंट को इन्फ्रिंज नहीं करना था, ट्वीक किया था फॉर्मूला. अब ट्वीक करते तो उनको तो पता नहीं होता कि ट्वीक कर दिया तो उसका पेनिट्रेशन क्रैश हो गया स्किन में से.
सही है.
तो अभी नेजो बिटवेल में भी एक कंपनी आ गई ऑलरेडी, बट हमने उनका मेजर कर लिया है. इट इज अनकंपेरेबली पुअर एब्सॉर्प्शन. ये क्या है, हमारा रेगुलेटर्स आर नॉट सो एक्सपर्ट. सो थोड़ा बहुत साइंस कोई झाड़ देता है तो उसको मिल जाता है प्रोडक्ट परमिशन. So don't seek the reward of protection. Otherwise, तो Troika today would have been 5,000 crore company भैया, only on innovation. But हमारा चोरी rampant हो रहा है. Satisfaction ये है कि we are the pioneers. People respect us, doctors respect us, and we are making good enough money here. Plus, we are serving the country with top of the line products at very reasonable prices. So don't सब कुछ देखोगे ना तो कभी innovation कर नहीं पाओगे आप. ये be prepared for all this nonsense.
It almost amounts to open innovation.
It is like an open innovation. ऐसा मान के चलो.
Like an open innovation where you survive on the quality not only of innovation but of productization. मतलब manufacturing जो efficiency है, manufacturing जो excellence है, quality control systems जो हैं, वो equally important है आपके.
और दूसरी एक बात है अनिल कि आपका इनोवेशन जब सक्सेसफुल हो जाता है तभी कॉपी करने लोग आते हैं। शुरू में जब चलता है तो कोई
नकल तो उसी की करेंगे ना जो अच्छी चीज होगी।
अब जब सक्सेसफुल हो गया तो आप तो लीडर बन गए हो, उसके बाद चिंता नहीं है।
सही बात। And there are people who would still prefer your brand. Very
yes,
seriously. There are. Brand formation also takes time, but it is a very valuable tool for building your
reputation. में आज more than 50-60 companies are there. फिर भी मेरा 65-70% मार्केट शेयर है, ग्रोथ भी आ रहा है, एंड बाकी जो बेच रहे मैं इतना समझ रहा हूँ कि भी वो 3 ml से सबका मसल डैमेज हो रहा था, वो नहीं हो रहा है यार। चलो इंडिया को तो फायदा हुआ।
हाँ ठीक है, ठीक है। That's a large
इनोवेशन के सिखाया हमने लोगों को भी मसल डैमेज बंद करिए।
सही बात। I think this is the kind of one last question I have taken is that there are certain lessons both in manufacturing and in formulation which you can put under open source guidelines. मतलब कुछ ऐसी चीजें होंगी, सबक होंगे जो आप ओपनली शेयर कर सकते हो। तो मेरा एक आजकल सबसे कहना है सभी एंटरप्रेन्योर से कि क्रिएट सम नॉलेज पब्लिक गुड्स। अपनी साइट पर कुछ चीजें ऐसी डालो, भाई देखो ये मैं लेसन शेयर कर रहा हूँ, आप सबके काम आएगा। अब उसमें क्या होता है कि only leaders can do that, Ketan. Only leaders can.
I know.
सब लोग नहीं करेंगे।
I fully agree with you, but and in fact when I was reading all these books which you had sent to me, I was thinking कि यार ये अनिल कितना काम करता है यार।
नहीं मैंने नहीं किया।
Honestly, I'm telling you, honestly, हमारा प्रॉब्लम ये होता है कि हम लिखने को नहीं बैठ सकते।
नहीं, लेकिन थोड़ा सा चाहे स्टूडेंट्स को इंटर्न ले लो, वो कर देंगे काम।
ठीक है,
take interns.
I will suggest कि you being such a kind of a, you are a stirrer of the hornet's nest. You arrange these kind of question answer sessions only. They can be recorded, and that will be the lesson actually for everyone.
Correct. That also is a good way. That's also once we will have only pharma kinetics के जो students हैं, pharmaceutics के जो students हैं, उन्हीं को ले लेंगे साथ में।
Innovation तो क्या है कि it is a generic word. Engineering वाले भी आएंगे, कोई फर्क नहीं पड़ेगा।
ठीक है, ठीक है।
So if there is a question answer, I think that's why that other day.
That helps.
कि भी मेरा speech मैंने 10 मिनट का ही कर दिया था because I wanted and see how many questions came up.
No, you got enough. I mean, now I have to curtail it, but there are, but then I can already see demand for that session. Already some people are putting on the chat, and I'm very happy that we will plan, Shivraj, that session. Nitin, we will plan a session for all the GYTI awardees and others who can join from other platforms. I'll ask Anil also. I'll ask Chakku also that if there are similar student startups at IT Bombay, IT Delhi, ISC, C Camp Morning में भी हम लोग के पास तस्लीम आरिफ था C Camp से, तो हम उनको भी साथ में ले लेंगे। इसमें जरूरी नहीं खाली GYTI से रेस्ट्रिक्ट करना है, बिग ग्रांट्स को भी ले लेंगे। But it will be nice to have some of your friends also with you, so that आप आपस में आर्गू भी करो थोड़ा सा, तो मजा आएगा बच्चों को सीखने में।
Correct.
हाँ, multiple viewpoints आएंगे ना थोड़ा सा।
Because when we speak from our perception. When they ask for what they don't know. So
correct.
We can't know what they don't know.
That's.
And I would also request the number of contact of Chakravarti ji because.
हाँ, I will. चक्कू जरा अपना नंबर डाल दो और email डाल दो chat में। वैसे मैं भेज तो दूंगा इनको। I will connect you.
ठीक।
No problem. He's a great designer, and if you ever would.
Exactly. Design is my passion, so I love.
तो वो मजा आएगा आपको।
इसलिए।
चक्कू, are you there?
Yeah, he's mute, I think.
अच्छा।
I can see him. Yeah, I can see his name. So.
So he will. I mean, I'll connect you both. So, Ketan, thank you so much for being with us. I thank all the GYTI participants. I thank all the faculty members, Anil Wali, and all others who joined us today. Dr. Vipin, Dr. Deepanvita, and Madhusudan from CCNB. All of you who found time for us today it was very kind of you to take the time off. Dr. Vandana Patrawale and Tasleem from C Camp. Dr. Vandana shared a beautiful story how one of our products is getting in 55 countries and how she has got, including US. So I think, and she has had several of our students were talking to you today. They are from her lab, ICT Bombay, Institute of Chemical Technology. So she's head of the pharmaceutics and has one of the very, very helpful faculty members. So I think the personality of the student is partly shaped by the faculty members,
Absolutely.
If they are generous, if they are kind, if they are open, then the students imbibe some of those values.
Yes.
And I think that's very nice. So thank you so much, Ketan, for being with us today. You have really stirred the imaginative part of everybody. And they have got inspired by many things that you have shared very candidly, very authentically, very boldly, and very frankly. And I think that is a very useful way of building capacity of the people. You're not sermonizing, you're not giving them only advice, you're also telling them, भाई मेरे से भी गलती होती है, मेरे से भी गलती होती है, आपने भी ये कहा आज, मेरे से भी गलती हुई है. मतलब जो स्ट्रगल होती है वो बच्चों को अगर ये महसूस हो जाए कि यार हमसे बड़े बड़े विद्वान हैं, उनको भी दिक्कत आती है, संघर्ष करना पड़ता है, तो शायद उनकी हिम्मत बढ़ेगी थोड़ी सी. और एक कल्चर जो बननी चाहिए कोलैबोरेशन की, क्रिएटिविटी की, सॉलिडेरिटी की, वो कल्चर बहुत जरूरी है. हम अकेले अकेले नहीं जा पाएंगे आगे. सो द नेटवर्क एंटरप्रेन्योरशिप की जो बात करता हूँ मैं आजकल ज्यादा, वो जरूरी है कि हम मिलके काम करें. So thank you so much, Ketan, for being with us. Thank you all the people, all the colleagues of GYTI Concalve. Our team has worked very hard to bring you all together. Chetan, Richa, and everybody else. भाई नाम बताओ अपने सब बच्चों. यश, हाँ, दिग्विजय ने रात दिन काम किया है और आप सबको फोन करता रहा, वो सबको बुलाता रहा. उसको डांट पड़ती थी बीच बीच में कभी कभी, लेकिन उसने फिर भी हिम्मत नहीं हारी. और सब लोग अभी अपना स्क्रीन ऑन कर लो भाई सारी GYTI team, please come on the screen so that Ketan can see you all. This is please all team members, हमारी team member को बोलो सब लोग आ जाएं सामने. तो चेतन, everybody should be, हाँ सब लोग आ जाओ. So ये I want to thank all of them. ये सबको एक साथ लाना, नितिन, श्वेता, all of you, Deval, Jayesh, and of course Ketan and Chakku and all of you to bring together. ये छोटे से बच्चे हैं हमारे जिन्होंने रात दिन काम किया और आप सबको एक साथ लाए. I'm grateful to them and to you all for being with us. It was such a learning value. Ketan, I'm really impressed. I have to update myself now. There are a lot of things that you have done lately, which I had to spend time and listen to your story.
Yes, once we
and thank you, Shweta. Thank you very much. Shweta, spread the word around. Keep your culture of cooperation intact. We need more Shwetas who will help others to solve their problem. हमारी दिक्कत हल हो ना हो, दूसरों की हल होनी चाहिए. हम आगे बढ़े ना बढ़े, दूसरों को आगे बढ़ाना चाहिए. और जब हम दूसरों को बढ़ाएंगे आगे, तो खुद भी बढ़ेंगे आगे. ऐसा कोई नहीं है जिसने खुद आगे तरक्की की और दूसरों को नहीं बढ़ाया. ऐसा नहीं होता. समाज हमेशा उन्हीं को आगे बढ़ाता है जो दूसरों को भी आगे बढ़ाते हैं. So thank you so much. God bless you. Keep in touch. Update your profile with us so that our platform becomes new. As I mentioned, I will conclude on mentioning that we are the largest provider of open source content in the world. We have techpedia.in platform, which has 200,000 engineering project information, engineering and pharmaceutical science and life sciences project information. केतन कभी देखना बैठ के उसको.
हाँ, मैं अभी मैंने रखा
उसको देखना. दूसरा हमारा प्लेटफॉर्म हमने बनाया है 0.9 मिलियन abundant US पेटेंट का डेटाबेस बना दिया. केतन, कैन इमेजिन? जो abundant हुए, मतलब जो ग्रांट हुए थे और रिन्यू नहीं किए गए, वो तो ओपन सोर्स होगा ना अब.
मैं वही बोल रहा हूँ कि काम कितना करते हो यार.
उसको यूज करो. I want you to use this database and develop some products out of that knowledge. तो उनको नहीं दिखी, लेकिन हम लोग उसको इस्तेमाल कर लेंगे.
नहीं, अभी मेरे कंपनी में एक न्यू प्रोडक्ट का सेल बनाया है मैंने.
हाँ, तो उनको मैं एक बार उनसे बात कर लेता हूँ.
हाँ, तो उसको लगाएंगे काम पे, इसमें से ढूंढो क्या है.
बस. And few examples, and then you help us to maintain this platform. क्या होता है, तुम्हारे जो कमाओगे ना उसमें से थोड़ा सा लगा देना इसके अंदर, बस, ताकि we can prove that this platform helps solve real problems of our society and our country. क्योंकि ये abundant database का, can you imagine? US में नहीं था कोई abundant database का, abundant patent का database. किसी ने बनाया नहीं. और दो स्टूडेंट, देविका कौशल एंड जायगम खान, इन दोनों ने मिलकर बना दिया. So I'm very happy to all of you, and please keep in touch. Nitin, Deval, all of you, Chakravarti, Chakku, thank you so much. Anil, thank you so much. All of you, grateful to you. Thank you. Bye. Jai Hind. God bless you.
Thank you so much.